Very good morning, all. We will start our day two session of Startup in few minutes. I repeat myself. Very good morning, all. I welcome you all to the second day of Startup. We will start our conference in few minutes. Thank you.
यशा यशा दीप्ती दीप्तीला बोलो लवकर बाहेर.
Dad?
A very good morning, everyone. Before we start a session, I request all the participants to stay on mute and kindly turn on your cameras. Again, I request you all to kindly stay on mute and turn on your cameras. Okay. Very good morning, all. I welcome you all to the second day of StartHub Journey from Innovation to Entrepreneurs in Herbals, organized by Association of Pharmaceutical Research, APR, and DY Patil Deemed to be University School of Pharmacy, Navi Mumbai. I'm sure you all had great study with us and hope to see the same enthusiasm from all our presenters and attendees of today. Association of Pharmaceutical Research APR and DY Patil Deemed to be University School of Pharmacy, Navi Mumbai organizes the StartHub Journey from Innovation and Entrepreneurs in Herbals, which is scheduled to be held on 14th and 15th October 2022 in the live on Zoom. StartHub, a journey from innovation to entrepreneurships in Herbals, aims to bring together leading academic scientists, researchers, and research scholars to exchange and share their expertise, experiences, and research results on all aspects of medicinal and natural products. It also provides a premium interdisciplinary platform for researchers, practitioners, and educators to present and discuss the most recent innovations, trends, and concerns, as well as practical challenges encountered and solutions adopted in the field of natural medicine. This conference is multidisciplinary program with broad participation with members from around the globe focused on learning about pharmaceutical science, research, methodologies, formulations, manufacturing, and its advances. We invite all the speakers and delegates from all over the world to attend this conference. It creates a perfect podium for global networking as it brings together renowned speakers and scientists across the globe to most exciting and memorable scientific event filled with informative and interactive sessions, international workshops, world-class international exhibitions, and poster presentations. WIC Health Products. WIC Health Products are pleased to introduce themselves as pioneer in manufacturing of pure vegetarian sweet taste jellies, which are soft and easily chewable for all age groups with sugar as well as sugar-free, depending upon choice of the valuable clients with the motto Eat Medicine with Tasty and Enjoy. Dawa Kalo Khel Khel Mein. WIC products recent research on stevia proves that it is very useful in the treatment of various viral diseases. WIC products are FSCI certified small scale industry and currently working with many pharmaceutical companies. With most of the products are plant-based and natural. WIC products have come up with the formulation in 100% natural herbals for increasing immunity in the body to fight against viruses. All the products are included with ingredients like pectin, sucrose stevia, glucose stevia gel, citric acid, natural flavors, and food colors. They are fortified with multivitamin herbs, chyawanprash, curcumin, avla, highly enriched with vitamin C. WIC products works as third-party manufacturers for big pharma companies and can make any products in jellies as per customer requirements. The benefits of WIC healthy gummies improves digestion, acts as immune boosters, very nutritious and helps in weight management, rich in antioxidants, minerals, and DHA. They are also source of multivitamins which provides energy and improves skin, hair, and nail health. Now, I would like to welcome all our honorable dignitaries and eminent keynote speakers. Now I welcome Dr. Bhairav B. Kulkarni, Professor and HOD, Dr. Ved Prakash Patil, Ayurvedic Medical College and Research Institute, Jalna, India. Dr. Satnam Singh, Associate Professor, Pharmacology, Adesh Institute of Medical Sciences and Research, AIMSR, Adesh University, India. Dr. Chaya Gadgoli, Professor, Saraswati Vidya Bhavans, College of Pharmacy, India. Vaidya Dr. Piyush Juneja, Founder, India Vaidyas, India. Dr. Renu, Deputy Secretary, Department of Labor and Employment, Government of Haryana, India. Dr. Ved Priya Arya, Scientist E, PhD Division, Head, Herbal Research Division, Patanjali Research Institute, Haridwar, India. And session chairs of our day, Dr. Rukia Sultana, Professor and HOD Department of Pharmacognosy, Yenipoya Pharmacy College and Research Center, Mangalore. Dr. Giriraj D. Kulkarni, Professor and Principal, Department of Pharmaceutics, Gokaraju Rangaraju College of Pharmacy, Hyderabad, India. Dr. K. Lakshmi, Professor and Dean, Chettinadu School of Pharmaceutical Sciences, Chettinad Academy of Research and Education, India. Dr. Shailendra Gaurav, Professor, Goa College of Pharmacy, Goa University, India. And I would also take this opportunity to welcome all our presenters from across the globe who have spared their valuable time in enlightening us with their latest research studies. I would also like to welcome all the attendees in joining us today. Again, I welcome you all once again to Startup Journey from Innovations to Entrepreneurs in Herbals. I hope you will have a great day with us today. Now, I would like to welcome our keynote speakers of today. I would like to welcome our keynote speaker, Dr. Bhairav B. Kulkarni, Professor and HOD, Dr. Ved Prakash Patil, Ayurvedic Medical College and Research Institute, Jalna, India, to deliver his keynote speech. Sir, please.
Thank you. First of all, thank you, APR Pharmaceutical and Research, who invite me such a wonderful platform. And I also thank you, DY Patil University School of Pharmacy. So can I share my screen?
Yes, sir, sure. Hey, that driver pa.
Is my screen is visible?
Yes, sir, it's visible.
Okay. First of all, again, I thank you, all the keynote speaker who are with me. It's my pleasure with Vaidya Piyush Juneja sir, with Dr. Satnam Singh sir, Dr. Joban sir, Dr. Renu ma'am, and Dr. Sabu sir. Today is my topic. I given one topic on a clinical study of evaluate the efficacy of Krushnadi Churna in the management of Tamaka Shwasa with special reference to bronchial asthma. This is my topic. Myself, Dr. Bhairav Bhimrao Taushikar Kulkarni. I'm a BAMS MD in Kai Chikitsa Ayurveda. And I also working as a PhD scholar in DY Patil Ayurveda College. Also, I'm working as a professor and HOD in Kai Chikitsa Department, Jalna. This is some brief introduction about myself. I'm also director of Sri Siddhivinayak Ayurveda Panchakarma Center working in Aurangabad. Also, I was a former principal of 3 Ayurveda Medical College in the past. I also advisory board member of Hungarian Ayurveda Medical Foundation, Budapest, Hungary. Well, also working as a editorial member of 5 international journals, working with registered Ayurveda specialist RAS, North America, APNA, member of Yoga and Ayurveda. This is some brief introduction about myself. So let's start about my topic, today's topic. So I'm talking on asthma. And in Ayurveda, it is mentioned Tamaka Shwasa. So what is a Tamaka Shwas? Shwas. Shwasati Vayu iti Shwasaha. Mentioned in Ayurveda classics, Hemachandra, one of the Ayurveda Acharya. Shwasati anena iti Shwasa. Shwasati iti Shwasa. Therefore, Tamak Shwasa denotes a pathological state where a sense of darkness prevails due to movement of pranavayu in reserved diuretics. So, my dear friend, in our classics definition of Shwasa, from above reference, it is clear that the Shwas word is used to denote a respiration and extent of air in the body. The word Shwas is used for both physiological and pathological states. Shwasaroga may be defined simply as a disease, which is the respiration and exchange of the air disturbed. So, in our Ayurveda Sanhita, there are mentioned Shwasa. So, 5 types of Shwasa mentioned in Ayurveda classics. Tamak Shwasa, it is a mention on the variety among the 5 types of Shwasa. But out of these, Kshudra Shwasa present as symptoms in most of the diseases. And It does not require any medication, whereas the Mahashwasa, Urdhva Shwasa, and Chinna Shwasa were present in the terminal stages of various diseases. So Tamaka Shwasa, it's a Swatantra Vyadhi. It's one of the common disease and having its own etiology, pathology, and a management. It is mentioned as a yapya. It means chronic in a nature. So bronchial asthma mentioned in modern medicine also, you know very well, and it resembles with Tamaka Shwasa as mentioned in Ayurveda. So these are some etiology of Shwasa is mentioned in our Ayurveda Sanhita. So these are the some etiological factors are mentioned in Ayurvedic Sanhita. There are Raja means exposure of dust, then Dhuma means exposure of fumes and smokes. Also mentioned exposure of breeze, living in the cold places, then excessive consumption of cold water, chilled or frozen water, excessive indolence in the sexual activities, also excessive walking, excessive consumption of dry fruits, consumption of incompatible foods, then flatulence, then excessive darkness of the body, excessive fasting, long-standing disability, injury to the vital area of the body, then diarrhea, fever, vomiting, chest injury, cholera, lazy stomach, anemia, liver disorder. These are the some etiological factors are mentioned in our classics. So in our Ayurveda Sanhita, two types are mentioned of Tamaka Shwasa. First one is Pratamak Shwasa. When Tamak Shwasa is associated with a Jvara and Murcha, is called Pratamak Shwasa. And Santamak Shwasa, Tamak Shwasa is occurred due to the Udavarta, Ajirna, then Rajakana, suppression of urges like Mala, Mutra, and which increase mostly during night, called as a Santamak Shwasa. So, in our Ayurveda classics, some Purva Rupa are mentioned, Dakshila, then Parshvashula, then Bhaktanna Dvesha. So, these Anaha, it means flatulence, distension of abdomen, then Parshvashulam, it means pain in the sides or flanks of the body, then Hruday Pida, it means heart pain or pain in chest. Then the Vayu running abnormal or restorage means upward direction. These are some Purva Rupa are mentioned in Ayurveda classics. This is the Samprapti mentioned in Ayurveda classics. Annava Strotas, this Vyadhi is belongs from Annava, Udakva Strotas Dushti, Vatava, Vata Dosha Prakopa, and Pranava Strotas Dushti. So these are involvement, and this is the picture. So I have less time, so I take a next slide regarding epidemiology. There are about 3 million asthmatics in Japan, of whom 7% of severe and 30% have moderate asthma. In Australia, one child in 6 under the age of 16 is affected. This is very dangerous, my dear friend. And in India, estimated 15 to 20 million asthmatics. In the Western Pacific region, WHO, the incidence varies from over 50% among the children in a Caroline Island to virtually zero in Papua New Guinea. In Brazil, Costa Rica, Panama, Peru, and Uruguay, prevalence of asthma symptoms in children varies from 20% to 30%. So I post one picture over here, diagnosis of asthma. One doctor ausculted one patient having a coughing, wheezing, shortness of breath, then chest tightness. These are some symptoms. So these symptoms are patterns, and taking a history of how much severity of this type of symptoms. And also the doctors taken a history of family history also. This is pathology of asthma. Some pictures in a upper side chest, and second one is normal airway, muscle, then lightening, then airway in the person with asthma, swelling and mucus in a second picture. What is asthma? I told you about our tamak shwasa. Now I told about chronic diseases of the may cause wheezing, breathlessness, chest tightness, nighttime or early morning coughing. These are symptoms mentioned in modern medicine. Episodes are usually associated with widespread but variable airflow obstruction with the lung that is often reversible either spontaneously or with treatment. The asthma cycle. This is the asthma cycle. Trigger of asthma. First one, inflammation, bronchial hyperactivity or bronchial reactivity. Then airway remolding. Trigger of asthma. This is one picture. Asthma prevalence, already told you, prevalence 150 million people around the globe, roughly equivalent to the population of Russian Federation suffer from asthma, and this is the number rising. It's very dangerous, my dear friends. Worldwide death from condition have reached 1,80,000 annually. These are some pictures who are suffering from coughing, wheezing, sneezing, backache, all this type of person who are suffering from asthma. This is the X-ray, chest X-ray, pneumothorax, pleural white line is mentioned over here. This is the X-ray of collapse lung. These are some bronchodilator used commonly in a modern medicine or regularly used patients in a who take a modern type of treatment. So this is my first aim of my study, to evaluate the efficacy of Krishnadi Churna in the stomach shwas, also to study the Nidan Panchak of the and modes of management of stomach shwas, evaluation of Krishnadi Churna in the stomach shwas, improving your Bala and Sthanavaiguna. This is my, and I use materialance method. I take 50 patients, 5 gram Krishnadi Churna with a 15 days medicine, and it includes Pimpli Sendho. Pimpli means Piper longum and Sendho means rock salt. So this is my inclusion criteria. Male, female both will take. CBC eosinophils and patient having symptoms of tamak shwasa mentioned in our Charak Sanhita. Shwasa kruchita means dyspnea, Kasa means cough, Gurgurak Shabda means wheezing or ronchi, Kastena Sleshma makes difficult in expectoration, Kastena Bhashya, it's means difficult in expectoration, and Anidra, it means insomnia. These are my exclusion criteria below 16 to 65 years. Patient, I excluded patient suffering from neurologia, patient suffering from meningitis, psychological disorder, any type of malignancies, lactating mother, lung disease like tuberculosis, carcinoma are excluded in my study. These are my drug review. Krishnadi Churna, it having a Pimpli and Sendava in the same quantity in a powder form. This is a picture. This is the herbal conference. Right now, start herb. So I want to explain about herbal medicine mentioned in Ayurveda classics. So I use this two medicines in our research work, Pimpli and rock salt. This is one of the shloka are mentioned in Ayurveda classics. Pimpli, it means long rejuvenating effect of the lung. Pimpli plays an important role in aiding the thermogenical response, that is release of metabolical heat energy. The effect is a very result, increase your thyroid hormone level in the body and makes Pimpli a typical Ayurvedic complementary component. Those benefit is to increase the bioavailability and enhance absorption of the other active ingredients. Some chemical constitution. The fruit contain 1% volatile oil, resin, alkaloids, piperine, and piperlongumine, avocy alkaloids. These are the chemical constituent. Piperlongum grows well in sandy loam soils which are well drained. So these are mentioned properties of pimply. This is the start herb conference. So properties of our herbal drugs. It is very important. So our drug is pimply. So pimply contains, it might have a anti-inflammatory properties. It is a process or possess antioxidant properties. It is a antimicrobial effect. It show anti-cancerous property. It exhibit anti-Parkinson properties, anti-stress activity. It having anti-epileptic activity, anti-hyperglycemic effect, exhibit anti-hyperlipidemic effect, anti-platelet effect, anti-arthritic property, anti-ulcer activity, then anti-asthmatic effect. It possesses anthelmintic properties, lots of properties of pimply, my dear friends. Potential uses of pimply. So potential uses of pimply is insecticial. According to the studies, fruit essential oil and addition of piperidine and alkaloids obtain P. longum as instead very good actions. Pimply is antifungal activity. Pimply might have fungicidal properties in essential oil of fruits. Also, pimply use antimicrobial activity. Pimply potential is anti-myopic benefits also. Then uses of pimply, immunomodulatory activity also. It has been CD4 plus and CD8 T cells are having in a pimply. So it is mentioned in pharmacology department also. So potential use of pimply is anti-diabetic effect also. Pimply has been proven have a strong anti-hyperglycemic and anti-lipid, peroxidative and antioxidant properties. So also pimply is antioxidant benefits also. Our second drug is Saindhav. Saindhav means Lavana, mentioned in Ayurveda. And in modern medicine, its name has rock salt. Latin name is rock salt. Benefits of rock salt is a full of minerals and vitamins, which improves your digestion, promotes bowel movement, and helps to clean toxic product from the intestine. It is also help to improve loss of appetite. The study result suggests that any disturbance is there. Result studies that rock salt can stimulate insulin in the body that prevents sugar cravings and loss of appetite. Rock salt having a digestive stimulant, so it is a might be due to the stress minerals. It promotes digestion and improves your appetite. Acidity, heartburn, and ulcers. It is very effective. It is a antacid, so it is a decrease your acid production in the stomach. It is also prevent your acid refluxes, which may result in a heartburn. So in Ayurveda, sendha namak used with sof means fennel seeds, coriander powder, and cumin seeds for reducing your acidity in the stomach and improving your digestion. This is one good medicine for your daily use for any type of heartburn or any type of acidity. Uses of Saindhava Lavana is a sweet, it is a carminative, is it a digestive, it is light and lubrication. So it is you improve your taste. It is a cooling and aphrodisiac. It is a suitable. It means it's a subtle, means sukshma and penetrates quickly. It is a good for your eyes. It is check the 3 doshas. So 10 to 13 grains and digestive 120 to 240 grains are emetic. So action in our Sahita, Sushruta, Murdu, Sita, Virindiyavat, Pitta, Haram. It is mentioned in our Ayurveda classic in Sushrut Sahita. So cooling when compared with Samruddha Lavan, digestive, preventive and antiseptic. Our third medicine, this medicine we use as Anupan, means this is medicine we can use with our two churnas, gingiber officinalis, it's adrak or ginger. So properties of ginger rich in various biological active compounds like phenolic and flavonoids. Ginger might possess a wide range of beneficial properties. These may be include it having antioxidant activity, it may have anti-inflammatory action, it may have anti-cancer activity, it may have anti-microbial activity, it benefit in keeping healthy weight, it boosts your glucose tolerance, augment lipid profile, uses. It is a aphrodisiac, it balance your kapha and ama doshas. It is useful to treatment of disease related to vata doshas. It clear your abdominal disease and worms, bronchitis of asthma. Then good remedy for gastric disorders, rheumatism, fever, and nervous diseases also. A mixture of ginger and honey includes motion and also cures your tympanitis. A recommended remedy for colic, then diarrhea, flatulence, dyspepsia, and loss of appetite is very good medicine or good herb. It relieves your pain, it relieves your swelling, it relieves your headache and toothache and internal paste also. These are some potential use of ginger. Use in respiratory system. Ginger may bronchodilation activity. Potential for ginger for liver. The phenolic compounds ginger may have demonstrated liver benefits effects. So potential of ginger for allergies, the anti-allergic activity. Ginger is managing allergic rhinitis also by suppression, psychotonic production. Potential ginger for pain killing activities. Indigestion of ginger might have display significant effect in relieving primary pain. So after study, this is observation and result. I give some medicine for the patient and I examine or I observed respiratory rate, expansion of chest, breath holding time, peak expiratory flow rate. These are the some readings are mentioned over here. And investigation criteria: hemoglobin, WBC, neutrophil count, lymphocyte, eosinophil counts, and erythrocyte means ESR. These are some before treatment and after treatment. These are my some readings. These are some readings regarding symptoms means lakshana, shwasakruchata, kasa, gurgurak shabda. I work on these 6 symptoms and I will get this type of result. These are the some readings. So let's conclusion. During the comparative study or values of both, it has been compared, conclusion were drawn. The same significant effect of Krishnadi Churna is more effective on the basis of study. It can conclude trial. Krishnadi Churna is very effective in the management of respiratory disease and enjoyment. This is my discussion. Features like appetite, digestion, gain improvement, then due to improved nutrition at the cellular level, Deepan Pajan, we call it as Deepan Pajan in modern technology, it's carminatives and digestives. And Agni Vardhan means increase your digestive power and individuals. Properties of Pimpli: improved your nutrition to reach or each of the body tissue, result improvement in the features like general and mental feeling of well-being. So this churna is having a vata kapha pacifying, stroto shadan and kapha nirasan properties, makes the pathway. So expansion of chest, breath holding time, sustained maximal inspiration, increase significantly. Hematocrit value, HB increases significantly. Lymphocyte neutrophils and ESR reduces significantly. Mild changes were shown in WBC and eosinophil count. No any type of side effect observed from the mild drug during the present study. Thank you very much. And thank you all the organizers who invite me such a wonderful platform. Thank you very much. This is my contact details and my email ID dr.bhairavkulkarni@gmail.com. This is my website www.sri-siddhivinayak-panchakarma-center. Then this is my contact number over here. If any query, then contact me. Thank you again. Namaste.
Thank you, sir. Thank you so much for your informative session. Participants, if you have any questions, you could ask our speaker. Okay, sir. I guess there are no questions from the participants. Thank you so much.
Sir, one question, sir. Sir, this is Dr. Srinivas. One question, sir.
Tell me, sir. So tell me, please.
So first of all, I would like to say the congratulations, sir. Just now I've seen what is you are showing that. So with this I've seen I got an inspiration, sir. And wholeheartedly I am congratulating you. And most of people who are taking especially the AYUSH system of medicines and of course Ayurveda and all these, people still not have confidence. And another thing is people are not having the option to take easily. So this is what I feel I'm sharing with you, sir. Instead of that, your all explanation towards the pharmacological action and action about the drugs, everything is nice, sir. Thank you so much.
Thank you very much, sir. In text message, someone asked about a dose of Krishnadi Churna. So I tell you, I use 5 grams morning, 5 grams evening after meal with the adrak swaras, means ginger swaras. So it is very effective. If you have any questions, so you can ask me anytime in my email ID dr.bhairavkulkarni@gmail.com. And also you can contact me on my mobile number also, which already I given to you. It's 99672-82076. Thank you very much. Can I stop my presentation?
Yes, sir. Like there are other 2 questions also.
Yeah, please.
The question is, what is the administration of drug in? That is the one of the questions, sir.
Administration of drug is orally. I already told you it's a orally administration. 5 grams morning, 5 grams evening after meal with ginger swaras.
Okay, and other question is from Ms. Sangeeta. So as a student, I want to prepare a churna. So where to have formulation test for same?
What, ma'am? Please explain again.
She want to prepare a churna so that where have formulation tests to be taken.
Formulation is Ayurvedic College Pharmacy. She can do.
So the person is asking in which form, like in water or direct powder they could take.
No. These are 2 products, Pimpli Churna and Sendhav. Sendhav means namak, right? These are 2 powders, mix same quantity and take with other ginger swaras, means ginger liquid, and taken orally. After that, you can take warm water.
Okay, sir. Thank you. Thank you so much for taking out of your time, sir. Thank you.
Any question?
That's it, sir. That's the
Thank you
questions.
very much, ma'am. Thank you.
Thank you.
Thank you.
Next, I would like to welcome our keynote speaker, Dr. Satnam Singh, Associate Professor, Pharmacology, AYUSH Institute of Medical Sciences and Research, AIMSR, AYUSH University, India, to deliver his keynote speech.
Good morning, everyone.
Good morning, sir.
First of all, I would like to congratulate all the organizers who have taken this initiative to conclude the talk on this topic, because it is a very happening area which is related to the healthcare system also. So secondly, I'm also very thankful to the organizers, those who have been given with this opportunity to speak in front of this August gathering, though it is a virtual mode, no problem. I would like to share my content to the PPT as well. Actually, my talk will be based on the simple knowledge of the herbal medicine, and as well as pharmacological profile, what are the myths and what are the realities, and how to use, how to increase the efficacy of those herbal preparations or herbal medicines or herbal ingredients. So many things are there. I would like to share my slide quickly. Now my screen is visible or not?
So it has been just started screen sharing. We couldn't see your screen.
Okay.
Yeah, now it's visible, sir.
Right, visible. Okay. And, would like to make it somewhat enlarged.
You could.
Not in the PPT mode, no?
Yes, sir.
Okay. Is it visible right now, ma'am?
Yes, sir. It's visible.
And ma'am, please check if it is.
It is in the full screen, sir.
Yes. Moving.
Yes, sir. It's moving.
So the topic is pharmacology and pharmacotherapeutic potential of natural products, especially. Pardon?
Sorry for the interruption, sir.
So basically, the talk is related to the clinical efficacy and future of healthcare, because nowadays this sector is becoming a very promising area in the field of healthcare system or healthcare domain. So I would like to start with the simple things. So what is herbal medicine? Basically, herbal medicine, which is also known as botanical medicine or botanicals or phytomedicine, basically are the ingredients or the whole extracts or the whole part of the medicinal plants or the plants which are having curative type of properties. So herbalism is a traditional medicinal or folk medicine practice based on the use of a plant seed, berries, root, bark, or flowers, and plant extracts for the medicinal purposes at different stages with the different requirements. So advances in clinical research are showing the value of herbal medicine in treating and preventing the diseases is much more, and the importance is gaining ground day by day. Researchers found that people in different parts of the world tend to use the same or similar plant for the same purposes. As far as the history is concerned, plants had been used for medicinal purposes long before the recorded history. Ancient Chinese and Egyptian papyrus writings describe the medicinal uses of the plants. This is the proof. Again, in the early 19th century, scientists began to extract and modify the active ingredients from the plants. In the last 20 years, in the United States, there is an increase in herbal medicine use due to the public gets dissatisfaction with the cost of prescription of the allopathic medications and interest in returning to the natural or organic remedies. Interest towards returning to the natural and organic remedies. Why? I will explain this later on. Why the people are returning to this. Again, recently, WHO estimated that 80% of the people worldwide rely on herbal medicine for some part of their primary health care. In Germany, about 600 to 700 plant-based medicines are available and are prescribed by some 70% of the German physicians. This is the data, small data of the world. So complementary and alternative medicine came in Palestine. The use and safety implications done by some scientists, they have basically concluded that 72.8% of the respondents have used at least one type of the natural alternative medicine. So the users were mainly middle-aged, low-income, educated women. Herbal therapy, prayers, and honey were also the most utilized types of the camp. But main is the herbal remedies. Herbal formulation. Now come to the herbal drugs. What are the herbal drugs? Herbal formulations or the dosage forms consisting of one or more herbs or processed herbs in specified quantities provide specific nutritional, cosmetic, and health benefits, and other benefits meant for use to diagnose, to treat, or to alter the structure of or physiology of the human being or the animals. Herbal preparations are obtained by subjecting whole plants, fragmented or just by cutting the plants or using the plant parts to treat, such as extraction, distillation, expression, fractionation, purification, concentration, or concentrated solution, or fermentation. This include muted or powdered herbal substances, extracts, essential oils, extract juices, etc. So nowadays, it is a well known fact that the old days are gone, where the herbal healthcare system was viewed as an alternative one. Now it is a great change which has been carried out with the great use of these medicinal plants or the herbal drugs. So now it is a change scenario. It is not only integrated into the mainstream healthcare system, but it is also attracting the attention of the big companies or the pharma players to produce different types of products for the benefits of the patients. So basically, herb were considered as a food supplements. So herbal supplements were classified in the past as a dietary supplement. So the one classification was also given by the US agency, which is known as Dietary Supplement Health and Education Act, which was generally passed in 1994 and covering all the herbal supplements, nutraceuticals, and all the things related to this area. The phytopharmacology, basically it is a branch of the pharmacology which deals with the pharmacological actions or the potential of pharmacotherapeutics, which are generally based or extracted from the plants or related to the plants or herbs or of herbal nature. So in the many cases, the scientists are not true what specific ingredient in a particular herb works to treat a condition of illness. But whole herb contains many ingredients and they may work together to produce a beneficial effect. It has been seen. It is a well known effect or established effect or reported basically research. If you will use single ingredient of a herbal extract or a ingredient from a plant, it will not work or will not show efficacy as compared to the whole extract. So there is a action which is based on the synergistic mechanism. So their action is synergistic. It may be additive in nature. So it has been seen when we are using a whole part of the plant, whole extract or the whole material, we are getting the good result and with a good efficacy. Secondly, there is a special mechanism to do the things to as far as the allopathic pharmacology is concerned. It is this pharmacology is slightly different, but processes are there. It has been seen if any herb is basically capable of killing the microbes effectively, but the mechanism is quite different. It will not. It will not kill the microbes as the antibiotics. It will just alter the paths of synthesis of proteins for the microbes in a different way. So ultimately, the microbes will kill. So there will be no alteration in the pathway or no toxic effects and no other side effects. Basically, there are some factors which contributing towards the parameters which are set for the collection of a the plant part used, because the type of environment, climate, bugs, soil quality, which generally contribute toward the proper amount of of the ingredients in which a plant grew. Different types of same species of the plant, when we using the same plant at different places or cultivating at a different levels in a different type of environment, we may get different types of ingredient in a different ratio. So it is very important. So that second is time of harvesting. It is very important because if we will delay the harvesting time, the content will be lost. And it is also important. Some plants are having the concentration of the active ingredient is much more in the morning, or some are at afternoon. If we will pluck them, we will harvest them during the evening, we will have the less content. So many herbs or many plants are used for their therapeutic effects in case of asthma, eczema, premenstrual syndrome, rheumatoid arthritis, migraine, headache, menopausal syndrome, and chronic fatigue, irritable bowel syndrome, respiratory disorders, liver ailments, kidney diseases, and very important, many types of cancers. Generally, it is a established fact, it is a recorded document or documented fact that the cancers are cured with the natural products with a great efficacy. The scope of herbal medicine is somewhat different as we are generally guessing. The first is they are having great nutritional value. So many of herbal products and the herbs are used as a food and nutritional supplements, and they are very effective and they should be used in proper quantities on routine basis, like coriander, garlic, nettles, and oats. And maintenance of health, the second domain where they are very high, they are having very high scope, the maintenance of the health. So like this dandelion and garlic, they are very effective in maintaining the health on a routine basis. Then treatment of disease. Third is the medicine. So which we can employ for the acute and chronic cases as well. So drugs from plant sources, many drugs are there, starting from aspirin, cocaine, quinidine, scopolamine, ibuprofen, pilocarpine, dioxin, morphine, gotamine, codeine, etc. Many examples are there. And many herbal medicines are available in different types of dosage forms. Like the herbs come in several different forms: teas, syrups, oils, liquid extracts, tinctures, and dry extracts, or in the form of pills and capsules also. As far as the safety is concerned, it is very important. We should know the safety level. We should be aware of regarding the safety concern. Many consumers believe that herbal medicines are natural and therefore safe, but it is not so true. It is not 100% true, because these are the some time before. Now there are some regulations put forwarded by the government for the processing, and just to go on the processing and to go on the safety concern, but still they are unregulated at a very different type of stages. They may contain additives and contaminants which may harm our health. As far as the purification is concerned, they may cause allergic reaction. Yes, they may interact with the conventional drugs. If the person is already taking the allopathic medicine, he or she should not take or mix these two pathies. The toxic, they may be proved toxic if used improperly or at a very high doses. So I think slides are not moving now. So, herbs with serious side effects. As per the FDA reports are concerned, I would like to highlight only few things. Herbal laxatives, usually sold as a diet tea, Scara, Senna, they can cause cramping, diarrhea at higher doses. They could cause bowels to no longer function without laxative stimulation, mean dependence. They can cause dependence. Similarly, ephedra in high doses can raise blood pressure, increase heart rate, and overstimulate the CNS. Then it may also produce the withdrawal effects in future. Again, it has been reported by the FDA that more than 800 of side effects are linked with the irrational use of this ephedra, including heart palpitation, seizure, stroke, chest pain, and heart attack are the main or major on the list. So this has caused at least 2 deaths also. Then there are some basically tricks by which some persons are generally projecting this herbal medicine as a magic remedy, but it is not so true. Basically, herbs are very effective in gaining or in reducing the weight. Certain herbs they trick brain into thinking the stomach is full. Basically, that's why the patient or the people or the person will not take the more food, and ultimately the fat will get started dissolving. So what others are thermogenic, speed up the body's metabolism. This is very true. So burning more fat, unwanted fat at a higher rate. So similarly, weight will be reduced. So thermogenic herbs contain stimulants that is very effective and very essential. There are some safe herbs to lose the weight. This kind herb, green tea, sawweed or kelp, nettle, ginseng, then hodia. So basically, they are stimulating the digestion or uh, uh, acting as a source of antioxidants or stimulant the BMR, or they are having thermogenic effect. All these effects are contributing toward the loss of weight or shedding of extra weight. So these are the pictures of these. So basically, a study to describe the side effects associated with the herbal products used for weight control is also there. It was conducted in Italy. So it has been reported that 64% of the suspected side effects were associated with the herbal products used for weight control, which was not basically used according to the instructions, and the instructions were not followed by the user. So women were involved in 85% of the reports. The reactions affected mainly the cardiovascular system, the skin, the digestive system, the CNS, the liver. At a large proportion of side effects were serious. So to be on the safer side, consult the physician first. Do not take a bigger dose of the label recommend. Take it under the guidance of a trained medical professional. Be specifically cautious or specially cautious if you are giving or prescribing to the pregnant women or lactating women, or if you are the prescriber or a nurse, basically. Therapeutic effects of some most common herbs. Now here, I would like to highlight some important herbs which are safer and which we can consume on routine basis. Their pharmacological actions are very simple. They are helping in improving of our health. First is garlic. It is very effective, and it has been seen that it is lowering cholesterol and triglycerides in our blood plasma. It can kill the worms, round worms especially. It can reduce the hypertension and improve the blood circulation. It is very effective in preventing the cancers, every type of cancer, because it has special ingredients. Effective in diabetes, effective in bronchitis, asthma, and allergies. Some side effects to some people, those who are taking it in a raw form. Skin rash may be there, but overconsumption can cause heartburn, not more than that. Ginger. It is a very, very good herb which should be consumed on routine basis in our house or in our diet. So it is very effective in cold, cough, and in treating the vomiting. It may reduce the angina episodes, and it is also affecting in lowering of cholesterol. It relieves nausea, motion sickness, morning sickness, menstrual cramps. Expectorant, coughs and bronchitis, parasite repellent. Then some side effects are also associated with this. It is narcotic in large doses. Any seed contains anethol, a plant hormone similar to human estrogen. So it should be avoided during pregnancy and used with cautiously in various conditions. The essential oil is useful and very effective in relieving some ailments, as I already discussed. Now the green tea. It is basically very effective and very good beverage. It has been reported that it reduces the incidence of development of cancer. It is a metabolic booster. It has the potential to lower the cholesterol as well. It relieves the inflammation and itching of the insect bite, reduces the pink eye inflammation, conjunctivitis, and it is a good source of antioxidant basically. But there is a myth that the green tea should be taken during the day for 3-4 times. No, it is not so true. First of all, green tea should not be taken in empty stomach as a bed tea in the morning. Secondly, not more than 3 serving of the green tea should be taken in a day. And third, after 5 p.m., it should not be consumed. So these are certain guidelines regarding the use of green tea. So overuse of caffeine can affect sleep and insomnia. Why? This is the reason why it is not consumed after 5 p.m. Lavender. It is very good herb and used to reduce the anxiety and stress. Its smell is very effective, and aroma is very effective pharmacologically. It sensitizes the brain and normalize the hormone level and neurotransmitters in the brain. So it is also effective in healing of skin abrasions, burns, and scales. And it is anti-inflammatory in nature. It has also antiseptic properties. In case of flea bites and rashes, it can be employed. So it can also used to relieve the fibromyalgia. Lowering of blood pressure and relaxing cardiac spasm, such as tachycardia, is also associated with its properties. It can also effectively relieve the arthritis and improve the circulation. Then it can relax the neck and head muscle. So it is also effective in relieving the nervous headache. It is also effective for the treatment of leukorrhea. Thyme. It is also considered to reduce the fever as an antipyretic agents. It is also effective in case of bronchitis, fighting toothache, tooth infections, very effective for the treatment of cough, flu, and sore throat. And in case of insect repellent. Side effect, excessive amount can affect menstrual cycle, only excessive amount. Thymol is toxic and should be used with great care in moderation. The essential oils should never be applied undiluted and is not suitable for use in small children. Especially, it is not suitable to be applied on the soft surfaces or on the mucous membrane. Otherwise, burning sensation will be there. Black seed, relieve rheumatoid arthritis, high blood pressure, anti-inflammatory, analgesic, antipyretic, antimicrobial, and anti-neoplastic activity is there. It is known to prevent hair loss also and provide healthy hair. Black seed, some side effects. Undiluted oil can cause skin irritation, not to be used in pregnant women. Chamomile, calm and anxiety, eases stress and may have a positive effect on attention deficit disorder. Used for irritable bowel syndrome, anti-inflammatory and antihistaminic activity are effective for nausea and morning sickness when used with ginger. Side effects, allergy for sensitive people. Those who are sensitive to this herb, avoid chamomile teas when taking blood thinners. So the tea of this herb should not be taken. It contains natural blood thinner as a coumarin. So dose will be increased. So an effective approach to increase the clinical efficacy of herbal drugs. It has been seen when we are administering the drugs, herbal drugs, by the conventional methods, there are some weak points, there are some disadvantages by which the herbal drugs are not working properly. As efficacious they can work when we are using them in specialized drug delivery system, the novel drug delivery system. So the approach is to convert them in a novel drug delivery systems or novel drug delivery dosage forms. Novel drug delivery system is advantages and delivering the herbal drug at a site of action which minimize the toxic effects with the increase of the bioavailability. In novel drug delivery technology, control of the distribution of drug is achieved by incorporating the drug in a carrier system or in changing the structure of the drug at molecular level. Herbal drugs are becoming more popular in the modern world for their application to cure variety of disease with less toxic effect and better therapeutic effects. The novel carriers should ideally fulfill two prerequisites. Firstly, it should deliver the drug at a rate directed by the needs of the body over the period of the treatment. Only the required amount at a particular site, at a desired site. No toxic effect. Secondly, it should channel the active entity of the herbal drug to the site of action, where the action is required. So the various drug delivery and drug targeting systems are currently under development. Novel drug delivery is a new approach to the drug delivery. It helps the drug to act longer and more effectively in case of herbal drugs, to minimize drug degradation and loss, to prevent the harmful side effects, and to increase the drug bioavailability. Which drug? Herbal drugs. So the drawbacks of the conventional dosage forms: poor patient compliance, increased chances of missing the dose of drugs. The unavailable fluctuation of the drug concentration may lead to the under medication or over medication. A typical peak valley plasma concentration time profile ID obtained, which make attainment of steady state condition difficult. The fluctuation in drug levels may lead to the precipitation of adverse effects, especially of a drug with small therapeutic index whenever over medication occurs. Advantages of novel drug delivery systems: enhancement of solubility. Some herbal drugs are not soluble in general solvents. Increased bioavailability. Yes, the issue of bioavailability has been solved or sorted out or solved with the use of novel drug delivery system for the administration of herbal or herb-based drugs. Protection from the toxicity. Yes, herbal toxicity can be prevented. Enhancement of the pharmacological activity. Yes, it is very important. Just to converting the medicine or the dosage form in a better way, by using new techniques, the pharmacological activity and the profile can be increased. Enhancement of stability. Yes, this is very important. It has also been increased. Sustained delivery of herbal dosage form, which was not there in the previous years. Then protection from the chemical and physical degradation. Again, very important. Advantages: lower risk of side effects. Why we are converting? Why we are relying on herbal drugs? So because there are certain advantages. One is low risk of side effects, more effectiveness, lower cost, widespread availability. Some limitations are also there. They are not suitable for many diseases. They cannot be combined with allopathic system of treatment. Lack of dosage instruction is there in some cases. Poison risk associated with wild herbs, where we will have to take the preparation very carefully under the medical supervision. Then lack of regulation, regulatory control, and longer duration of treatment sometimes. But still, it is very effective domain. So different novel drug delivery system for herbal drugs. We are converting. It has been reported that many herbal drugs are generally prepared in different drug delivery systems for administration. So these are liposomes, phytosome, nanoparticle, implant, this transferosome, microemulsions, and ethosomes. Liposomes are biodegradable colloidal and spherical vesicles composed of a bilayer membrane entrapping an aqueous core. Aqueous core enclosed in one or more phospholipid. They are used to convey the vaccine, drug, enzyme, or other subs to target cell or organ. This is the picture showing the core where the drug will be retained by this phospholipid bilayer and it will be delivered at a targeted site. So Lee et al. Prepared the liposomes containing extracts of T for the thin film dispersion method. Then advantages of liposome formulation: hydrophilic and hydrophobic drug can be delivered. Liposome herbal therapeutic act as a carrier for this, and then the cytotox molecules can be removed. The and macromolecules can be used. Sustained and controlled release of the formulation can be possible with this technique. So this is the basically chart which is showing different types of plant constituents starting from 1 to 12 and their biological activity, starting from anti-cancer, digestive, analgesic, anti-cancer, lungs, antimicrobial, and application of these particular plant constituents or plant products in the form of liposomal dosage forms with the latest technology. So we can see improved therapeutic outcome, bioavailability has been improved, action has been prolonged and enhanced. So all these outcomes are basically received with the use of different types of technologies to convert the conventional herbal dosage form into novel drug delivery dosage form. Now the phytosome process, the basically phosphatidyl choline and the plant components actually form a 1:1, 1:1 or a 2:1 molecule complex, depending upon the substance complexing involving chemical bonds. So it is also very effective. Advantages: phytosomes are better bioavailable botanical extract, dramatically enhanced and delivered the faster and improved absorption of, especially in case of intestinal tract. Phytosome permeates the non-lipophilic board extract to be better absorbed in intestinal lumen. Phytosomes will be given in small quantity and desired results can be achieved. Small quantity and desired results. So similarly, phytosome is widely used in cosmetics due to their more skin penetration and have a high lipid profile. So this is the phytosomal herbal products. The list is showing starting from curcumin, membrane. All these are the plant constituents or ingredients, their biological activity starting from antioxidant, antibacterial and antidiabetic. All the biological activities are listed over here and their application in the form of a phytosome will increase the bioavailability, solubility enhancement, absorption enhancement, improvement in the efficacy, improvement in the absorption again. So by just converting these, we can enhance the pharmacological profile and the clinical efficacy of the same. Now the ethosomes are vesicle composed of the phospholipids and high concentration of ethanol. So high concentration of ethanol in the vesicles led to the enhancement on their permeability through the skin by fluidizing the lipid domain of the skin. So implants, these are the polymeric devices which are used for the controlled and sustained drug delivery in the system. These are directly placed in the body by just given a incision, and these type of implants are generally placed and stitching is done. These are of two main types: biodegradable and non-biodegradable. Non-biodegradable, after the core will release the whole material, whole drug in the bloodstream. After some time, a gap of few months, the non-biodegradable is generally extracted out. They're just given again incision and it is replaced. It is, and in case of biodegradable, the shell is biologically degraded in inside the skin. So implants of the extracts of this tension were developed using by the chitosan and gelatin type of material. Microemulsions, nanoemulsions, and microemulsions are the emulsions of oil and water type having size range of several microns. So they are prepared by using the surfactants, which are considered safe for the human use and approved by the FDA also. So this is the structure of this microemulsion particle. You can see. So these type of emulsions have higher surface area and hence can easily penetrate through the skin. So they are also non-toxic and non-irritant in nature and can be used in the animal and veterinary purposes. So nanoemulsion can be prepared by the high pressure homogenization and microfluidization techniques. Transferrosomal particles. Transdermal are the phospholipid vesicles which acts as potential carriers for the transdermal delivery of the drug as they overcome the difficulty of penetration through the intercellular pores of the skin due to their flexibility. So this Patil et al. Prepared transferrosome containing the curcumin gel and an increase in the penetration was observed when compared with the simple gel through the skin. Marketed novel drug delivery formulation of plant active particles and extracts. So this is the brand name. We are having this white tea in the liposomal form, green tea liposomal form, white hibiscus liposomal form, and here aloe in the liposomal form, then guarana liposomal form. Plant active ingredients and extracts. Camellia sinensis, white hibiscus, aloe vera, guarana. Type of the NDDS. Liposome, 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 and the last is phytosome. So these are the name of given by the company. So these are the available dosage form by. Now the recent patents on novel herbal formulation. Yes, this is the table which is showing the US patent number. These are the patent number. Active ingredient, opioid analgesics and aloe. Then this ginseroids, isoflavones, alkaloids and econitum. Then glycine containing 7S globulin, protein extracts and curcumin, ginger and officinal. Ginger officinal is very important again. So the novel system incorporates this. The first one for, in the form of nasal spray. Second is microencapsulated and controlled release formulation. Again, the third one, isoflavone, which was converted in microencapsulated formulation. Then alkaloids and econitum, transdermal delivery systems. And the last one in herbal tablets dosage form. So we can see how the development is going on in this domain, how the herbal medicine are gaining ground and importance day by day. So the conclusion in this case, novel drug delivery systems of herbal drugs have a potential future for enhancing the clinical efficacy and activity and overcoming the problems associated with the plant medicine also. Herbal drug market, just an overview. India has been using its rich biodiversity for healthcare for at least 4,000 years through its traditional system of medicine like Ayurveda and other herbal system, but requires an appropriate efforts as below to build the market. Number one, test the concepts as well as the products through the modern biology and chemistry. The next is characterize the product at molecular level. Third, develop the newer products which are far simpler, more benign and effective than the ones which we are employed nowadays. As far as the market is concerned, in case of the global market, it has been reported that the herbal healthcare industry is estimated to be around $47 billion globally, while it is in India is contributing is only $1 billion in this industry. As per recent survey is concerned, the same estimated market about rupees 2,500 to rupees 300 crore with the growth rate of around at the rate of 25% every year, including OTC plus herbal. The rate is, this is growing at the rate of 25% every year. You can see the future and scope of the herbal medicines. So around 16% contribution in total pharma market. 1,400 crores herbal OTCs and 1,000 crore ethical herbal markets. Approximately 8,000 herbal manufacturers are there. The herbal healthcare sector is witnessing business. It has come up as an alternative to monopolistic conventional synthetic products. The revival has ensured longevity of herbal companies, and hence there has been a flurry of companies entering the market. Globally too, this market has been growing rapidly, hence big opportunity available for Indian companies who are venturing out to exploit the opportunities offered by the green field, including the big names like Vexi, Cadila, Wokat, Lupin, with the interest shown by both by FMCGs and pharma companies, and now considered one of the high growth industry. Spark of the future perspective. If we talk, products of traditional systems of medicine are unlikely to compete successfully until they have an edge over their competitors. The only weapon of knowledge is appropriately used against competition. Thus, in future, India shall have knowledge-based products by way to develop special preparations either as single molecules or of multiple molecules to make existing preparation trimmer and molecular defined, to investigate synergism amongst various fractions of the extracts, as I already discussed, to develop specific preparations which accelerate recovery after suffering from debilitating diseases, to develop specific detoxifiers. Available opportunities. Growth at the rate of 25% in phytomedicine market has been recorded. The data collected only in case of big companies under their operations, bearing with the local players taking a huge share from the market. Ever increasing health costs and side effects associated with allopathic drugs, customers are now shifting towards herbal medication, which I have highlighted in the beginning, that I will tell you why herbal drugs are required. 2-3 other important things are there. Number one, allopathic drugs are associated with serious ADRs. Secondly, there is incomplete cure. Third, relapses are there. Fourth, there is a very big challenge to the healthcare system in the coming years, that the WHO has reported that in the coming years, the incidence of antimicrobial resistance or the death due to antimicrobial resistance will be much more as compared to the death occurring by the deadly diseases like cancer and other diseases. In the next coming 30 years, it will become more worse. So the herbal drugs are not producing any resistance. That's why, and they are safer. The future of the herbal pharma market in serving the personal and healthcare consumer across the world look bright and promising. As appears, the many more opportunities available to explore the phytomedicine market, and the same could be possible through increasing the prescription base through modern medicine practitioners, herbal medicine practitioners, and prime focus on the general practitioners, which requires experienced, trained, and skilled field force, which may have an ability to identify the customers and focus with them. Phytomedicine market may turn around the overall pharma industry picture in future, which is stagnant with the growth at the rate of 16% to 18% from years together, which came from new molecules price revisions. The phytomedicine market should play a vital role in the growth of pharma industry in future, as per the growth rate mentioned by ORG. Herbal products, particularly hepatic protective, menstrual modulators, antioxidants, immune modulators, anti-arthritis, aphrodisiac, hemostatic, bowel regulators, and bronchodilators. In short, the products are being used in the chronic conditions where prolonged treatment is needed, considered to be effective, safe, dose convenient, and cost effective, rather economical treatment over the modern medicines. Himalaya drugs, Chark Pharma, Dabur, Amil drugs are being considered as a big players, followed by Solomon Labs. As per the available data, there are some states like Uttar Pradesh, Punjab, Rajasthan, Madhya Pradesh, Chhattisgarh, Gujarat, Maharashtra, Kerala, Andhra, Karnataka, and Tamil Nadu are being considered to be the major contributor, as the climate suits for the cultivation of the major medicinal plants and herbs over there. The government is also supporting. The phytomedicine is gaining the status of modern therapeutic. Last slide, ma'am. Therapeutics due to the continuous pouring of the knowledge as a result of increasingly intensive research output. So it is gratifying to note that the Department of Health and Family Welfare is now taking the initiative by involving the industry and R&D experts and the bureaucracy to work towards standardization, validation, and for bringing about cohesiveness between different systems of traditional medicines to protect the national interest and image. There are some weaknesses also. The phytomedicine products are selling as a very special selling entity. The customer selection and promotion should be there for the product. So with the changing scenario, phytomedicine market becoming a prime focus with the big Indian companies along with the MNCs. The herbal medicines are poorly regulated. This is the weakness. Assurance of safety, quality, and efficacy of the medicinal plants and herbal products is a key issue which needs to be addressed. So, natural plants and microbial derived products are efficacious in maintaining the human health and cure of diseases as efficacious pharmacotherapeutics. The exact nature of their significance and mechanisms by which these natural products regulate the cellular homeostasis that maintains the cell survival, differentiation, and death, leading to the pathological consequences required further detailed investigation. Therefore, the identification and development of the important natural products as novel pharmacological agents could help to combat or neutralize the deleterious effects and would be the next important targets for future drug discovery studies for the coming healthcare setup. The natural novel pharmacotherapeutics or the pharmacological agent will definitely play an important pharmacological role in terms of costs, improved efficacy. These are the very important factors which I am highlighting in my last slide. Costs, improved efficacy, decreased dose burden, nil side effects, no adverse reactions, with no incidence of drug resistance or relapses of disease episodes. Hence, novel pharmacotherapeutics or pharmacological agents will be proved as a backbone of healthcare system in the future. Thank you very much. Thank you. I think I have finished within the stipulated time. Am I audible, ma'am?
Yes, sir, you're audible, sir. Thank you. Thank you so much for the presentation. The Sir, they are asking to share your contact details.
Contact detail, ma'am.
Yes, sir. Kindly drop a message of your contact details in the chat box. No.
मैम आपके पास है नहीं? Okay, मैं there are some questions I think which are posted over here.
Yes, sir. There is a question like there are any herbal resistant as like antibiotic resistance.
No, ma'am. No resistance has been seen so far. Antibiotic resistance is due to the different mechanism, different type of pharmacology. It has been established that if we are using these herbal products or natural products or plant extracts, which are capable of killing deadly microbes or pathogenic microbes, they are basically killing these microbes in a different way. The microbe is requiring some different set of cellular reactions. Basically, these ingredients will block the reactions occurring in the microbial cell in a different way, and they will not produce any toxic effect over there as well. So different types of mechanisms are there, or they can never be produce resistance in future because the system is different. Another is there drug-drug interaction occur when we give lavender oil and chamomile oil? No, chamomile and lavender can be given, no drug-drug interaction, but allopathic drug or herbal drug may have drug-drug interaction.
Thank you, sir. Thank you so much for the questionnaires also. As we are, sorry.
Any query, ma'am? Any other question?
I guess that's all the questions, sir.
If a plant extract has anti-cancer property, what kind of scientific proofs?
Sir, kindly answer the questions in the chat box as like we are running out of time. So sorry for the interruptions.
Nice question, but okay. Okay. Ma'am, I think I have got muted myself during some slides. My voice was missing.
Yes, sir. Actually, there was an interruption between.
Sorry for it, but I do not know how it happened. And so, if you allow me, then I can. If a plant extract has anti-cancer property, what kind of scientific proofs needed?
So sorry, sir. Like other speakers are also waiting. Like if you want to answer, like kindly answer in the chat box. So sorry for this. So kindly drop your email ID, sir, so that if any of the participants have questions, they would directly contact you.
Okay. Email ID. Can I stop sharing?
Yes, sir.
Ma'am, you are from DY Patil?
No, sir. I'm from Association of Pharmaceutical Research.
Okay.
Thank you, sir. Thank you so much for your informative session, sir. Next, I would like to invite our next keynote speaker, Dr. Chaya Gadgoli, Professor, Saraswati Vidya Bhavan's College of Pharmacy, India, to deliver her keynote talk. Ma'am, please.
Yeah, I'll share the screen.
Yes, ma'am.
Is the screen visible?
Yes, ma'am, it's visible.
Okay.
And just as guidelines for all the keynote speakers, ma'am, like kindly stick on to the time.
Yeah.
20 minutes for the presentation and 10 minutes for the questionnaire. Thank you.
Yeah, definitely. Definitely. Is the screen visible properly?
Yes, ma'am, it's visible, but
Okay. Am I audible?
yeah, ma'am, you're audible. Kindly
Okay.
full screen your.
Yeah, I'll do it full screen. Is
Yeah,
this visible now?
yes, ma'am, it's visible. Thank you.
I'll just change and just see whether the slides are getting changed.
Okay.
Getting
Yeah.
changed?
Yes, ma'am, they are getting.
Okay.
Thank
Okay. Okay. Thank you, madam. And we'll start with this. So I'll be presenting few case studies which we had carried out in our laboratory. So the title of my talk is Research and Development of Natural Product. As you can see, there is a cycle. There is a traditional medicine available. Intervention of science and technology can lead it to the phytopharmaceuticals, nutraceuticals, and cosmeceuticals, and many more products like your functional food and so on. So some case studies I'll just narrate through these presentations. So going ahead, this is just a normal journey which everybody takes from herb to develop a herbal product through the research. That is, you select a plant or mineral or from plant origin, mineral origin, or animal origin, then authenticate it, then extract it for the required metabolic metabolites, and then you do the toxicity and try to develop the products. So this is just a general, and everybody follows, I think, the same path. Coming to the extraction of phytocosmetics, of course, this is also general. For every constituent, you have to do the trial and errors and extract the constituents accordingly. So some overview of this is given. Alkaloids you can extract by water, methanol, then glycosides also water, methanol. Some terpenoids, steroids, you can get it in organic solvents. However, if you are using organic solvents as extracting solvents, you need to do the residual solvents because these are quite toxic. Otherwise, your extracts may not be accepted. Then tannins and flavonoids, almost the same way. Next, few case studies. This is one of the case studies that I would like to narrate. This all has started with some information in the general magazine. Somewhere I read that the Parijatak, that is Nyctanthus arbotristis, has the constituent similar to saffron, and they are using this as a saffron substitute. However, I couldn't get any reports. So I just carried out the TLC analysis of saffron, as well as you can see here, the flowers, the orange color calyx, we separated and did the TLC. Now we found that the constituent is the same one, that is crocin. Crocin is an apocarotenoid. You can see the structure on the slide, and this is just a published paper in the said journal. These flowers, the beauty of these flowers is that, you can say beauty or unfortunate thing for these flowers is that these wither, these blossom at night and wither the next morning, and they get perished within half an hour. So why not, if they have this potential to substitute saffron, why not to explore this? With this, we started the journey of this, exploring Nyctanthus arbotristis or Parijatak flowers. Later on, we prepared some formula, that is phytosomes. Phytosomes, because that apocarotenoid is highly unstable. So as Dr. Satnam Singh was telling about the phytosomes, liposomes, we did prepare these and incorporated into the gel. Along with that, we also incorporated the Taget spatula, that is the lutein present in the Taget spatula. We did standardize all these extracts, and then we developed the gel formulation. This gel formulation was evaluated as usual for particle size, then zeta potential. Then we did even the antioxidant studies in vivo parameters like glutathione levels. Then further, we explored this for the genetic level for expression of collagen and elastin gene. So when we found that there is upregulation of both these genes, that can be useful for anti-aging activity. We did only cosmeceutical products. So however, if you can give it orally also, it can exhibit antioxidant activity. Next, going ahead with the crocin and lutein formulation. This was the paper that was published in the in vitro enzyme studies, that is collagenase and elastase enzyme inhibition studies. Then we did the anti-angiogenic study because we wanted to explore if it has the anti-cancer. We found that it has the angiogenic study, so it can have the potential to be the anti-cancer agent. However, we got the biphasic response. At higher concentrations, it is anti-angiogenic, and at lower concentrations, it is pro-angiogenic. So angiogenesis has something that both the activities can be used in different situations. Going ahead, we carried out the genotoxicity, whether the Nectanthus flower has genotoxicity. We found that these are all the published papers. Both the Nectanthus flower extract and crocin have been shown to be non-genotoxic. So it can modulate the response of standard mutagen. Then we carried out the subacute toxicity, that is 28 days subacute toxicity profile for the phenolic extract, which is rich in crocin. And it is found that there should be a caution to be exercised with the Nectanthus extract for treatment of the concentrations at concentration of 250 milligram per kg and above when required to be consumed for the period or greater than 14 days. Further, this was evaluated for wound healing activity. Again, the crocin extract was incorporated into the phytosomes, as well as the crocetine, the A glycan of the crocin, was incorporated in the phytosome because there is good amount of crocin in the extract, are about 33%. So we did isolate the crocetine by hydrolysis method, and then we incorporated. We found that it exhibits good wound healing activity. Further, the phytosomes were also evaluated whether they can increase the bioavailability, not only causing the enhancement in the stability, but these could increase the bioavailability. As you can see the diagram here, the levels of crocetine have increased when they were administered to the rats in the phytosomal formulation as compared to the Nectanthus extract. So finally, coming to the journey of this Nectanthus arborestis flowers. First, we established that yes, it can be used as substitute to saffron. Then we did carry out the pharmacological activities, antioxidant, analgesic, anti-inflammatory, immunomodulatory, wound healing, cosmetic utility like fairness. However, we couldn't get any fairness activity for the same, but anti-aging we could get. Then we went ahead to develop the formulation and carry out that anti-aging activity at genetic level. Now, we are planning for development of some nutraceuticals and functional food using this as substitute to saffron so that it can be made affordable to a common man. So this is about the journey of the flowers right from its establishment as substitute to saffron till the development of the formulation, and still the work is still going on and many students have worked on this project. Going ahead with the next case study, we have started with the bark of Onosma ecoides, which is similar to alcana, and this contains a naphthaquinone, and the naphthaquinone that we isolated was a dimer of the naphthaquinone, which is an alkanine or chiconine derivative. And this we carried out this isolated compound itself exhibited good wound healing. Of course, the plant is reported for its wound healing activity, and it is used quite a bit for healing the wounds. So we carried out this activity to know what is the ingredient that is responsible. Another interesting fact about this Onosma equals is that this is called as Ratanjo. Somewhere we read that it is put in the castor oil, and that castor oil, when you apply it on the lips, so it gives glow to the lips over the period, maybe for 15 days or so. So we thought, why not to prepare the lipsticks out of this? So we went ahead with the extraction of the dye, and we tried to incorporate in the lipsticks. But we were finding it very sticky and not appealing product. Hence, then we searched that the NBRI, that is National Botanical Research Institute, has filed a patent by preparing lipsticks of this particular bark. So then we were thinking what novel we can do, and we were finding that it is not a very appealing product. It is very sticky and not releasing the dye properly from the base. We converted it to the lakes using the bhasma preparation. As you can see, the dye is naphtha phenol.
कुछ बता रही हाँ
Yes.
APT
Yes. Can I go ahead? Okay, so this particular we prepared the lakes out of this dye. The dye we had already isolated this constituent, and we standardized this dye. Even we standardized the bhasma preparations, and we adsorbed the dye on the bhasma. So we found that the dye contains a naphthaquinone, and these all quinones have the ability to change their colors when they come in contact with alkali or alkaline earth metals. So we could get different shades, as you can see, the lipsticks with the different shades when we treated with different bhasma preparations. Then where we incorporated this into the lipstick base, we found that the lipstick base requires only little amount of dye or the lake to release the color on the skin or the on lips. So then we filed a patent, and we were granted the patent. We filed it in 2006, and we were granted in 2009 this patent. The dye can also be used for preparation of other cosmeceutical products. Why am I calling it as cosmeceutical? Because this does have the wound healing activity. If you have any cracks on the lips, then it can also be useful for the same. So therefore, it has some medicinal activities, especially related to skins. So we can call this to be the cosmeceutical product. Further, we thought, why not to use this for coating the tablets? So we prepared these two shades by treating it with the plasma preparations, and these lakes, then you can use the aqueous base for preparation of the suspension, because these are the lakes, and industry does prefer the aqueous bases or solvents rather to your organic solvents. And we found that you can very well use it for the coating of the tablet. However, this dye or the lake is not useful for drugs which are basic in nature. Another story about development of the or designing of the apparatus for extraction of volatile oil. Generally, the conventional steam distillation or Clevenger's apparatus, everybody is aware, looks like this, that is on the right hand side. So what happens is that we load the plant material in the round bottom flask, and then we add water or steam you put, and then you distill. While loading it is very easy, but to take out the plant material after the extraction, it becomes very difficult to take out. So hence, one of my students, out of this difficulty, she designed this apparatus, wherein what modification she did was, this is a round bottom flask, there is a frit over here, and there is a roll of the stainless steel sheet, which is the perforated. And in that she packed her leaves and she just inserted. Again, this all can be dismantled. And this is also frit. So she packed it in this and water was added in this. There is outlet or there is an inlet, you can say. You can pass the steam or you can heat the round bottom flask as it is. The steam passes through the, because it is a frit or it is perforated, the steam passes through the leaves or the material that is packed in the roll of a stainless steel sheet. And then there is also frit over here. That steam carries the volatile oil or the hydrosol, or you can say azeotropic mixture, and it goes to the condenser. And this part is similar to your Clevenger's apparatus. By designing this, what we found that it is not only that the leaves can be or the material can be packed and unloaded easily, but the quality of the oil was also better, the quantity was better. So we filed the patent and we got it granted in the month of May 2022. Now what she did further, she took that oil, and this is the oil that she got, and she got this, the beta caryophyllene through the column chromatography, and she did the anti-angiogenic or angiogenic activity. And this is the untreated, and we found that the oil does have the anti-angiogenic activity. The next case study is of linseed formulation. When we thought of preparing linseed formulation, we thought of preparing the formulation for the content of SDG, that is seco-iso-laricy-resinol diglucoside. It is a lignan which is useful for treatment of postmenopausal problems and other. It has many activities. It's a very good nutraceutical apart from the linoleic acid and other constituents. The ethanolic extract, after you defat this linseed, you get ethanolic extract which is rich in this SDG. This is present in the form of polymer, and when you hydrolyze it, you get SECO, that is seco-iso-laricy-resinol. This A glycon was then appears in the blood, and this then gets converted to enterodiol and enterolactone, which has phytoestrogenic activity. So what we did, we isolated the SDG by hydrolyzing the polymer, and then we standardized the extract for SDG, and then SECO was isolated by hydrolysis, and then it was the formulation was prepared. It was evaluated for all regular parameters. It was just a capsule formulation which was granulated and then filled in the capsule. We did the standardization of the capsule for SDG content, and then we did the pharmacokinetic studies. There is a mistake here. There was some mistake in preparing the graph, so please excuse for that. It should be 0, should start with 0. So you can see we found out the absolute bioavailability of the SDG, that is with respect to SECO, and then the SECO, the bioavailability of formulation. You can see the bioavailability of formulation is better as compared to the pure SECO when it was administered. So what Dr. Satnam was telling ahead, that the extract gives better results as compared to the pure constituents, which you can make out here. So these are the case studies which I wanted to narrate. And the best way to get something done is to just to begin with something you will definitely get. And thank you for your patient listening. Thank you.
Thank you, ma'am. Thank you so much for your presentation.
Okay, should I stop sharing?
Yes, ma'am, please.
How to stop sharing? Stop sharing. How to stop sharing it?
Ma'am, I will be stop sharing your screen. It's fine.
Okay. Okay. Okay.
Uh, Participants, is there any questions for ma'am?
Yeah, ma'am. Good morning, ma'am.
Yeah, good morning.
It is about some of the technical doubt. No? See, this patent is that we can apply through online.
Yes, it is available online.
So to establish, to introduce any things which we are observing from the traditionally from the childhood.
Pardon?
From traditionally, we are observing so many herbs,
Yes,
some of the tribal areas and some of the people,
yes,
what is known as traditionally they are giving some medicines using all these. We to make patent, we can go through online.
yes,
We,
yes. Online we can go, but this was filed when there was no online facility. So it is manual, but in Indian patents website it is available.
manual means we need to get from Delhi only.
No, Mumbai.
Mumbai.
Yeah, we filed in Mumbai only. However, you will get
Every,
any office, no, no problem.
every state has its own patent.
Yes, yes, sir.
It comes under which department?
This is from pharmacognosy department, sir.
Yeah, ma'am.
Pharmacognosy related to natural. Pardon, sir?
Pharmacognosy related to?
Yes, pharmacognosy that is related to natural products.
Every state has its own board.
There is a question from the chat box, ma'am.
Yeah.
Is there any other method for extraction of essential oils other than steam distillation?
Yeah, there are many methods available. Supercritical fluid extraction is there. Then your hydrodistillation is there. Then UCLI method is there. Then enfleurage method is there. Enfleurage is generally used for flowers. UCLI for citrus peels. These are all specific methods for a particular type of the plant material. And another is your hydrodistillation. If you have a dry material, you can do it by hydro. Supercritical fluid is also there.
Okay, ma'am, thank you. And the other question is extracting the active ingredient is mandatory to prove the properties of the plant and seed?
Yes, it is mandatory to prove the properties of the plant as well as for standardization of this. And when you extract active material, you are enriching your extract. And that enriched extract the dose can be reduced and it will give better activity as compared to your the full plant material.
Thank you, ma'am. And the other question is for patent. Should we have scientific proofs and experiments done to provide its properties?
Yes, 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 you do need. If you have any idea, you can file a provisional one. And after 1 year, you get 1 year to file a complete specification after doing your experimentation. But you will have to complete that in 1 year and file it. Because you may think that somebody else can file that patent, then your idea may go waste. So you can do that, provisional you can file and then complete the experimentation.
Okay, ma'am. Thank you. Other question is, have you done anything for the standardization?
Yes, we have done, that is isolation of the active marker or the marker compound. Like in case of onosma, we isolated that dimer, naphthalene dimer, and we standardized our extract with respect to that content. Then for crocin extract, we isolated crocin and standardized the extract with respect to crocin content. Using HPTLC, mainly we use HPTLC. HPLC also you can use, but HPTLC has greater advantages as compared to other.
Okay. Lavender oil is good for skin, but it causes skin irritation. What can we do?
You can prepare this novel drug delivery system and utilize that in the low concentration.
Is intellectual property rights, IPR, different from pharmacognosy for patents?
Yes, slightly different, because if you want to patent activity, you cannot patent. But the product which has enhanced properties, you can patent that. and you have to take you have to give in writing or get the permission from National Board of what do you say then? National Board of NBA, that is National Board of Authentication. I think that is situated at Chennai. You have to take permission from them and state that it is not from textile category. If they permit, and you have to share some profit if at all you commercialize your patent. So that MOU you have to sign with the NB, not accreditation. It is not accreditation. It is something else. National Biodiversity Authority, sorry. National Biodiversity Authority, it is not accreditation.
Okay, ma'am. Thank you. Kindly drop your mail mail
Okay,
ID
in the chat box, I'll just drop it. Yeah.
So
I'll
that if
just drop
any
it.
more questions, they could contact you.
Yeah, definitely. I am dropping that mail.
Okay, ma'am. Thank you. Thank you all the participants, and thank you, ma'am, for the informative session. Now,
Thank
next,
you.
next, I would like to move on and to welcome our keynote speaker, Dr. Piyush Juneja, founder, Indian Vaidyas India, to deliver his keynote speech. Sir, we welcome you.
Right. Am I audible?
Yes, sir, you're audible.
Just a second. Let me just check my presentation as well. Just give me a second.
Yes, sir, sure.
Hello.
Yes, sir, your screen is visible, sir.
Yeah, I think there is a power cut. Just a second. Yeah, fine. So is it visible now?
Yes, sir, it's visible.
Okay, fine. So first of all, thank you so much, the organizers, the entire team. I think I got connected with Karthik from your organization. And I will not take much time. My presentation is very brief and crisp. However, there are certain ideas and there are certain entrepreneurship thought process which I will be giving in today's presentation. And I request all of you who are listening to this session today. See, we have excellent speakers, as I was listening to a couple of speakers prior to my session, in terms of the technical knowledge, in terms of the like scientific, technical, research, pharma, all those. We have excellent speakers yesterday and today, and you can gain tremendous knowledge from all of them. You can be in touch with them. You can take it forward in terms of any aspects are there. However, there is one very important aspect which I want to discuss here. I think predominantly the pharma fraternity and the pharma community is right now listening to this session. And as we see in IITs, in engineering, in IIMs, in management, and many other domains, people have this kind of these days thought process of being an entrepreneur, starting a startup, maybe just after the college or during the college or after some years of job and all. But all of them have that thought process and mindset to start. However, in pharma, in medical, in herbal, in all these domains, I have seen very less people taking up and in fact non-pharma, non-medical, like scientific people generally do start these kind of startups, which is also good. However, if people like you, people like pharma background and experts can get involved, that can be an added advantage. So on that note, I want to start my session today. I'm Dr. Piyush Juneja, Dr. Piyush Juneja, founder indianvadyas.com and hempvadyas.com. Basically, it's a portal where we have enlisted either the doctors across India. Like it's an aggregator platform. We have more than 1 lakh Ayurveda doctors, and hemp is our upcoming portal for medical cannabis and hemp as well, which is again growing. So let me just quickly share some innovative ideas. In fact, it's some aspects are already in process, or maybe some people are doing some aspects on that, but not in that way, which I will be discussing today. So maybe any one of you, my idea is any one of you. I request all the other people to be on mute to avoid the disturbance. Thank you. So, or maybe the host can mute all the other participants so that we can have the session without any disturbance. Thank you. So the idea was to discuss this, and the idea was to present this today to ensure that all of you will get a thought process and a mindset that how we can take it forward. For example, in these 10 innovative ideas, the first one is digital herbal mandi. So we already have, I think, a couple of startup, but not at that much scale. And the idea is we have so much of herbal cultivators, we have so much of like raw material suppliers across the country. However, there are very less aggregators or platforms like having like, okay, so somebody wants to purchase, some company wants to purchase, some researcher want to purchase. I get several queries from the research, from the pharma people, from like people who are doing M-Pharm or PharmD. Sometimes they need herbal raw material to for their research, and many other researchers also. The companies, the manufacturing companies, the I request again to be on mute, please. Thank you. If can host can mute the participant because there is some background noise coming up. Hello. Can I request host? Am I audible? Hope I am audible. Yeah, thank you. I think it's not there. So as far as digital herbal mandi is concerned, we need to understand that we can aggregate the entire ecosystem of people who are cultivating, who are having raw material herbs, and they can connect to the companies and they can connect to the respective buyers as far as digital herbal mandi is there. Then second is marketing of native cultivation. Means what happens is we have so many native cultivator in all over India. In maybe your state or your city, you will see so many medicinal plant cultivators are there, but we don't have any kind of a mapping of them. So if you can start from your city level or state level for that matter, an interactive map that somebody will click on the map and they will check, okay, these people are there who are cultivating this particular district. It has to be started at a ground level from the maybe district level. There are so many villages in any district having so many native cultivation of the medicinal plants, and when you interact with them, you will get that knowledge and insight, and you can put that on the map. Third is medicinal plant and herbal identification blueprint. So what happens is many times, being an Ayurveda doctor also, I have faced that challenge, and many other people are facing this, that they're not able to identify the raw material sometimes, or the herb or the medicinal plant identification. Although we got this training, we got this know-how, we got this like as a subject, it was there in our colleges. So but somehow all of us are not able to identify all the medicinal plant and herbs. Some people from botany can do, some people from like in pharma, I think you people have that subject in which you understand and read about these herbs. I think cognosy is there, so you can get that understanding in some way. But yes, if we have experts who can identify, and we can create some kind of a digital tool or a blueprint or a kind of an app, mobile app, in which we can just scan a particular herb and we can get the name or maybe get the details. So that can be an excellent tool, because in our nature, in our day-to-day life, wherever we live, we have so many plants around us, but sometimes we are not able to identify the potential or the benefit just because of the lack of knowledge in terms of identifying that plant. In fact, that can be an excellent app or tool for the masses also. So that is one more idea which we can thought. And in fact, all these ideas which I am discussing right now, anyone of you want to take up, we can help you out. Our platform, that is Indian Vidyas and Vidyas, are there to help people to grow in terms of business, in terms of, we are already working with several professionals. So all of you can just note down any idea which clicked to you, and you can let us know. I will share my email ID, and we can have a detailed discussion after this session to implement and to execute and to help you to take it forward as a particular business also. Then digital herbal education again. As today I have got an opportunity to see, and excellent session by both of the speakers from prior to my session. I have seen their session. So they have excellent knowledge in terms of herbal education, the pathways, the physiology, the pathology, the way the herbs are interacting inside the body, the way the herbs are treating the disease, the way the herbs are getting involved with the mechanisms in our body. So all these things are there. Any drug interaction, any side effect, by the way, sometimes we do see side effects also of the herb. So all this can be there with the community, with the professionals, with the doctors, with the scientists, with the pharma people. So that kind of education and that kind of consortium is very much required. In fact, on that note, this particular event which is going on is an excellent event. I think you people have done an excellent job to compile and to create a platform where all the experts are together discussing on this particular aspect. It is one of the first, I think, innovative event which I have seen in my last 16 years. Then herbs properties in various systems. Look, for example, we have seen in Ayurveda, there are so many herbs which are mentioned in Unani system of medicine, homeopathy, Siddha, and phytopharmaceutical is a separate domain. You have certain properties which are mentioned in scientific data as well. So when we compile, if there is any tool which we can compile all that information, we can get plenty of additional source of information, plenty of additional like information in that compilation. Because see, sometimes what happens is we know a particular benefit from a particular herb, but when we compile and when we study that, okay, this system is saying this system is saying next about this herb, about the property, sometimes we can get some additional benefit also, which maybe we are not aware right now if you are following just one system. So ultimately, end of the day, the idea and the thought process is for a particular herb, for a particular plant, if there are n number of benefits which has been documented, quoted, and sometimes being researched, or if not research, we can always do the research as well. So I think in that note, we should develop this kind of a platform as well, where all the system benefits can be there for a particular herb. Somebody can click and get all the details. In fact, all these ideas are very much business viable as well, by the way. So you can earn a good amount of money and you can grow financially also whenever you launch any of this kind of app or a platform to start with. Now the next stage is, for example, there is one more issue and one more dearth, or maybe say one more bottleneck, that when we talk about medicinal plant or herbs, so the cultivation training is very less to the farmers. Sometimes they are locally connected and they are locally having that grooming or understanding, but sometimes or many times we have found that still there are challenges and still there are issues in terms of plant cultivation or herb cultivation, and that training and that orientation to the farmers is very, very crucial. And now 4G, and after 4G, 5G is also coming up, so we can have digital training sessions, we can have digital online sessions, and all the villages, till the villages high speed internet is connected now. So it is very much feasible these days to create this kind of a training, and definitely offline also can be added to that. But first, if we can do online, or maybe some small videos, some DIY videos, or some like very, very simple training sessions in their vernacular languages also, by the way, which farmer can understand. So that is very much required, because I have seen farmers are willing to grow, and this is a very important segment. We can't ignore them when we are talking about herbs and herbal domain in our country, or any country, by the way, for that matter, because ultimately, end of the day, farmers will grow the herbs, farmers will grow the medicinal plants, and then we'll be able to take it forward. So the cultivation training to the farmers is missing, and sometimes due to the lack of that, they are unable to cultivate and they are unable to grow financially also. And there are so many aspects of this training which we need to understand. Then next is hemp industry. Hemp industry is growing at a very fast pace. Anyone of you is aware about hemp is a plant which has more than 100 of properties. It is used in nutritional, it is used in cosmetics, it is used in medical aspects, it is used many ways, fabric, industrial, hemp fuel. I think in our country, I have seen one video last week. First kind of its experiment people have done, that they have started preparing hemp fuel from this particular plant, and they have done some kind of a trial as well. In entire world, the research is going on. So we can consider this particular plant, this particular herb, this particular domain as a very serious one, because in the entire world it is growing at a very fast pace, and in our country it has a potential of growing huge as well. So any one of you, we are working on several aspects in this particular domain, and any one of you want to interest, having an interest, we please connect us and let us know. We can always help you out in this as well. And there are so many like in fact, if I start talking about this particular industry, in fact there are so many innovative ideas we can discuss and we can take it forward, because this is at a very initial stage in our country. Next is herbs as food. Very, very important point. See, sometimes we need to understand that so many herbs and so many medicinal plants are also considered as a food. In our kitchen also, if we see, there are so many herbs which we are using in day-to-day life as a food also. But exactly the benefits and sometimes the overall scenario in which how much quantity, how much okay, the frequency, the duration, or sometimes what can be taken as a food, what cannot be taken as a food, because there is a very sensitive like aspect to all the entire discussion which we are doing today. We need to understand as a herb, as any herb or any medicinal plant. On one side, we always say that it does not have any side effect, but on the other side, there are so many herbs which are not, if taken under medical supervision, can do side effects as well. For example, when we talked about hemp, it is under schedule even in our country, means only to be taken under medical supervision, not to be taken without prescription or medical supervision. That is mandatory. And so many other herbs and so many other plants are also like this, which our regulation is saying. But many times we have seen, due to the nature of this domain or maybe due to the business interest, many companies and many people are promoting directly to the consumers, to the patients, to the people who can take, without involving the medical field or the doctor ecosystem in this. And then there is a problem. In pharma also, there are people who are trained on allopathic pharma side, but there are very less resource in our country. In fact, we want more and more pharma people to get trained on herbal and these kind of aspects, so that they can also take care, because in allopathic, the pharma fraternity is taking care well in terms of the drug dispensing, drug giving counseling to the patients, how to take it, how to what are the side effects, or what are the main compliance aspects, or many aspects are there. So that domain is missing in herbs or in Ayurveda or any other system of medicine, which is Indian system of medicine, and predominantly using herbs as the treatment modalities. So that we need to understand, and we need to differentiate that, okay, which can be taken as a food, which should not be taken as a food, and so on. So that is very important. And I think if some kind of an app or some kind of a platform is there which can guide people, in our country also, in FSSAI, there are certain herbs which are listed as Ayurveda or food also, but not all. So that is very important. Then international herbs. So many times we have seen, when we are talking about herbs or when we are talking about medicinal plants, predominantly Indian herbs or Indian plants are being discussed many times, several times. Although India is a herb and India is a leader in terms of natural cultivation, however, China and other countries are way ahead of us in terms of the business, which is huge in terms of the herbs and medicinal plants are concerned. And China is one country, or maybe some other countries, they are very smart. They have adopted and they have infused all the other international herbs also in their country along with their native herbs, to understand and to take it forward in terms of growing and emerging as a leader in terms of the herbs or medicinal plants or herbal market is concerned. But in India, it is very lacking, or it is very at a very primitive stage, that many of us are like, we are not having the basic knowledge of international herbs. We are not having the basic knowledge of taking it forward in terms of a business or in terms of a domain as far as the international herbs are concerned. So that is also very, very important, and we need to understand and take it forward in terms of getting the entire benefit. Because see, ultimately, end of the day, if you talk about this era, this is the era of globalization, and when we are doing globalization, the demand of herbs and the demand of medicinal plants is increasing day by day. And internationally also, if we are aware that what kind of herbs are going on and what kind of herbs are there in demand, and if we focus on that as well, and we cultivate, or maybe we see that in India, because we have a vast dynasty and we have a vast range of states and the climates and various different situations and environmental aspects are there, in which easily we can at least cultivate most of the international herbs which are there. But still, yes, maybe there are some challenges for some herbs, but not for all. But if we thought, if we give a thought process on that, that can be a very good and excellent tool for all of us. And the final thing is the herbal research encyclopedia. See, what happens is, whenever we talk about herbal research, whenever we talk about any R&D or any research, in fact, in our country, and I give credit to pharma fraternity specifically, and because I have worked very closely from last 16 years in my career, initially in the clinical research and many other related domains, I have seen that pharma people are doing excellent efforts in terms of research, in terms of creating certain footprints of how the herbs are working, the mechanisms, the research in terms of the overall plant, and then the physiology and how they are working on the pathology as well. Preclinical studies, many times we have seen, are being done in the pharma institutes and the universities. So that encyclopedia, that kind of a compiled knowledge or a compilation is required, because whenever we want, we can go for any platform, that is fine in terms of doing the search or in terms of doing the like overall homework of how we are mapping our research knowledge. But if there is a dedicated platform, if there is a dedicated encyclopedia, we can always quickly click and see the entire results, at least for Indian scenarios, or maybe add on in a next phase for the international research. So that is very, very required these days, because that will help in many aspects to the researchers, to the clinicians, to the entire fraternity who are there involved in this particular domain. So I just thought to share all these 10 ideas today with all of you. Hope all these will make sense, and any one of you wants to take it forward, any one of you wants to ensure that yes, herbs are actually my concern is, I have been into various sessions previously as well, and again in many fraternities there are certain or other challenges also, but specifically to pharma, I have always been like we are always getting very good response. People are very much having a thought process of doing certain kind of innovations, or maybe a thought process of adapting this and taking it forward. So most of you are I think looking forward to start your careers or professional life, I guess, as a student. And if any one of you have any kind of a thought process, please implement as soon as possible, whenever is possible for you. The only issues can be the fund sometimes, or maybe the execution aspects, or maybe you don't know how to take it forward, how to start. You need some hand holding, mentorship, guidance in detail. We are always there to help you out. If you are ready to work, you can kind of start. I don't know, maybe DY Patil is a big brand, so you must be having incubator in your university, in your setup. But if not, or if in this segment you need, we can help you out in that as well. And incubation is required for at a student level. We can get some seed funding, and accordingly you people can start doing action on these, any of these, or maybe any other idea or any other domain you want to work on in terms of the herbs and medicine plants are concerned. My only request is, my idea, my intention of doing this session today was not just to just present a session. It was just to trigger some thought process in all of you who are listening to this. And as I can just quickly brush up the session. So these are 10 ideas which I have shared, and all these 10 ideas I request any one of you can take up, and we can help you in many aspects in this. But yes, let's start, and let's start some action, because it is very much required. If we can ensure that each one of this particular aspect can be taken care of in terms of either a platform or a mobile app or a software or whatever it can be, it will definitely help. Some of them are already there, but in sometimes at a very initial stage they are. So any one of you know, and any one of you is already doing something in this, please let us know. We can market and we can propagate and disseminate that to lakhs of followers and lakhs of people who are connected with us. Recently we did an analysis, more than 35 lakh people are connected with us through various platforms. So we can always propagate and we can always disseminate that as well, if you already are involved, or if you know somebody who's doing any of this aspect. But yes, if not, let's start
Sure. Dr. Piyush. Dr. Piyush,
Hello, am I audible?
yes, sir, you're audible.
Yeah, sorry, I got disconnected. So hope session went well, and maybe we can have queries now. Thank you.
Yes, sir. There are questions from the chat box, sir.
Right.
Please
Actually, I got disconnected, so maybe you can just post again, or maybe you can just read because I'm not able to see. Thank you.
Yes, sir, I will be reading out the questionnaire. Sir, will you help in case of licensing process if required for startup, for herbal production and or formulation?
Yes, we can help in that. We are already helping several organizations and several individuals as well, so we can help that. That is not an issue.
Okay, sir. And there is other question also. Please plant useful for Vidarbha region to cultivate. I have a company want to do work for elimination of farmer suicides of Maharashtra state and want to run AYUSH plant cultivation for production of AYUSH use medicine.
See, actually, that is the issue which I was saying. I have shared my email ID and mobile number also. Hope it is visible in the chat box. So that is an issue. Like we all know that in our country, in Maharashtra and other states also, farmers are in a very bad shape, and many of them do like this kind of suicides, and all these issues are there. So I request whosoever has raised this query, please let's connect as soon as possible, because we are already helping certain organizations and certain like groups, and they are working with the farmers, and we have experts, we have cultivators, we have people who know how to cultivate and take it forward, and they can give training to these farmers also. So let's quickly connect and take it forward, because we are doing at a very initial level right now, but we want to scale it up, and that's why that was the idea of putting this in this slide also, that we need to groom and we need to train. And first of all, we need to do an orientation. Sometimes we have found that farmers are already trained and they know how to cultivate, but the problem is they are not aware about who will purchase and how it will be purchased, and how like the returns will be there. And with this new farm laws in our country, I think that was a very positive step, that any farmer can connect directly to the company, and they can do a contract cultivation also, which is also possible now. So let's take it forward, and we are very much ready to help any these kind of issues which are there at farmer level, because they are the backbone, and we need to ensure that they are stable, and they are there, and they are sustainable, so that we can get the maximum benefits from their cultivation.
Thank you, sir. Thank you so much. I guess there are no more questions for you.
Sure. Thank you.
Thank you, sir. Thank you so much for your informative presentation. Next, I would like to welcome our next keynote speaker, Dr. Renu, Deputy Secretary, Department of Labor and Employment, Government of Haryana, India, to deliver her keynote speak. Ma'am, please.
Good morning, everyone. So, yes, is it visible?
Yes, ma'am, it's visible.
Okay, it's 10:35. I have to finish before 11, right?
Oh, before 12.
Oh, sorry, before 12. Yeah, yeah. First of all, I would like to congratulate to the entire team for organizing such a wonderful conference. And I was actually looking forward to visit Mumbai also, but somehow it has been converted to webinar. So I'll start my presentation because the title of this presentation, this conference is Journey from Innovation to Entrepreneurs in Herbals. So basically, I will sum up my presentation, which will only round about the such things, how it's an innovative approach in minimizing the gap between research and industrial production of AYUSH drug. There is actually a gap. People are doing wonderful research. They are publishing the papers. Everything is going on fantastic. But when it comes to the industrial production, when it comes to the production of medicines, there is a gap and there is some lacking, because one of the speaker also told that the regulatory aspect of the herbal medicine is very it's not good in our country. So I will focus on that also, how our country is going to enhance such things. So myself, Dr. Renu, I'm working as a Deputy Secretary in Department of Labor and Employment. But I am a former research officer also in the Ministry of AYUSH, Government of India. I worked there for 5 years. And I was heading the specific section that was drug standardization, where I was working on the entire AYUSH department, like Ayurveda, Yoga, Unani, Siddha, and Homeopathy. So I will well verge to the our theme of the conference. So here I'll start my presentation. Like herbal drug is a traditional medicines, because now, as you all know, there are only two kind of medicines nowadays. It is hot topic, the conventional medicines and the traditional medicines. See, the traditional medicines covers so many alternative, adjuvant, and complementary medicine. It is an ancient and cultural bound practice in every human community. Everybody basically, it has come from the folklore medicines, or the some nowadays it is known as a tribal medicine and folklore medicine. But in our ancient, in our Rig Ved, in our Charak Samhita, everything is mentioned as a like Ayurvedic and plant medicines. So what I was telling, that when we use the traditional medicine, there are three types. One is alternative medicine. When we use in place of conventional treatment, then it's known as a alternative medicine. And when if we give the herbal or the plant medicines along with our complementary medicine, that is a adjuvant medicines, and we integrate those that conventional medicines with this herbal, then it is known as a integrative medicines. Now the country, or the all over the nation, is facing such challenges, which is also known as a disease burden in case of non-communicable diseases, where 71% of the deaths are globally due to the communicable diseases. It is apart from the epidemic or pandemic time to time, which is going on in case of COVID, in case of SARS, in case of MERS. So this is the integration of traditional medicine with modern medicine. So now people are actually looking forward for the harmless medicines, which is a traditional medicine. In such case, the side effects are in at the very lower side. That is why a huge emergence and a huge demand has been come up basically during the COVID times. Everybody, you might have seen, you are around, everybody was taking ashwagandha tea, giloy tea, everybody was taking ayush kadha, everybody was shifted towards the natural medicines. Now, because it is a growing interest in the traditional medicines shown by consumers, when you have the interest, when you develop the interest, then there are some responsibilities and challenges also are there. And there are three important challenges at present. First of all, the public and the user of traditional medicine, everybody is requesting for the safe, quality controlled, and effective medicines. This is the first challenge. And second challenge is the scientists, because all over the scientists initially they are very skeptical towards the plant medicine, because they think there are less scientific data, there are less effectiveness, there is less reproducibility kind of science. And the third point is government need the updated mechanism for the regulation of traditional medicines, how these are being cultivated, how these are being manufactured, and how these are being sold, because there are a lot of misleading advertisement you can see on the TVs or on the newspaper. There are a lot of misleading advertisements. So government need to regulate all these things. So for that, Government of India had announced a separate body, namely Ministry of AYUSH, to encourage the traditional system of medicine and to set up the set of standards of the traditional system of medicines, which covers the Ayurveda, Siddha and Unani drugs. So it comes in the existence in the 2014. Earlier, it was considered as an Indian system of medicine and homeopathy. Now it has been converted to AYUSH, which covers the yoga, naturopathy, Unani, Siddha, homeopathy, and Sova Rigpa also. When we talk about the herbal medicine, it is not only the Ayurveda. There are other medicines also. Like if I talk about the Siddha medicines, people in the Chennai, they must relate to the Siddha, because utilized and being faith in this, specifically in the Tamil Nadu. Sova Rigpa is a system of medicine which is being used in the Tibet and Sikkim area. So all consolidated, it is a AYUSH medicines. So what people are think, if we are talking about the herbal medicine, it is only Ayurveda. No, but it is not like that. Ayurveda nowadays Ayurveda and that complement conventional medicines are more or less same, because mostly all the chemical constituent active pharmaceutical ingredient, you can say like cinchona from the chloroquine from the cinchona, and the digitalin from the digitalis. Now this being synthesized and manufacturing as a conventional drugs. So now the legal aspect of AYUSH medicine, which is very important, because until unless any medicine will be regulated legally, that can be treated as a illegal medicines. So in Indian legal system, there is a legal provision regulation under section under second schedule and 4A under the Drugs and Cosmetic Act, which is a chapter 4A from section 33B to 33O, which is regulating entire this medicinal plant, initially from the definition, initially from the cultivation, their identification, their adulteration, their phase 1 trial, their phase 2 trial, everything, so that the scope or the vision of the this AYUSH Mantralaya was basically to set up the standard of the AYUSH medicine, so that they can become the at par to the conventional medicines. So this is the hierarchy. Initially it was only a small department of AYUSH, then it become the ministry. Now this ministry is also working on the developing education, research, propagation of indigenous alternative medicines, improving the healthcare access. As you know, during the COVID time, Ministry of AYUSH has distributed the AYUSH Kavach, AYUSH Kwath, and Haldi Doodh. Everything this has been propagated. This has been awareness to the people, so that giloy, to take giloy in your tea. So such things, it is inherited in our Indian culture. So during this COVID, this has been immersed, and you might have known also, you might have aware also, that during that period the production of ashwagandha, the production of giloy was accelerated like anything. The export of these herbal drugs was also escalated like anything. So considering all these, Government of India, Ministry of Finance announced the 2000 crore rupees for the fund, basically for the cultivation of the AYUSH plants to farmer. In my last previous speaker, someone had asked the question, like if the farmer wants to cultivate AYUSH medicine, what should they do? So there is a organization NMPB, which is known as National Medicinal Plants Board, which comes under the Ministry of AYUSH itself. They are providing the buyer sellers meet who will buy, who will sell. If a farmer will produce the ashwagandha, or if a farmer will produce the giloy, then who will buy, which pharmaceutical country will buy. So they are providing a platform where the farmers and the buyer seller meet they used to conduct, so that a farmer need not to run here and there, that my take my product, I had produced such things. So this kind of activities already is being going on and carried out by the government of India. I repeat again, it is a National Medicinal Plants Board, which is known as NMPB, working under the Ministry of AYUSH itself. Also, this NMPB is providing more than 100 crore rupees of project, of the scientific project. Many of our listeners who are participants, many must be from the student, they must be from the research. So they can submit their proposal to NMPB also in ways of pharmaceutical pharmacognosy, phytochemistry, pharmacology. They can do the project, they can do take the funds from that. They are giving the project, they are giving the funds more than 100 crores. So everyone, please look into that. And if whoso are try to work, try to research, or try to become an entrepreneurship, they are also providing such a platform also. Now, what is the industrial, what is the gap, and what is the journey from the innovation to entrepreneurs in herbals? Like, which is, what is the innovative approach in the minimizing the gap between research and industrial production? See, when we do the research, we have to do a set of protocols, set of standards. Then if we come up with such a data, then there will be very easy process that you can go for the production of AYUSH medicines. So this is the introduction and country perspective. Like, traditional system of medicine has become significantly more popular all over the globe because of the effective and curative nature of the chronic disease with less toxicity. So since India is the mother hub, so everybody is keeping eye on the India, and we have to work on this set of standardization. Means we have to standardize the medicine in a prescribed set of standards of inherent characteristic, constant parameters, definitive qualitative and quantitative values, efficacy, safety, and reproducibility. Reproducibility, it is a very important aspect, which is for this herbal medicines or AYUSH medicines being criticized, because this reproducibility, to set the reproducibility is a very challenging task in the case of AYUSH medicines. Now, in the process of developing, agreeing upon the technical standard, specific standards are worked out by the experimental. There is a document of WHO, quality parameters. We all are following, and government of India, everybody is following that WHO document. So there are a set of standards which everybody, herbal medicines are used to do this. So standardization is a tool in the quality control process. So for every AYUSH plant medicine, this set should be this set is a mandatory initially from the identification of the drug, because there are a lot of adulterative medicines, there are a lot of adulterative plants, they are same genus, same species, but the chemical constituents may be different. I'll give you an example: digitalis. If you collect the leaf of digitalis in the first year of growth, you will find the very less quantity of digitalin or digitoxin. If you collect the plant in the second year of the growth, then you will find the valid quantity and most of the quantitative of the digitoxin and digoxin. So such things need to be taken care of. Now, the important criteria for quality as per pharmacopoeia, because everybody, because in our country there are Ayurvedic pharmacopoeia of India, homeopathic pharmacopoeia of India, Siddha pharmacopoeia of India, and Unani pharmacopoeia of India. All are being followed by the WHO itself. So everybody has some common parameter like identity. First of all, if our plant will be the genuine, then all the process will be beneficial. If our plant is not genuine, the entire process will be go in the vein. Then purity, our plant should be, because nowadays arsenic and limit test, in fact IP also introduced some kind of herbal pharmacopoeia set. There is a separate column in Indian pharmacopoeia for the herbal. They are specifically focusing on pesticidal residue, toxicity, and arsenic and heavy metals. So this is the major concern nowadays. Now the content of essay, levels of specific phytochemicals, which is the key key importance for the industrialist for the production of AYUSH plants. Now the safety studies assure better saleability of the drug. People are saying yes, Ayurvedic and plants don't have any side effects. It is not like that. Because of the plant, because of the soil, there may be some arsenic, there may be some cadmium. So there it is important. It is equally important to take the safety studies. And in the last, it is efficacy. For wider acceptability of AYUSH drug, there must be some efficacy study should be there in case of preclinical or pharmacological studies. So now the rationale why these studies required, I guess I had already explained, like for the purity, for the safety, and for the efficacy of the drugs. Now this is the technique of herbal extraction. How we come up with the extractions from the plants, because whether it is Ayurveda, whether it is homeopathy, whether it is Siddha, whether it is Sovarikpa, everything come from the plant. The thing is, after that they diverted in some of their route of administration or something else. But the technique of herbal extract, in homeopathy it is known as mother tincture, toxicity, in a potency. In homeopathy it is known like that. In herbal, in our conventional, it is known as extracts, and that alcoholic extract, and we will give the successive filtration. So this is water extraction, conventional technique like dry herb, fat soluble component, heat component. These are the basically techniques by which we can come up with the herbal extraction. These are non-traditional extraction like headspace trapping. There are total 12. Microwave distillation, controlled instantaneous decomposition. So there are some techniques. Now the journey from innovation to entrepreneurship in a herbal areas. First of all, we have to come up with the phytochemical bioassay guided fractionation. This is the very important point, because from plant we used to do the fractionation depending upon the solubility and as per the chemical. First of all, we'll go for alcohol, then chloroform, then hexane, according to their polarity. And as per their polarity and as per their solubility, different compound will be come out from the fractionation. So which goes to the bioactive compounds, and this goes to the phase 1 to then metabolism distribution. If we go for the in vivo study, before that standardization is a very important aspect. And nowadays some few innovative techniques are there, like microscopy, DNA barcoding, TLC, NMR, and MS. DNA barcoding, initially it was for gene library, now it is for the plant DNA barcoding also. We need not to go for entire work. If we have this DNA barcoding, we can come out with the areas of the plants also. Now the schematic representation of herbal drug development. First, it will go for the selection and collection of plant. Second, it will go for, it is a consolidated part of research and production also. So selection, then standardization, then extraction of plant materials, and after that the standardization and quality control with respect to physico-chemical properties of the extract, and then evaluation of biological activity and other various kind of activities. So these are the various pharmacopoeia of AYUSH medicines: Ayurvedic pharmacopoeia of India, Siddha pharmacopoeia of India, Unani, and homeopathic pharmacopoeia. Everything is available on the website of PCIMH. PCIMH is Pharmacopoeial Institute of Indian Medicine and Homeopathy. PCIMH, you just go and log in. You can available all the data available on the there. If you want to do any research, if you want to do any literature survey, if you don't have the data, you just log into the PCIMH. They have every kind of pharmacopoeia over there. Pharmacopoeial Commission of Indian Medicine and Homeopathy, PCIMH. Now this is the parameters which one should be followed, either in the case of industrialists, either in the case of researcher, or in the case of academicians. So these are the various parameters. This is the microscopy, how it looks like if you studies. This is the, it is a diagnostic characteristics also. Some plants have various peculiar diagnostic characteristics which can identify from the adulterated drug also. And one example is HPTLC. There are some RF values which by which we can provide, the researcher can provide to the industrialists, so that they do not waste their time They just check and they just check the standard parameter and they go for the production. Now I'll share one thing, that AYUSH clinical case repository. There is a AYUSH Sanjeevani app where all the doctors, all the Ayurvedic doctor, homeopathy doctor, Siddha doctor, they generate their data, acceptance and usage of AYUSH measures, which was created during COVID-19. They also provide and promote the AYUSH knowledge. They provide the AYUSH advisories related to immunity boosting measures, and to develop AYUSH interventions and solutions to reach out the target people 50 lakhs. So I have given the that website also. If anybody wants, they can go through and they can get some idea from there, and which will facilitate not just dissemination of the information, but also for the analysis and research purpose also, how the AYUSH system for the treatment disease conditions. So now this another portal that Ministry of AYUSH had launched. One is AMAR and second is RMIS. AMAR is AYUSH Manuscript Advanced Repository Portal, where you can find all the manuscripts, all the rare books and the journals, which is not available anywhere, all the rare books from the ancient times. And another one is Research Management Information System, which is a rich collection of research tools, reference and guide materials. Many of researchers, even those who are writing the research papers, they can do the research also here. Such type of resource therapeutic product would be useful tool in TCM, and it may be incorporated into as well. Now, so this is the herbal medicines database for drug discovery, where you can go and think herb, impact, DC, TDD, OMIM. This is the data, basically database. If one is starting their research, they can go to such database if some kind of work has already been done. Now the rationale of standardization and quality control of AYUSH medicines. This is the main important point is just to produce evidence rated research scale, and historically the isolation technique, bioassay technique, mechanism as a promising agents which may be incorporated to the AYUSH medicines. Applying the new strategies to the traditional medicines, they can amalgamate and they can produce a better scientific techniques. See, these are the rigorous basic science research isolation, because plant, the simpler it looks like, it is more complicated to research on that, because isolation and structural elucidation is very complicated. Dosage bioavailability is also again complicated. So it is necessary and it is mandatory to amalgamate the traditional medicine with the modern system of techniques to produce the good quality products. Now there are some central sector scheme for augmented quality control system, what I was telling, that there is a scheme which is like Ministry of AYUSH is providing. This is, I'll just show you. It is again from the National Medicinal Plant Board. They are providing lot of funds. Those who are keen to do the research. See, eligibility criteria, funding pattern 6 crore. For government pharmacies, they are providing 6 crore. For the private, they are giving the 50% of amount of the if you propose the proposal for 90 lakhs, this NMPB, the committee will evaluate your proposal. They'll give you 90 lakhs, 18 lakhs, according to your, if anybody wants this document, I can share that. So where is my presentation? I think I'm just finishing my presentation. My slide is visible.
The central sector scheme for augmenting that slide.
That is visible. Okay, I need to close this. Oh,
you want to stop the?
Yeah, I want this. Actually, I hyperlinked this, so I want to close this. Anybody from your host can, if you minimize this.
Now kindly try to share them.
Again, I need to share the slide. Okay. Yes. So I was my last slides. Oh, so this was the central sector scheme. It is a huge. There are a huge project. If somebody want to take research on the, or if you people are establish the nursery, or if you people are establish the herbal garden, they are providing the funds to that also, for the institution, for the college, for the, in fact, for the house, for the hospital. So there are a lot of funds which is being given by the this National Medicinal Plant Board, Ministry of AYUSH, and for the entrepreneurship, for the production also. So I know nowadays there is a major issue which is fund, and this is the solution. This organization is providing you the funds. So now I come sum up my presentation with the thanks. I think I had completed in the time. Thank you so much, and open for the questions, please.
Everyone are asking to share the document for the central sector scheme, ma'am. If you could share, kindly drop that document in the chat box, please.
Okay. Apart from this, okay, I'll do one thing. I'll email to this DY Patil email ID. You can share to everybody.
Okay, sure, ma'am.
Okay.
And everyone are asking to share your contact details, ma'am.
Contact details. Okay. My share. I'm writing my email ID also.
If any participant have questions, they will be contacting you directly, ma'am. Like,
Yeah, sure. Thank you so much, and thank you the team to giving me this opportunity, and congratulations for convening such a wonderful conference, and looking forward to coordinate with you in future also. Thank you. Bye bye.
Thank you, ma'am. Thank you so much. We are happy to have you here today. Next, I would like to welcome our next keynote speaker of our day, Dr. Vedpriya Arya, Scientist E, PhD Division, Head, Herbal Research Division, Patanjali Research Institute, Haridwar, India, to deliver her keynote talk. Ma'am, please. Ma'am, we are sorry, like you're on mute.
Am I audible?
Yes, ma'am, you're audible.
Yeah. So good afternoon, everyone. And I would like to congratulate the organizers for conducting such a nice conference. And today my topic is World Herbal Encyclopedia from Contemplation to Creation. As Dr. Piyush Juneja ji also said that there is a need to make herbal encyclopedia, and with very much proud, I would like to share some of the details of World Herbal Encyclopedia project, which is completed by Patanjali Research Institute. So today I will share some of the brief highlights with you, and I think this will be very important for the juvenile scientists as well as the senior scientists who are searching for some of the herbal medicines, that how they can be used. And we are not covering just Indian medicinal system, we are covering the world medicinal system with more than 900 medicinal system of the world. So let's begin with that. So I am first of all offering my salutation to our gurus, Swami Ramdev ji and Acharya Balakrishna ji, under whose guidance entire Patanjali team and the entire Patanjali herbal research team is doing such kind of works. Before starting the World Herbal Encyclopedia, I would like to make some of the emphasis that is what is the global scenario of medicinal plants. When we are talking about the global scenario of medicinal plants, what are the total number of medicinal plants in the world, what is their taxonomic status, and if there is unbiased information is available for all of the medicinal system of the world. What our concept is, why we are saying Indian medicinal system, why we are saying basically Chinese medicinal system, because a plant is plant and its medicinal property will be focusing for the disease. So it means to say we should have the information of all of the medicinal system. But at present, in our world, in world scenario, there is no a single checklist of medicinal plant in which all plants are documented. We have studied extensively in this regard. You can see some of the stats which are very interesting. We have found from the FAO that there are 52,000 medicinal plants, and from other resources that there are 72,000 medicinal plants. But we have found that these lists are based on estimation, and there is no detailed list of plants. There is no resources that how these plants are derived, and moreover, common plants among different countries, like if plant is growing in India, it is most probable that it may also grow in the China. So common plants are also not excluded from the list. When we are talking about Royal Botanical Garden Kew, they said there are 28,000 medicinal plants and which belongs to different species. There are the 28,000 medicinal plants around the globe. When we are talking about how they are derived or what are the resources they have justified, there are the 143 databases, 415 basically unique names of the plant-based medicines, and from where they have derived this data, it is available. When we are talking about the Kew medicinal plant or Kew botanical garden, then MPNS system is there, and which shows that there are the 26,000 medicinal plants. In a nutshell, when we are talking about the global status of medicinal plant, WHO, there are the 21,000 medicinal plant, but there is no checklist. When we are talking about the FAO, there are the 55,000 medicinal plant. Again, there is no checklist. When we are talking about Kew, there are the checklists for the 26,000 and 28,000 medicinal plant. But when we are talking about that, how much medicinal plant in the world, this is the absent, this is the questionable. Medicinal system of the world, this is again questionable. Total number of synonyms, again questionable. Plant names in world languages, that how much plant names are available in or are being spoken in different world languages, it's absent. Then Ayurvedic plant checklist or any of the system, medicinal system based plant checklist, taxonomic status clarification, as well as the medicinal list of the lower plants like the algae, fungi, bryophytes, pteridophytes, etc. There is no list available. So what we have done in Patanjali Research Institute, we have made a multi-voluminous compendium, which is of 109 volume compendium. In this 109 volume, one volume comprises of 1,000 pages. This is the extensive compilation which is, you can say, breaking all of the records. I will share some of the key facts that what we have done in the Patanjali Research Institute. There is the Vedic nomenclature. When we are talking about the nomenclature system, there is very ambiguity. I think if there are some plant taxonomists or the botanist in the audience side, they will be agree that if a plant it is being named in the binomial botanical nomenclature, for example, Acacia nilotica, in India, it might be possible that it is named some other name like Acacia, basically some other species name in some other countries. This is going on at present in the taxonomic era or in between the taxonomic scientists. So we have produced the Vedic nomenclature, which is completely based on the basically the morphology of the plants. Like if someone knows that particular name, he go to the field and he can perfectly identify that, okay, this is the feature, this is the morphological feature, and yes, this is the name. But when we are talking about the binomial nomenclature, there is no such kind of things. There are the evidences that some of the plants are named on the friend's name, some of the plants are named on the enemy name, some of the on the wife name, some on the like the son name, on the sister name, etc., etc. So when we are talking about that, this is the plant, this is the Acacia nilotica, or you can say this is the basically Acacia occidentalis, we are not able to derive its morphological characters. But yes, through Vedic nomenclature, which is the first time in the history of the world, we are able to identify the morphological characters in the field itself. So for this kind of work, we have the 900 scientists or Ayurvedic scholars. We have identified 7,500 plant genus, 50,000 plant species. We have not provided just the checklist, but there is the detailed information of these 50,000 medicinal plants. Well, this encyclopedia, this extensive compilation, it is in 108 volumes. We have covered 2,000 tribes of the world. 2,000 tribal information, like this tribe is using this condition. We have included around 2,000 tribal people information in this particular text. We have also referred the 14th century Ayurvedic texts like the Rajnigandhu. There are the 2,200 folk formulation, and very interesting fact, there are the 30,000 canvas paintings. Each and every plant, it is hand painting on the canvas, and there are the total 30,000 plant canvas paintings and 35,000 line drawings. There are the 6 lakhs bibliographic sources. This whole information, which is compiled in 109 volumes, these all are authentically compiled in with 6 lakhs bibliographic sources. There are the 2.5 lakh plant synonyms, 12 lakh vernacular names, and very interesting fact is that these 12 lakh vernacular names are being pronounced in the 2,000 languages of the world. We are identifying here 160 botany-based medicinal system over here, as well as 4.5 lakh unique reference codes. Well, this whole information, in which more than 50,000 medicinal plants are compiled over here, there are the 6 phylogenetic groups in which these are arranged: algae, fungi, bryophytes, pteridophytes, and the gymnosperms and angiosperms. You can see here, these are the different genus, like the algae, fungi, bryophytes, pteridophytes, gymnosperms, and angiosperms. As you can see, there are the 47,000, moreover, 47,000 plants in the angiosperms. When we are talking about the Vedic nomenclature, we have find out the morphological characters plus what are the gun, dharma, and dosh, which is a crucial factor in the Ayurvedic system, Ayurvedic medicinal system. For example, if the Vedic name is the Pichu Mardaka Nimbakam, the name is based on the ancient Sanskrit manuscript, and it qualifies its morphology, anatomy, properties, and action. This is the medicinal history of the world. You can find out there are more than 8,500 bibliographic resources through which this information is compiled. 5 volumes are totally dedicated to the medicinal history of the world, like how this plant is derived. It is covering Indian medicinal system, it is covering Chinese medicinal system, Aboriginal peoples of Australia, as well as the Native American people of America. We have covered 962 healing practices. A very important fact that we have discovered in this particular compilation, there are more than 900 tribal-based medicinal system. But when we are talking about the medicinal system, you can say principle-based medicinal system, like the Ayurveda, like the Unani, like the Chinese medicinal system, there are around 10 to 15 medicinal systems around the world. We have covered the pharmacological aspects. These are also covered under the World Herbal Encyclopedia, and it is very important. Like if Dr. Juneja also said that if this plant it is having in vitro, in vivo studies, a lot of work is going on, and our pharmacologists are doing the marvelous work. But there is not a single document in which these all information are compiled with reference-based text. So we are compiling the pharmacological aspects of the encyclopedia. In encyclopedia, you can see that the postdoctorates, PhD holders, more than 300 scientists are over here who are currently working for the World Herbal Encyclopedia. In this World Herbal Encyclopedia, we have covered the ancient manuscript preservation as well as the republication. Like some of the manuscripts which are in the Bhojpatra, some of the manuscripts are in the Tamrapatra. We have identified those Sanskrit scholars who can read those manuscripts, who can translate those manuscripts, and we have provided that in the English version of the, we have also conserved and republished these manuscripts. At present, we have 63 lakh pages of ancient manuscript that we have digitalized at present. It includes 50,000 palm leaf based manuscripts which are in different language. Some of are in the Bengali, Malayalam, Marathi, Nepali, Udiya, Tamil, and so on. And there are the several subjects for when we are talking about the Ayurvedic content. So Ayurveda, different subjects are there, like the Brahman Granth, Darshan, Dharma Granth, Jyotish, Kavya, Kosh, Mantra, Niti Granth, etc., etc. Another very important point, that is the World Botanical Dictionary that we have done. If any particular plant, out of 50,000 plants, it is named, why it's named. In the modern medicinal system, we have compiled this information. There is a potentially herbal research, herbal museum. Basically, in this herbarium, this is accredited by the Index Herbarium, New York Botanical Garden, in which we have conserved more than 10,000 plant specimens over here. This is the herbarium, and yes, because students are also here, so if you want to identify a particular plant, we are having the taxonomist as well as the voucher specimens are also available here. You can facilitate this facility from the Patanjali Research Institute. Then we have the Patanjali Herbal Painting Museum. You can see each and every painting, this plant painting, these are the handmade paintings, canvas paintings. These are also available. These are available in these sliders. And this is the work for the centuries, not for the 10 or 15 years, because any of the plant which is endangered at present, which is extinct, or it's not identified by, renowned taxonomist, it is available over here, and the students as well as the faculty members can come and you can say they can match and they can find out that what is this plant. Then this is the Patanjali Herbal Garden, more than 300 species of herbs, shrubs, climbers, these are over here. 5 artificial caves which are providing the Ayurvedic basic Ayurveda basic knowledge, beautiful sculptures, as well as our ancestors who are preparing churans, what is asam, bhasm, etc. over here. Then we have this is the Patanjali Research Institute main basically campus, and there are several divisions. I'm just going through some of them, like the Drug Discovery and Development Division, Herbal Research Division. Well, these are some of the pictures of the our laboratory, Drug Discovery and Development Division. What's our vision? Our vision is to cover the old or ancient Ayurvedic manuscripts, or you can say Ayurvedic formulations, and on the base of the evidence-based medicinal system, we want to prove them at the basically global level. Then you can see here we have all the research strength and accreditations at the Patanjali. We have the NABL accreditations, DSIR accreditations, CPCA guidelines, and the NABH accredited hospital. So this is the brief history in a short span of basically around 4 or 5 years. We have made several interesting things. These are the different analytical chemistry, biological research capabilities, and the workforce. Near about 300 scientists are working here, and these belong to different area of expertise. And this is our whole team of scientists. And I would like to share a message that If anybody is interested in this kind of concept of the World Herbal Encyclopedia, most welcome at the Patanjali. If you want to identify a plant, any of the plants, so Patanjali Research Foundation Herbarium is there, and there are lots of opportunity to work in collaboration as such. Thank you so much.
Thank you, ma'am. Thank you so much for your valuable information. Participants, do you have any other queries? Ma'am, kindly drop your mail ID and contact number, please. Thank you, ma'am. Thank you so much. I request all the participants to turn on their videos for gallery photo. I repeat, I request all the participants to turn on the video for gallery photo. Thank you, everyone. Next, I would like to welcome our Wig Health Products are pleased to introduce themselves as a pioneer in manufacturing of pure vegetarian sweets, tastes, jellies, which are soft and easily chewable for all age groups, with sugar as well as sugar-free, depending upon choice of their valuable clients, with the motto, eat medicines with taste and enjoy. Dawa kalo, khel khel mein.
Health Products are pleased to introduce themselves as a pioneer in manufacturing of pure vegetarian sweet taste jellies, which are soft and easily chewable for all age groups with sugar as well as sugar-free depending upon choice of their valuable clients with the motto, eat medicine with taste and enjoy, dawa khalo khel khel me. Wig Products are FSSAI certified small-scale industry and currently working with many pharmaceutical companies. With most of the products are plants-based and natural, Wig Products have come up with a formula in 100% natural herbal for increasing immunity in the body to fight against viruses. All the products are included with the ingredients like pectin, sucrose, stevia, glucose, stevia gel, citric acid, natural flavor and food color. They are fortified with multivitamins, herbs, chyawanprash, curcumin, avla highly enriched with vitamin C. The benefits of Wig Healthy Gummies: improve digestion, acts as an immune booster, very nutritious and helps in weight management, rich in antioxidants, minerals and DHA. They are also source of multivitamins which provide energy and improve skin, hair and nail health. Wig Products hopes that you will take interest in our above products and give a chance to serve you better. Apple cider vinegar jellies, gummies. Ashwagandha and ginseng extract gummies. Ashwagandha with vitamin D and zinc jellies. Biotin jellies, gummies. Chyawanprash jellies, gummies. Curcumin lozenges. DHA jellies, gummies. Multivitamin and minerals jellies, gummies. Curcumin with mint lozenges. Ashwagandha and ginseng booster jellies. Chyawanprash booster jellies. Cough and cold sore throat jellies. Spirulina powder jellies.
Now I call upon Mr. Vig Mahendra Raja from Vig Health Products to speak few words about the company and their health products. Sir, please. So kindly unmute yourself, please.
Now you can hear me?
Yes, sir, you're audible.
Yeah. Good afternoon, everybody. I am chairman of Vig Health Product, founder. We are manufacturing gummies, jellies in all vitamins, herbals, everything. So it is good for health and easy to eat. Our proverb is dawa khalo khel khel mein. Every child can eat, old age can eat, and it is such a soft jelly. You put in your mouth the vitamin and minerals or whatever herbs are there, so that goes in your body. So there is no chewing gum or anything like child के दांत में फंस जाएगा, teeth में ऐसे कुछ भी throat में. It is very easy for all ages, old age, young age, and anywhere you can take Chyawanprash, or else otherwise Chyawanprash you have to take with spoon. Our Chyawanprash with this is you can open the flap and eat 2.5 gram. This 100 mg is there in every herbs. So all details are there. So we are having full plant with all equipped machines and hygienic plant at Lonavala, Mumbai. Offices at Mumbai. Hello. 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 Can you hear me?
Yes, sir, we can hear you.
Yeah. So any question, anybody? Hello. We are third party manufacturer also. We are having our own brand, but we are mostly doing third party manufacturing for pharma companies, herbal companies, any doctors or anybody, we can make their brand and give them.
Is there any questions from the participants? Okay, sir, I guess everyone are clear with the thing.
Okay. My numbers are there, email address is there, website
Yes,
they
sir.
can see. You please display.
We will be sharing in the chat box. So if any other queries are there, they will be contacting you.
Please. Thank you, madam. Okay. Thank you. Thank you very much.
Sir. Now, I thank all the keynote speakers and all the participants to be enthusiastic. Now, I would like to continue with our technical session. I welcome all the participants for the technical session. All the respective participants join your respective halls through breakout rooms. If anyone are facing difficulty, kindly drop a message in the chat box so our coordinators will be guiding you to how to join the halls. Now I call upon Ms. Bhargavi to take over the main hall as host. Thank you.
Yeah, thank you, Roshni. So I hope we can start our technical session now. I hope all the students and the participants have been joined in the main hall. So today our session chair is Dr. Rukia Sultana. I would like to welcome the session chair, Dr. Rukia Sultana, who is Professor HOD of Department of Pharmacognosy, Enipoya Pharmacy College and Research Center, Mangalore, India. I welcome you, ma'am. So, ma'am, today we are having completely of 17 presentations where each participant will be provided with 10 minutes. So, before entering into our session, I would like to give some instructions to the participants. The time provided for each and every participant is 10 minutes, which includes the discussion session also. So 7 minutes you can allocate for your presentation, and the remaining 3 minutes you can allocate for the question and answer sessions. So I request each and every participant to stick over to the time and go as per the timings for their presentations. Dr. Rukia Sultana ma'am.
Thank you. Thank you.
Okay, thank you ma'am. Welcome you. And can we start the session ma'am with your permission?
Yes, please start.
Yeah, thank you ma'am. Today our first presentation is oral presentation by Sujata Marmatha Mandala, NG Halgar Institute of Dental Sciences, Rajiv Gandhi
Excuse me.
University of Health Sciences, India.
Hello.
She's
Hello. Yeah.
hello.
Yeah. Just want to know how to join the respective halls, hall A, B, like that.
Yes, sir. I can guide you. Top to bottom to your screen. I hope you can able to find the option called breakout rooms. May I know the device that you're using? You're using mobile or laptop?
Laptop.
Yeah. Bottom you can see the option of breakout rooms.
Yes. Just
If you can click the breakout room, you can find out the hall names, hall A, hall B, hall C. So respectively, you can join in your session, sir. Sorry for the interruption. Now I would like to welcome our first participant, Sujata from NGH Institute of Dental Sciences, Rajiv Gandhi University of Health Sciences, India. And today she is going to give her presentation topic on tea tree oil, a boon in the field of medicine, RGUHS grant study. Dr. Sujata, you can start sharing your screen and you can go with your presentation. I wish you all the very best. Answer it. Dr. Sujata, you can start with your presentation. Sorry to interrupt, Dr. Sujata, we are unable to hear you. Dr. Sujata, if you have any technical issues, please let us know. We are unable to hear you. Dr. Rukia ma'am, could you please confirm us whether the presentation was audible or not?
No, not audible yet.
Yes, yes. Thank you, ma'am. Thank you for the confirmation. Dr. Sujata? If you have any technical issues, please let us know so that we can have your presentation after a few minutes. Ma'am, meanwhile, can we go with the second presentation?
Yeah, please.
Yes, ma'am. Today, the second presenter is Vilas Shinde from Yashasri Hospital, India. He is going to give his presentation topic on novel hypoglemic agents from some Indian medicinal plants. Dr. Vilas V. Shinde, you can start sharing your screen and you can go with your presentation. And I request Dr. Sujata to stop sharing her screen so that another participant can share the screen. Dr. Vilas, sir. Dr. Vilas, sir. Ma'am. I request Dr. Sujata to stop sharing her screen. Dr. Sujata, ma'am. Dr. Sujata, ma'am. Dr. Sujata, ma'am. Sorry for the inconvenience, ma'am. Just give us 1 minute time and we'll sort this out.
Sure, sir, no problem.
Dr. Vilas Shinde, could you please let us know whether you can able to share your screen or not? Dr. Vilas, sir? Dr. Vilas Shinde? Please let us know whether you can able to share your screen or not so that we can sort this out.
Am I audible?
Dr. Sujata? I hope Dr. Sujata is there online. Joe?
Hello. Hello.
Hello.
Are you presenting, Dr. Vilas? Are you presenting? No voice.
Yes, ma'am. Dr. Vilas, sir. Dr. Vilas, sir, we are unable to hear your voice. Dr. Vilas, sir. Hello. I request Dr. Vilas, sir, please confirm us. Your voice is not audible to us.
Vilas sir, kindly unmute yourself so that you will be audible to the participants. Dr. Vilas, actually you are not audible. So kindly unmute yourself so that we could hear you. Sir, you at the down bottom of your screen, you could see a mic icon so that if you click on that, you could unmute yourself. She joined with this name, HPP, Priya Desai. Sir, I guess there is a technical glitch from your side, so we couldn't hear you. Okay, so we will move on to our next presentation. Oral presentation by Desai Priya Ajit Kumar, Tatya Pharmacy College, India, on a topic of effect of Panchagavya and Panchagavya Gitra in treatment of lung cancer in Vistar albino rats.
Hello. Hello.
Yes, ma'am, you're audible.
Okay. Just wait for 2 minutes. Can you all audible to me?
Yeah.
Hello. Hello.
Yes, ma'am, you're audible.
Yes, ma'am, you are
Kindly
audible.
go.
Okay. Shall we start?
Yes, ma'am, you can.
Okay. So first of all, good afternoon to all of you present over here. I, Priya Desai, from Assistant Professor of Tatya Pharmacy College. Today I'm going to share a presentation which is on my research work of effect of Panchgavya and Panchgavya Grita in the treatment of lung cancer, which is Vistar albino rat, and presented over the Start Hub from Innovation to Entrepreneurship Hub in 14-15 October. Today I am going to cover introduction and backgrounds, aim, objective, material, method, result, conclusion, and the references of my research work. So first of all, introduction and background. The malignant tumor. We can say that malignant yani cancerous process. Cancer is what? The uncontrollable growth of the whole cells. That is called as a cancer. And the malignant cancer, we can say this physiological parameter. We can say different physiological parameter. Okay. For example, you can particularly talk about the lung cancer. So malignant lung cancer are the. Can I audible, ma'am?
Yes, you are audible. Please carry on.
Hello.
You are audible, dear. Please carry on.
Okay, thank you, ma'am. Malignant cancer is which various histological parameter arise from the different such type of cell is epithelium, bronchial, or the alveolar other type of the glands. So this cell is divided in mainly two categories. This is a small cell lung carcinoma and non-small cell lung carcinoma. That small cell lung carcinoma is 85% of total of all the lung cancer type. And in this particular research, I will develop my model by the DEN, diethyl nitrosamine, that is a chemical. And this chemical is used in the animal, which is visceral albino pup. And I developed the cancer for my treatment model. And traditional medication, which is the remedy of the lung cancer treatment. Nowadays, whole world is towards going to Ayurvedic, and particularly I choose this topic. Cowpathy is belonging to a long year back for the different treatment of the medication as a different disease. I select the Panchagavya and Panchagavya Grita. That means a mixture of 5 cow product, that is milk, dung, curd, urine, and ghee. This whole Panchagavya and Panchagavya individual component are reported for the immune booster for the cancer treatment of particular breast and prostate. There is no such research is done on the Panchagavya and Panchagavya Grita as an individual parameter or both comparative parameter in treatment of lung cancer. So today I choose, in research I choose this particular subject, that is Panchagavya and Panchagavya Grita in the treatment of lung cancer. Now, my aim and objectives are, the aim is to study the effect of Panchagavya and Panchagavya Grita in treatment of lung cancer induced by diethyl nitrosamine, that is a such chemical which induce the cancer in any model of animal. Now, my primary objective is to determine the induction of lung cancer. Before I taste my drug, I should know whether this particular cancer is inducted or not. So I use particular DEN for the lung cancer induction model. To evaluate, second objectives are to evaluate the effect of Panchagavya and Panchagavya Grita in anti-cancer agent in treatment of lung carcinoma induced by the DEN. And third objective is to compare anti-cancer activity of Panchagavya and Panchagavya Grita. So ultimately we are going to be test induction model. Secondly, it individually Panchagavya and Panchagavya Grita's treatment effective, and both are comparatively effective. We are going to be tested. Now, material and methods. In vitro study I will use, in which animal model is induced in 15 days old pup of visceral albino red. I will give a particular, each group contain 8 animals. My study require only 6 animals because there is a cancerous study. So many problem or issue associated with the lifespan. So I will take 2 animals extra in each and every group. So totally each group contain 8 animals. And I select either sex animal for this study. So their weight is 10 to 15 grams. And route I selected for the administration of drug, that is DEN in intraperitoneal route, whereas the treatment is oral route. So Panchagavya and Panchagavya Grita will be used by the oral route, and DEN will be given by the or introduced by the intraperitoneal route. Now, procedure for the model induction as well as the treatment. Now, 15 days old visceral albino pups are weighted, and 48 pups are divided into 6 groups. As I told earlier, a 6 group per group we have 8 animals, which in study I require only and only 6 animals. Hello. Now I would like to ask. Hello, is there any problem? Hello.
Please carry on, no problem. Please
Okay.
carry.
Okay, thank you, ma'am. 48 pups are divided into 6 groups, which is only 6 group contain 6 animals and the 2 animal extra per group. So first group is only normal control group, that is given by saline or treated by saline only. Second group is model control group and treat with the DN 100 milligram per kg in 5 divided dose. Yani, it means 20 milligram per kg per day. Now the third group is treatment group 1. First group is DEN 100 milligram per kg for 5 days, and Panchagavya low dose, that is 500 milligram per red. This dose selection will be done by the toxicity study already reported for the breast cancer. Now, second treatment group is DEN 100 milligram per kg plus Panchagavya high dose 100 milligram per red. Now, the fifth group is the treatment group 3, DEN 100 milligram per kg and Panchgavya low dose 150 milligram per kg, as well as high dose 300 milligram per kg, which was my 5th group, and the treatment group is 4. Now, second is every morning. Now we are going to start with our experiments. Every morning we are choose the collect the Panchgavya and Panchgavya oral route a 90 days treatment, and after that in 61 day, the period of, you can see in this timeline, from day 1 to 60, I will continue with my this Panchgavya and Panchgavya. From 61 to 65, I will treat with both Panchgavya and Panchgavya plus add-on cancer induction model. I use DEN intrapenetrator route for the 90 days, and DEN only given by 61 to 65 days for study. So perform biochemical test evaluation for cancer induction at the end of the experiment. Treatment for 90 days for both non-Panchgavya and Panchgavya grade. Evaluation parameters start before 60 days and after at the end of the study, that is 19 days. So we are carrying the study for 90 days. On the 61 to 65 days, I was used to induction of model. So before induction and after induction, I will be take some parameters to evaluation. Now, evaluation parameter: before in control, nothing treated; in model, nothing treated. Panchgavya and Panchgavya will be treated. And after the cancer model induction, 61 day to 65 day after, in model control group there is a DEN 100 milligram per kg, Panchgavya 50 and 100 milligram per rat, milligram per rat per day for Panchgavya plus DEN, as well as Panchgavya and Panchgavya grade for the same for Panchgavya grade doses. After the end of this study, at the end of 90 days, the blood collection from the evaluation for biochemical parameters and biomarkers, for example, C-reactive protein, serum creatinine, and other parameter, histological parameter will be assessed. Now, we can start with the procedure of DEN and model induction. Now we are going to be introduce the procedure of Panchgavya and Panchgavya grade. So Panchgavya first of all, in which all cow product, that is dung, ghee, urine, and the milk will be collected and morning it every morning. So we have to collect fresh product of milk, dung, and urine. After that, that product will be equally used for this preparation. So first of all, what we are going to be do, urine and dung will be mixed well and filtered for 8 times from cotton cloth, and after that filtration will be added into milk, ghee, and curd in this filter in equal proportion. After that, again filter 1 time, so we can get a Panchgavya. And after a Panchgavya grade, that means early in the morning, collection of fresh cow product, milk, dung, urine, and freshly indirect product, ghee and curd in equal proportion. After that, urine and dung will be mixed well and then filtered for 8 times from cotton cloth. After that, all the filtration is collected. That means filtered are going to be achieved in our hand. In this, we are going to be add milk and curd and again filter well. After that, boil the whole product up till the water is removed, and after that equal amount of ghee will be added in the filtrate, that is called as the Panchgavya grade. In Panchgavya, we are just adding raw material and filter it. In Panchgavya grade, we have to remove excess amount of water and that add into equal quantity of ghee. Ma'am, can I audible?
Yes.
Okay. And now we go for the results. Now we can see the body weight. If general thing is in mind, that is a present amount of ghee that will be direct increase the adipose tissues of our body. So you can say this first is for control group. This blue line you can see, that is a control group in which we are only treated the saline. In model control, we are going to be only use the DN material. But if you are saying that the effect of Panchagavya and Panchagavya Grith on the weight of the animal, the high dose as well as the Panchagavya high dose will be increase the maximum effect of their weight. So they are increasing maximum weight. You can see these two eras as compared to that control or model control group. Same as the second parameter is serum creatinine. If you see this, control level of serum creatinine is this, and model control is maximum elevated level of the serum creatinine. And you can see this parameter or this result will be so the highest or model control group contain maximum amount of creatinine, serum creatinine, whereas decline level of treatment group option. So this ratio or these particular parameters shows our treatment drug, that is Panchagavya and Panchagavya Grith, will be useful in the treatment or effective in this treatment. That is another parameter for the estimation. Now, C-reactive protein. If you can see this before treatment, there is a negative. You can see this, no uniform distribution of the sample shows the negative result, as well as the positive result means you can formation of clot or aggregation. As you all know, that is a normal control, model control, treatment 1, 2, 3, 4 are group. So only after the treatment, model control group have a positive result. You can see in next slides. See, these all are the uniform distribution of result, whereas in these standard group or model control group contain shows that aggregation or clotting of particular sample. So this shows that this particular component or this particular sample shows your positive result, as I saw in earlier slide. That is C, negative, all are negative, and in only model induction group there is a positive. This particular C-reactive study be done on based of this positive and negative control. How much is the how much cancer is spreaded or how much induction, that will not represent over here. Now the last parameter is histological parameter. You can see these all histological slide are undertaken of 10x, whereas on right side you can see 40x image of all histological parameter. So A represent a normal control group, B represent the model control group, C represent low dose of Panchagavya, whereas D represent Panchagavya high dose. And now E and F will be represent Panchagavya low and high dose respectively. You can see this purple color dot, that is a nucleus which is maximum growing in the 40x slide of model control group. That means our model group, or we can say induction group, will be shows the maximum cancerous cell or maximum growth, in which there is low as compared to all other groups. And this F means Panchagavya Grith high dose will be same, shows the result as we known in the as we seen in the normal control group. That means that kill all the cancer already controlled, already in use in this study. Now my conclusion from the result of present study, it concludes that both Panchagavya and Panchagavya Grith are effective in treatment of lung cancer induced by diethyl nitrosamine. I will add something in which particular ghee contain conjugated linoleic acid, which is used as a cancer treating agent. In dung, that is a disinfectant agent component. Milk contain lactoferritin and urine contain uric acid, and the curd contain a protein. That all 5 components are mixed together in Panchagavya as well as Panchagavya Grith. That will be shows particular anti-cancer activity which is reported in my studies. That all factor individually reported for other studies, but there is no single evidence reported for the lung cancer treatment. Now, a conclusion, another is it is defined from the study that high dose of Panchagavya Grith is better treatment cancer in induced by the diethyl nitrosamine. This study is done only primary base Still, further confirmation studies are significant evaluation parameters are suggested for better discrimination. Those are my reference, and thank you.
Yeah, my question is,
Yes.
You have selected 2 doses, right?
Yes, high dose and low dose for both.
How did you select that?
Based on their toxicity study done on the breast cancer.
Okay, you have already collected from the articles.
Okay, yes, ma'am.
Okay, and which is the route of administration of your these 2 formulations?
Oral route. This is mentioned over here. Treatment is oral route.
Okay, fine. This is one more thing. What I want to tell you is,
yes,
you have not done any blood parameters, right?
Serum, no, ma'am. I will done with the blood parameter serum creatinine. That is blood parameter. Serum
creatinine you have done, but when it comes to the cancer,
yes.
When it comes to the cancer, your WBC, RBC, hemoglobin, neutrophil, all these things also plays a very important role.
Yes, ma'am.
So even that need to be determined.
Ma'am, particular for that, I will choose C-reactive protein, that is a biomarker also, and serum creatinine. That both tests will be done for the on basis of blood sampling collection.
How did you collect the blood sample? Which route?
Retroorbital plexus, ma'am.
Okay, thank you.
Thank you, ma'am.
Thank you, Dr. Desai, for your presentation.
Welcome, ma'am.
Now, I would like to welcome the first presenter, Dr. Sujata, from the Institute of Dental Sciences, Rajiv Gandhi University of Health Sciences, India. I hope, Dr. Sujata, now you can start sharing your screen and you can go with the presentation.
I request Dr. Sujata to be stick on the time given to you.
Yes, a small guidelines for all the presenters. So the timings that is provided for each and every participant is 10 minutes, 7 minutes for your presentation, and 3 minutes would be the discussion time. I request everyone to stick on the time and you can go with your presentations. Dr. Sujata, you can start your presentation. Dr. Sujata, your voice is not audible. Dr. Sujata, could you please acknowledge us whether we are audible to you or not?
Hello.
Hello.
Dr. Sujata, we are not able to hear you.
Sorry, Sujata, we can provide you the presentation this next time. So please check with your network connections and all for the next time. So ma'am, can we move to the other presentation, ma'am?
Yeah, please.
Yes, ma'am. So now I would like to invite Dr. Sheikh Sana to give her presentation.
Good afternoon, one and all. Is my voice audible?
Yes, ma'am. Yes, ma'am. You are audible.
Okay. Just give me a minute. I will start my presentation.
Yes, ma'am.
Is it visible?
Sorry, ma'am. We are unable to see your screen.
Am I audible now? I'm Sujata. Just.
You're audible
now. Sorry, ma'am. Sorry. Yeah.
Sujata
Okay.
ma'am, are you there online?
Yeah, ma'am. Yeah, ma'am. Shall I start?
Yes, ma'am, you can start sharing your screen and you can give your presentation. Dr.
Okay.
Sheikh Sana, could you please wait for a few minutes?
Yeah,
after this
sure, no
presentation?
problem. Yes, yes.
Yeah. Thank you, ma'am. Thank you.
So you can see the screen, ma'am, now?
Yes, ma'am, we can see.
Yeah. So I'll start with my presentation. Sorry for so much of inconvenience creating. So this is Dr. Sujata Bahati, professor and the primary investigator for the Rajiv Gandhi University of Health Sciences grant-related work on tea tree oil as a boon in the field of medicine and in vitro study. These are the contents of my presentation. Coming to the background, natural essential oils play an important role in the cancer prevention and the treatment. Various mechanisms such as antioxidant, anti-cancer, and anti-proliferative, enhancement of immune function and surveillance, enzyme induction, and the enhancing detoxification, modulation of multi-drug resistance, and synergistic mechanism of volatile oil constituents are responsible for chemopreventive action of tea tree oil. More than 60% of anti-cancer agents which are currently in use are derived from the natural resources. Tulsi, Curcumin longa, Nigella sativa are to name a few. Penfold in the year 1920 has invented more than 100 different components of tea tree oil. Tea tree oil has wide range of mechanism of action. It's a very good antimicrobial agent, antibacterial agent, antifungal, antiviral, and anti-inflammatory agent. Currently, lot of importance has been given for its anti-cancer property. Many commercially available beauty products are utilizing this great antimicrobial action of tea tree oil in the form of face washes, cleansers, and lotions. The basic mechanism of the action of tea tree oil is as follows. The cytotoxicity of the tea tree oil might be due to the interaction of lipophilic components of the oil with a phospholipid bilayer of cell membrane with a consequent alteration of cell growth and activity by inducing cell necrosis. Keeping these properties of tea tree oil in mind, this study helps us in assessing anti-cancer activity of tea tree oil against various cancer cell lines by using MTT assay. We also found its efficacy as a root canal agent and antibacterial agents. The objectives of my study are as follows: to evaluate whether tea tree oil has any anti-cancer activity against standard strains of cancer, specifically concentrating on oral cancer, and also to check there is no cytotoxic activity against mouse fibroblast cells and the weather, that's multi-kidney cell lines, which were used as control in our current study, and also to decide the minimal inhibitoric concentration 50 and the minimal bactericidal concentration 50 value of various concentration of tea tree oil. This study we conducted at the Central Research Laboratory at Maratha Mandal Snataji Rao G. Halgekar Institute of Dental Sciences, Belagavi, Karnataka, after obtaining the ethical clearance. And the cell lines were procured from National Center for Cell Science, Pune, and we have received around 85,000 rupees of the grant from the Rajiv Gandhi University of Health Sciences. We received this commercially available plant extract from Crystal Aromatics, New Delhi, imported from Australia with a refractive index of 1.475. At 25-degree, weight per ml was 0.8850 grams per ml. This oil is obtained through steam distillation process and always stored in the amber color bottle to prevent the oxidation. MTT assay. MTT is a calorimetric assay developed by Mossman with a modification was used to screen for cytotoxic activity of tea tree oil, that's a plant extract. The cells were seeded in 96 well plates at the density of 10⁴ cells per well in 100 microliters of culture medium. More the cells are viable, more the purple color, more the cell undergo lysis, less purple and the light color. Absorbance, that is optical density, was measured at 570 nanometer wavelength using microplate reader LISA Plus and quantified by spectrophotometer. The quantification of inhibition of proliferation shall be done by manual counting of cells under microscope by using Newborn's counting chamber. The percentage of cytotoxicity and the cell viability were determined by two standard formulas and the data obtained were statistically analyzed. MTT assay was performed on the following cell lines: laryngeal cancer cell line, cervical cancer cell line, colon cancer cell line, leukemic cancer cell line, oral cancer cell line, and the breast cancer cell line. Apart from this, NIH3T3 and the mouse fibroblast cell lines, which were used as a control, they were also checked for the cytotoxicity with the tea tree oil. And the MTT assay was performed at the interval of 24 hours and the 48 hours of duration. Similarly, as a dental application, the cytotoxicity was also checked for the fibroblasts present in the periodont ligament and the pulp fibroblasts at the interval of 5 minutes, 24 hours, and 48 hours, so that this particular reagent can be used as a root canal irrigant. And also, we checked for the antibacterial activity of this particular TTOL to check the sensitivity and the resistance on the oral microbiota and the minimal inhibitory concentration and the minimum bactericidal concentration of the TTOL. After checking the Spearman's row correlation, we could see that the statistical significant results were obtained with all the different cell lines except for the HT29, and the control which were used in our current study also had a statistical significant results, that is NIH3T3 and Vero. Pulpal and the periodont ligament fibroblasts also showed the statistical significant results with the p-value being less than 0.005. So the IC50 value of TTOL for the different cell lines were obtained for different cell lines to help us to proceed further to conduct the animal study and then with the human clinical trials. Further, this also helps us in producing with the patency of a particular drug dose. So the IC50 value of TTOL, which were checked for the laryngeal cancer cell line, after treating it with the TTOL for the 24 hours, we obtained it as 0.781 micrograms per ml. And this is a dotted plot, which is also depicting the same results. IC50 value for the oral cancer cell line after treating with the TTOL for 24 hours is 25 micrograms per ml. IC50 value for the TTOL for the cervical cancer cell line after treating with the TTOL for 24 hours is 3.125 micrograms per ml. IC50 value of TTOL for the colon cancer cell line after treating with the TTOL for 24 hours is 12.5 micrograms per ml. IC50 value of leukemic cancer cell line after treating with the TTOL is 3.125 micrograms. IC50 value for the breast cancer cell line after treating with the TTOL for 24 hours is 25 micrograms per ml. Similarly, just to check the cytotoxicity of the normal cells that which were used as a control in a study, that is monkey kidney cell line, also called as a Vero, and also NIS3T3 mouse fibroblasts, which were used as a control in a study, we could see that cytotoxic effect of TTOL was not found. Rather, we could see the excessive proliferation of the cells. Similarly, the cytotoxic activity of the TTOL is also checked on the PDL fibroblast and the pulpal fibroblast at the interval of 5 minutes, 24 hours, and 48 hours. We could see that the cells were excessively proliferating rather than undergoing rather than undergoing cell lysis. So this is the beauty of this particular reagent, so that we can be using this particular agent as a root canal irrigant in the coming future. So the minimal inhibitory concentration were checked for the oral microbiota, that is streptococcal mutants, actinobacillus actinomycetum comitans, Fusobacterium nucleatum, and Porphyromonas gingivalis. The antibacterial action, which has shown that it was for the streptococcal mutants, it is sensitivity was seen up to 6.25 micrograms per ml of TTOL. For the AA organism, till the 7th generation of TTOL, and for the Fusobacterium nucleatum, the first concentration itself was showing a high sensitivity. Porphyromonas gingivalis had the sensitivity up to 7th concentration of the TTOL. The minimum bactericidal concentration also had shown there was no growth appreciated for the streptococcal mutants till the 6th concentration of the TTOL, that is 3.12 micrograms per ml. And Actinobacillus actinomycetum comitans also had shown no growth till the 7th concentration. Fusobacterium nucleatum also has shown no growth in the first concentration itself. And Porphyromonas gingivalis had shown there was no growth appreciated till the 6th concentration of the TTOL, that is 3.12 micrograms per ml. Coming to the conclusion, the efficacy of a TTOL as an anti-cancer agent with its minimal inhibitory concentration 50 value in our current study has shown a better results than the other naturally available plant extracts from the previous literature. This variation in the result also could be due to the differences in the method and the concentrations used from the previous studies. Hence, this IC50 value of TTOL with its greater efficacy related to its anti-cancer activity can be brought to the level of clinical trials in the coming future. Similarly, it also has been seen that TTOL has a very good antibacterial agent against the various strains of oral microbiota. With this, I would like to conclude my presentation. So this is my special thanks to Dr. Kishore Bhatt and Head of the Department of Oral Medicine, Dr. Renuka, and Dr. Chetana and Dr. Ravi Shirati for the statistical help. Thank you.
Yeah, Sujata, it was a good presentation. I want to know, have you checked the bioactive components which is present in the tea tree oil?
That is the next step, ma'am. We are applied for the grant, ma'am. So that we are going to proceed with, ma'am. So there are more than 100 components, as I mentioned you. So we are trying to find out the main components which are responsible for the anti-cancer activity of this particular agent.
Yeah, true,
Yes,
true.
ma'am.
Okay, okay, thank you.
Okay.
Thank you for your presentation. So, ma'am, shall we go with the other presentation?
Yeah, please.
Yeah, thank you, doctor, for your presentation. Now, I would like to invite Ms. Poonam T. Taru for oral presentation from Vishwakarma University to give her presentation. Ma'am, please.
Ma'am, just a minute. I'm sharing my screen.
Yes.
Yes, ma'am. Is it visible to you all?
Yes, ma'am.
Okay.
Your screen is visible and you're audible. We can go with your presentation.
Okay, ma'am. Thank you so much. So shall I start? Okay, very good afternoon to all. My name is Poonam Taru from Vishwakarma University School of Pharmacy. And my topic is a review on the natural products and traditional medicines for COVID-19. So as we all know that, just wait, ma'am. Yes, so all we know that the COVID was generally originated from the Wuhan, China, and the cause for this virus is a novel coronavirus, which is called as the SARS-CoV-2, and that is the severe acute respiratory syndrome coronavirus. And the disease is mainly transmitted through close contact with the infected individuals via respiratory droplets, either from the sneezing or coughing or by the touching to that infected persons. And the natural resources and medicinal health plays a valuable and important role in managing this COVID-19. Already we have seen this scenario and these how they are going to help us. So it can provide both like the preventive and the therapeutic aid in the COVID-19 conditions. So introduction is in the pandemic infections. It is clear that those with the weak immune systems are highly susceptible to those infections, and it is a worst outcomes. In this regards, immune enhancing herbs may definitely be helpful for the body to fight COVID-19 infection. So here we provide a review of some immune boosting herbs, and many supplements basically from the medicinal plants may help improve immune response. So what are the different herbs which we can use in the COVID-19? So first herb is so I have focused on the very few herbs over here as for the time concerns. So first herb is the Tinospora cordifolia. That is the Tinospora cordifolia act against the SARS-CoV-2 infections. It is the source of the various types of the bioactive components, including the alkaloids, steroids, glycosides, aliphatics. So spotlights of this current study is to find the essential drug for the COVID-19 disease using the antiviral activity of this compound. So we know that how we are going to use this Tinospora cordifolia. So we generally used to take the kadha, which is mentioned into the Ayurveda. So we generally put the directly stem of this Tinospora cordifolia into the boiling water and get it half, and then we generally use it for to boost the immune systems. So likewise, we have used the Tinospora cordifolia. Second drug which I have included in my presentation is the Azadirachta indica, which is also known as the neem. So this Azadirachta indica is a common tree that will be present in all the houses, and its leaves, barks, trunk, seeds have been already known for its antiviral and antibacterial properties. Already it is proved around the world, and Azadirachta indica had antiviral property against coronavirus too. Next drug is the Gingiber officinale, that is also known as the ginger. So this is the third drug which I have included in my presentation. So the major constituents of the Gingiber officinale belongs to the family Gingiberaceae is the 6-gingerol, which is having the which is act as a promising drugs to treat the COVID-19. Because of this, 6-gingerol possesses the excellent drug likeliness parameters with zero violations of different rules and it has a very good ADME pharmacokinetic properties that is also reported. I have gone through the many reviews, and from that I came to know that. And then the 6-gingerol proves the antiviral. Sorry. 6-gingerol proves antiviral efficacy against the SARS-CoV by showing the highest binding affinity and interactions with the multiple targets of a COVID-19, including viral protease or the RNA binding proteins or the spike proteins. So in this way, this 6-gingerol is going to affect into the COVID-19, means it helps to improve the immune systems. Next drug is I have included is the curcuma longa. So this curcuma may have the beneficial effect against the COVID-19 infection via its ability to modulate the various molecular targets that contribute to the attachment and internalization of COVID in many organs, including like the liver or the CVS, that is the cardiovascular systems, and kidney functions. This curcumin could also modulate cellular signaling pathways such as the inflammations, RNA replications. All I have gone through the many review articles, and after that I have come to know these things. So the curcumin may also suppresses the pulmonary edema and fibrosis associated pathways in COVID-19 infection. And in this way, how the curcuma is going to affect on the COVID-19. Next drugs I have included is the Ocimum santum. Yes, Ocimum santum, which is also known as the Tulsi, and it helps in controlling the COVID-19 because already it is proved that the Tulsi is having the pain, it is acting in the pain and the diarrhea, fever, cough. Generally we use in the day-to-day practice also. So if we have the kadha of this Ocimum santum or decoction of this Ocimum santum, so we'll get the relief from these symptoms whichever we can find into the COVID-19. Next term is the next up is the Allium sativum. It is a potential unique therapeutic food rich in the organosulfur and the flavonoids components to fight with the COVID-19. So from these drugs, there are so many drugs which we can use, like the ashwagandha and so many. So what is the conclusion of my review is that the immune system in the body plays a very important role to fight against unhealthy environment and the microbes such as the viruses, bacteria, fungi, etc. Since these botanical plants having low cost, they are found easily anywhere, and they're having the minimum toxicity. So it has a potential to enhance immunity to fight against COVID-19 and other infectious diseases, and plays an important role to become a fit and healthy. So for this, we can press the preclinical. Yes, sorry. So for this, we can maintain the proper diet, and if we follow the Ayurveda, so we can, these are the preventive measures through which we can prevent the disease conditions. So thank you all for your kind patience and listening to me. Thank you.
Okay, it was a good presentation.
Thank you, ma'am.
Can you list out the components from these plants which are beneficial for treating COVID-19 or suppressing the COVID-19?
I have mentioned the Tinospora cordifolia, that is Gulwell.
That is a plant name, right? I'm talking about the phytoconstituents.
Phytoconstituents, yes. Gingerol is there, ma'am. Some alkaloids, steroids. I have not gone in depth, ma'am, actually. In neem, we can, already it has the azadirachtin, which is having the antiviral activity.
COVID-19 is a, now it is a topic of the time, right?
Yes.
So many reviews are there. So if you are taking that as a topic, you have to have lot of information.
Information, yes,
Only
ma'am.
the plants means it is nothing new. Everybody knows it, right?
Yes, ma'am.
Along with the plants, which are the constituents or the recent research, that also if you have reviewed, that would have been very helpful.
Yes, ma'am. Surely, definitely in future, I will focus on that, ma'am.
Okay, good.
Thank you. Thank you, ma'am.
Thank you, doctor. Thank you for your presentation. Now we are moving with our other presentation, oral presentation by Dr. K. Karpagavali from National Institute of Siddha, India. Her topic is on evaluation of hemantic activity of polyherbal Siddha formulation. Karisalakani Churnam. Ma'am, you can start sharing your screen and you can go with your presentation. Dr. Karpagavali, may I know, are you
yes,
there
ma'am, I'm
online?
sharing. Yes, ma'am, I'm on online.
Yeah, okay, ma'am. You can start sharing your screen.
Yes, ma'am. Good afternoon, ma'am. Is anyone is audible my voice?
Yes, you're audible.
Yes, ma'am. Does my screen is
Yeah, both your screen is visible and
okay,
you're
ma'am.
audible.
Okay, ma'am. Thank you. Today, my presentation is evaluation of hematinic activity of polyherbal Siddha formulation, Karsalangani Churnam. I'm Dr. Karpagavalli presenting it. Introduction to my topic is Siddha system is one of the 6 recognized streams of Indian systems of medicine. It is well received among the masses of South India. Siddha has both safe herbal and herbal mineral formulation and treating various conditions from ulcer to cancer. The herbal formulations, Karsalamani Churnam is also one among them, which is specifically given for anemia, that is more specifically iron deficiency anemia. This study has been conducted in order to prove its efficacy in the treatment of anemia. Anemia is present when the hemoglobin level in the blood has 2 standard deviations below the mean for the particular age and sex. Physiological definition for anemia is a condition in which tissue hypoxia occurs due to inadequate oxygen carrying capacity of blood. WHO criteria for diagnosis of anemia is children of 6 months to 6 years, that is below 11 grams per dl, and the children of 6 years to 14 years, it's for about 12 grams below is considered as anemia. The aim is to find out hematinic activity of Karsalamani Churnam and albino rats. Materials and methods. Formulation was prepared as per SOP. Preparation of animals. Hemoglobin content were estimated, statistically analyzed. The SOP of Karsalamani Churnam is a dried whole plant of Eclipta prostrata, dried whole plant of Borhavia diffusa, dried raceme of Gingiber officinal, dried seed of Piper nigrum, dried fruit of Piper longum, dried fruit pot of Terminalia tribula, dried fruit of Philanthus emblica, dried fruit coat Terminalia bellerica, dried wood of Coccineum fenestratum, dried fruit of Corianthrum sativum, dried root of Glyceris glabra, dried seed of Nigella sativa, dried leaves of Abies spectabilis, dried seed of Elateria cardamom, dried seed of Cuminum simnum or that taken. In this, the ratio is Eclipta prostrata only taken for 4 tola and the others were in an equal ratio 4:1. This is the final product of Karsalamani Churnam. A method of preparation was the previous method mentioned drugs were purified as per the literature evidence, dried in the shade and grind into fine powder. The dose mentioned here is a human dose, 200 mg to 500 mg twice a day, and the adjuvant is honey. The duration is 20 days. Preparation of animals. The animals were taken. The albino rats were obtained from CL Baidmata College of Pharmacy. The rats were housed in an animal house with temperature and rat pellets were provided to the animals and animals at libidum. The animals were daily monitored for their behavior and health. Acclimatization. Prior to any experimentation, the animals were allowed to adapt to the animal house experiment for 15 days. During this period, no experiments were performed on the animals. The males and females were separated and kept in a clean cages. Suitable temperature, humidity, and lighting conditions were maintained and fed and water were supplied at libidum. Cleanliness and hygienic conditions were maintained as required. The animals were monitored regularly for signs of any infection or injury. And grouping of animals as follows. The acclimatized animals were subsequently divided into 5 groups, each consisting of 5 rats, 2 males and 3 females, and tails and ears were marked accordingly. Group 1 constituted the control animals that were not induced for anemia and did not receive any treatment for anemia. Anemia was experimentally induced in animals of Group 2, Group 3 and Group 4. Sorry, except in Group 1, all the other groups were induced for anemia. The Group 2 animals served as anemic control animals that did not receive any treatment with the test formulation. The Group 3 animals received Karslangani churnam 100 grams per kg, and Group 4 received Karslangani churnam 200 milligrams per kg. And the Group 5 animals received conventional chemotherapy, that is allopathic treatment, positive control. Okay, this is the evidence. And the control group is the hemoglobin was measured, and the control group was 16.6 plus or minus 4.1, and Desperal treated animals were 10.5 plus or minus 2.5. Desperal plus Karslangani churnam 100 milligrams treated animals, their hemoglobin is 12.6 plus or minus 2.1. Desperal plus Karslangani churnam 200 milligrams 14.8 plus or minus 3.2. Desperal plus ferrous succinate plus folic acid, the hemoglobin in that rats are 16.1 plus or minus 2.1. Okay, and thus in this study, Desperal plus Karslangani churnam 100 milligrams given albino rats, the hemoglobin noted is 12.6 plus or minus 2.1, and the Desperal plus Karslangani churnam 200 milligrams received albino rats, their hemoglobin raised to 14.8 plus or minus 3.2. Okay, thus this is my conclusion. This study was designed to substantiate the traditional claim of ametonic activity for the polyherbal formulation Karslangani churnam. Preclinical studies are prerequisites for the standardization of the drug. Further clinical trials should be done to prove the effectiveness of the drug. Thank you.
Hello.
Yes, ma'am.
Yeah. Good presentation, but I have few questions. Number one is you have written the quantity as tolas, right?
Ma'am, audible, ma'am.
The ingredients list, you have written the quantity as
yes,
tolas. Tolas.
Yes, ma'am.
Yeah, tolas means like actually when you are taking in your Siddha system of medicines,
okay,
you have to also prescribe as how much we say in the modern system of medicine. Then only others will understand, right?
Okay, ma'am. Okay, ma'am.
Yeah. So what do you mean by 1 tola?
Yes, ma'am. 1 tola is equal to 2.4 grams, ma'am.
Okay. Okay. So I think if you made another column, which is telling 1 tola means how much, it would be useful. And then this study was on animals, right?
Yes, ma'am.
Okay. Okay. Fine. Yeah. Good.
Thank you.
Bhargavi, please unmute yourself.
Bhargavi, you are muted.
Yeah, unmute yourself.
I'm sorry. So, ma'am, we are going with the other presentation by Dr. Vilas V. Shinde from Yashasri Hospital, India, on the topic of novel hyoglymic agents from some Indian medicinal plants. Sir, I hope you are having some good network connectivity now. You can start your presentation. Sir, kindly unmute your mic, sir. It's in mute.
Okay.
Yes, sir. Now you are audible.
Okay.
You can start sharing your screen also.
Just a second. Hi, everyone. Is it visible now?
Yes, sir. Your screen is visible and you are audible too. You can go with
Oh.
your presentation, sir.
Okay, thank you very much. Hi, everyone. Myself Vilas Shinde. Today I'm presenting a review article on novel hypoglycemic agents from some Indian medicinal plants. So, as you may be knowing that diabetes mellitus is very common non-communicable disease which affects all human population throughout the world. It is a metabolic disorder with a common manifestation of hyperglycemia of global prevalence and incidence. Chronic hyperglycemia, which is most observed in uncontrolled diabetes mellitus, causes long-term damage, dysfunction, and failure of several organs in human body. Like we can say kidney damage or liver damage, that sort. But that will be in a chronic conditions. Now, as far as diabetes is concerned, it has got two main types. That is type 1, that is insulin-dependent diabetes mellitus, and second type is non-insulin dependent diabetes mellitus. According to World Health Organization in 2014, it is found that 422 million adults were affected by diabetes, and majority of which were type 2 diabetes. Now here comes the role of our Indian medicinal plants. Nowadays, many drugs are used conventionally for prevention and management of diabetes, such as sulfonylureas, bigonides, metformins, metformins, etc. But still, most effective treatment against diabetes may yet to be achieved. Moreover, use of synthetic drugs has certain limitations for use and many side reactions. The Indian medicinal plants played an important role as they are found to be more useful in the management of diabetes. It is estimated that more than 25% of modern medicines are directly or indirectly are derived from the medicinal plants. Now we have studied about 10 some Indian medicinal plants, and we have this is as you know that this is a review article. So every plant has been studied, like we can say the preparation of the extracts and their hypoglycemic activity using different diabetic animal models. So Ajati Rasta indica that you might be knowing neem leaves. It's an ethanolic extract in rabbits that is an alloxan-induced diabetic model that is also hypoglycemic activity. Next is Caesarelia esculenta. The part is roots and it's aqueous extract of in male albino rats, which is in streptozotocin diabetic mice. Next is Eucalyptus chloridura leaves, which is its aqueous extract that was tested in albino rats, and it was confirmed in alloxan diabetic model. Next is Momodica charantia. Its fruit is ethanolic extract of the fruit is quite tested in rabbits, and which was again tested with the alloxan-induced diabetes mellitus. Then Phyllanthus neruri, Piper longum. So these are all we can say the most commonly used the plants which we are using in diet also. Like for example, Seasonum jambolana, jambul seeds, even methi seeds. So these are all the plants which we have just reviewed and selected for this further studies. Now the important thing about this medicinal plant is that they are having the bioactive principles, like we can say alkaloids, flavonoids. So these are the most active compounds, and these are generally prepared by using the different solvent systems, or we can say column chromatography. They are separated and they are tested in experimental models. And we have selected the most hypoglycemic agents, the plants, for these further studies. And our aim, like we can say, we can conclude from this review that number of diabetic patients has been increasing dramatically over the past few decades and demands special attention towards diabetes management. As we discussed before, the conventional therapies are either expensive and often related with the side reactions, like we can say sulfonylureas, type vitamides. They are having certain side reactions also. Therefore, medicinal plants provide a better opportunity as they are less toxic and affordable than synthetic drugs. Like as we are, since morning, we are just discussing about this, many herbal drugs or herbal medicines, they are quite available, most affordable price, and even they are most potent also. So considering all these factors, so further research will be appreciable for studying these novel hypoglycemic agents into the therapeutic segments of diabetes mellitus. Yeah, the references. Thank you very much.
Hello, sir.
Yes, ma'am.
I was very much exciting seeing your title.
Yes.
But when you're presenting, I didn't find that what is the novel hypoglycemic agents from the plants. Nowhere you mentioned that.
Yeah, that is, ma'am, it's a prelim review. So, I mean, in the next references, we may be having the information about that.
But your title should be what you're presenting, right, sir?
Yes, that I will take care.
Your presentation and title is not at all matching. And number two is, you are giving reference of 2014. When you are reviewing now, which is 2022, and you are giving dated back 2014. So many research might have done it during that period of time.
Yes.
So you should have been given reference.
That is there, but yes, ma'am.
Which is
tell me.
recent. Not 2014 is very old, right? So the data should have been given 20, 21, 22. That will show you a proper review.
Okay.
I can see that you have collected a good number of things, but the collection should be from the recent. So that will be the proper reviews.
Okay, ma'am.
Okay, so thank you.
Thank you very much.
Yeah, good afternoon.
Thank you for your presentation, sir. I
Yes.
request all the participants to mute their mics. Ma'am, now we are going with the other presentation by Dr. Nityamala Indrakumar from National Institute of Siddha from India. The topic of her presentation is antiplatelet aggregation activity of Siddha drug. Ma'am, you can go with your presentation.
Ma'am, I have a question. I have a question for the Monday, sir. Ma'am, I have a question for you.
Yes, sir. Please tell me. Please tell me, sir.
Yes, sir. Good afternoon, sir.
Yes, sir. Good afternoon. Please let me know your query.
It's a nice presentation. Sir, my question for you, sir, you have taken the plants which are using for the hypoglycemic only in which system of medicine, sir? You have taken the plants, which system of medicine? Is the plants are using which system of medicine? Ayurvedic system of medicine, Unani or homeopathy, like that. My question is that, sir.
So this question is to Dr. Vilas V. Shinde. Hello. Hello. Dr. S.N. Babu sir, you can text your query in the chat box so that Dr. Vilas sir will be answering your questions in chat box.
S.N. Babu sir, can I reply your question?
Yeah, my question is that the sir is explaining about the hypoglycemic, all these hypoglycemic, what he has taken the review. Is these drugs are medicinal plants with the hypoglycemic activity? Is these drugs are taken from which system of medicine? Whether the Ayurvedic system, whether the homeopathic system of medicine, whether Unani system of medicines? Is a review? But what is the potency and what these drugs are using in various systems? My question is that whether Azadirachta, Dragonella, Syzygium, Piper, Philanthus, these drugs are taken which system of medicine drugs?
Sir, he has taken the articles for the research. Whatever the research has been done based on any system of medicine on the medicinal plant which is used as a hypoglycemic agent.
It's not clearly mentioned.
Not specifically which system of medicine. Whatever the research has happened, he has collected the article and then he has just summarized it.
Can we get the providing about all these things in the further? So
yeah, you can start doing review, sir, in that way. Your system of medicine, you can start doing review in your way.
So, that's I'm finding out that because of I couldn't find out the Azadirachta and some of the drugs, the review. Thank you, madam.
Okay, sir. Okay.
Excuse me,
Yeah,
ma'am.
thank you, sir. Ma'am, shall we go with the other presentation now?
Yeah, yes, sure.
Okay, ma'am. Now I would like to invite Dr. Nityamala Indra Kumar from National Institute of Siddha, India. Her presentation topic is on an anti-platelet aggregation activity of Siddha drug. Ma'am, you can go with your presentation now.
Good afternoon, ma'am. I am not able to share the screen since the other screen is shared.
Yes, ma'am. Dr. Vilas Kumar sir, Vilas sir, I request you to stop sharing your screen so that another participant will able to share.
Hello.
Yes, sir. Dr. Nityamala ma'am, now you can try.
Okay, ma'am. Now you are able to see the presentation, ma'am.
Yes, ma'am. Yes, ma'am. We are able to see, and your voice is also audible.
Okay. My topic is anti-platelet aggregation activity of Siddha drug Icharamuli Churnam. I am a research scholar, Dr. Nityamala from National Institute of Siddha. Introduction. Indian system of traditional medicine like Siddha pioneers the art of healing from ancient time as it is systematically documented and well-established practice guideline framed by the traditional healers, otherwise called as Siddhas, ancient physicians. Siddha text prescribes several lakhs of medications and formulations that are indicated for various diseases. Aristolochia indica lin, commonly known by its vernacular name called as Indian birthwort, family Aristolochiaceae. In Siddha medicine, it is called as Ichuramuli. In southern parts of Asia, this herb is considered to be possessing potential medicinal properties. Cardiovascular disease cause increased percentage of socio-economic disability around the globe. According to the recent statistic, CVD associated with comorbid conditions like dyslipidemia, diabetes, obesity, poor dietary habit, custom-made social habituation like tobacco and alcohol intake have increased the tendency to cause and worsen the condition of CVD. In vitro studies, in CVD, the worst scenario is the abnormal accumulation of platelets on the biologically significant surface of the blood vessels. Increased platelet aggregation contributes to pathologically important factor in the physiology of CVD, which might be the result of multiple different mechanisms. In vitro alternatives provides considerable evidence-based results for treating CVD. Collection and authentication. Plant materials were collected from southern zone of Tamil Nadu. The collected specimen was identified and authenticated by the Director, Plant Anatomical Research Center, Chennai, Tamil Nadu, India. The specimen voucher was deposited in the National Institute of Siddha, Chennai, 47. Aristolochia indica lin. It is commonly known by its vernacular name Indian birthwort. In southern part of Asia, this herb is considered to possess potential medicinal properties. Distribution: this plant is a perennial climber with greenish white woody stems found in low hills and in plains of India, Nepal, and lower Bengal to Chittagong in Bangladesh. Phytocomponents: this plant is known to have p-coumaric acid caryophyllin. Materials and methods: formulation of Icharamuli churnam. Collected plant materials was washed thoroughly and were extended for shade dry procedure, followed by the formulation as per the standard method. Dosage 1 to 2 gram twice daily, and indication: heart diseases, Irudaya Rogam, as mentioned in Asuda text. Antiplatelet aggregation activity was carried out by using the platelet-rich plasma 0.13 into 10 power minus 7 for each assay, resuspended in thyroid buffer pH 7.4. Aggregation of platelets was induced by calcium chloride at a final concentration of 2 micrometer. The platelet aggregation was documented by the transmittance value of spectrophotometric measurements at 600 nanometer. To determine the in vitro platelet aggregation characteristics, various concentrations 100, 200, 300, 400, 500 microgram per ml of Icharamuli churnam was added to the platelet suspension for 1 minute, exposed at 37 degrees Celsius before treatment with platelet aggregating agents. Aspirin at 500 microgram per ml was used as the standard. Antiplatelet aggregation was calculated using the below formula. AP is equal to AT minus AC by AT into 100. AP, the percentage of antiplatelet aggregation. AT, absorbance of the extract and calcium chloride treated cells. AC, absorbance of the control sample. Statistical analysis was carried out using GraphPad Prism 5 computer program. Results: the effect of EC on antiplatelet aggregation activity. In vitro antiplatelet aggregation characteristics of Icharamuli churnam at various concentrations 100, 200, 300, 400, 500 microgram per ml. When compared to standard drug aspirin showed 91% mean activity. 100 microgram per ml 27.93, 200 45.33, 400 microgram per ml 69.93, 500 microgram per ml 80.67. The control was the standard drug was aspirin 91%. 500 microgram per ml showed 80.67. This proved the antiplatelet aggregation activity of the test drug Icharamuli churnam. It is tabulated. The same results are tabulated: 100 27.93, 200 45.33, 300 69.63, 400 74.67, 500 80.67. It is the pictorial representation. Conclusion: in the present study, in vitro antiplatelet aggregation characteristics of Icharamuli churnam evaluated at various concentrations. Aspirin showed 91% mean activity, and the results were 27.93, 45.33, 69.63, 74.67, and 80.67 for the various concentrations of the Icharamuli churnam. This proved the antiplatelet aggregation activity of the trial drug Icharamuli churnam. Thank you.
Thank you for your presentation.
Okay, ma'am.
Which was, you have done in vitro studies, right?
Yes, ma'am.
Okay. So what was your, what you have used for in vitro studies?
Ma'am, aspirin was used as the standard, ma'am.
Okay.
The platelet-rich plasma was suspended in thyroid buffer. Aggregation was induced by calcium chloride, ma'am.
Okay, how did you collect the platelet?
Ma'am, actually this study was done in Satyabama University, ma'am.
Okay, but you should know the procedure, right?
Okay, ma'am.
Okay, fine. Thank you.
Thank you.
Yeah, thank you, Dr. Nityamala. Ma'am, shall we go with the other presentation now?
Yeah, please.
Yeah, okay, ma'am. The next presentation is by Dr. B. Anusha from Sri Satya Sai Institute of Higher Learning, India, and the topic is on assessment of anti-diabetic and anti-hemolytic activities of aerial parts of Trichosporium amiel. Dr. B. Anusha, you can start sharing your screen and you can go with your presentation. Dr. B. Anusha? May I know, are you there online or not? Ma'am, I hope she might having some network issues. So we'll be moving with the other presentation by Dr. Sheikh Sana from YB Chevan College of Pharmacy. Dr. Sheikh Sana?
Yes, I am there, ma'am.
Yes, ma'am. You can go with your presentation, ma'am.
Yeah, sure.
Thank you, ma'am.
I hope my screen is visible.
Yes, ma'am. Yes, ma'am. You are visible and your audio is also audible.
Okay. Good afternoon, one and all. First of all, I would like to thank Startup Conference for giving me this opportunity to present my poster today. Respected Dr. Rukaiya, my topic for today's presentation is antifungal activity of various solvent extracts of Costus speciosus and Costus pictus and their preliminary screening studies. I am carrying out my PhD work under the kind guidance of Dr. Abu Bakr Salam Bawazir, in charge principal, YB Chauhan College of Pharmacy. As we are all familiar with oral candidiasis or oral thrush, that is one of the most common fungal infections. The main causative agent behind this mycosis is Candida species, mostly Candida albicans. Many traditional medicinal plants have proven to be effective against oral candidiasis. One of such plant species, Costus, is also very effective and show potential antifungal activity. My main objective of the work to assess antifungal activity of various solvent extracts of both the plant, that is Costus speciosus and Costus pictus. Solvents such as hexane, methanol, ethyl acetate, and water were used for the extraction process. These solvents were selected on the basis of thorough literature survey. The plants were procured from local nursery. They were collected, authenticated, cleaned, and further they were converted in the form of extract. They were further converted in the form of extract. They were also given a wide accession number and were authenticated from Dr. Bamu University, Aurangabad. Basically, the leaves of Costus pictus and the rhizome part of the Costus speciosus plant were used for the particular activity. Maceration process was found to be suitable for extraction of active constituent present in the plant. One of the method, that is agar well diffusion method, was used to determine the antifungal activity against 5 different Candida species as mentioned in my poster. A known quantity of extract was transferred in well containing pre-swabbed agar plates. Clotrimazole was used as a positive control and the results were recorded after 48 to 72 hours of incubation. From all the extract, hexane extract show a strong antifungal activity overall, while the methanolic extract shows a lesser antifungal activity against particular strain such as Candida glabrata and Candida parapsilosis. Ethyl acetate extract exhibited activity against certain strain such as Candida glabrata, while aqueous extract exhibited no antifungal activity against the various fungal strains. The zone of inhibition was recorded for each extract, and the maximum zone of inhibition was shown by hexane, that is 34 mm against Candida albicans species. Thus, from the present study, it can be concluded that both the plants demonstrate promising antifungal activity, and in future, additional work can be executed to elucidate possible mechanism of action and active constituents responsible for the same. I would like to thank the team Startup Conference to giving me such a huge platform to share my part of my research PhD research work. Thank you so much.
Hello.
Yes, ma'am.
Yes, Sana.
Yes, ma'am.
Good presentation.
Thank you, ma'am.
But you are mentioning maturation, right?
Yes, ma'am.
Yeah, and you are using how many solvents?
I am preliminary. I have used 4 solvents, from which hexane extract showed good activity against all the Candida species.
Okay, so what you did, can you explain the method of maceration, how you did?
Yes, ma'am. I had taken a certain amount of drug. Like initially, I have taken 2 grams of drug in around 500 ml of hexane extract, hexane and other solvents like ethyl acetate, methanol, and water. Then I have, yes.
All separately?
Yes, all separately, ma'am.
What made you to choose this method of extraction?
Ma'am, first of all, the main reason for choosing maceration was that the constituents which are present in both the plant are heat sensitive. So above the 60 degrees Celsius temperature, I had gone through the literature. So it is mentioned in the literature that above 50 degrees Celsius, the phytoconstituents get degraded. So mostly the maceration technique is used for extraction of all the phytoconstituents from the plant.
Okay, okay. So you have chosen based on the polarity and you have found hexane extract is most effective, right?
Yes, ma'am. Yes, ma'am.
What might be the reason you have done the preliminary screening also?
Preliminary studies for this, ma'am. You are asking for antifungal activity?
I'm asking for the what may be the constituents.
In hexane extract, we have got certain constituents like octadecanoic acid, hexadecanoic acid, and so on, which is already in other plants. They have already proved that these phytoconstituents can be responsible for the antifungal activity. So further, ma'am,
I'm asking your preliminary screening you have done, right?
Preliminary screening of what, ma'am?
Of your extract?
Yes, yes, I had done.
Phytochemical screening you have done,
Yes,
right?
yes. Phytochemical I had done.
What you have got? What category of the constituent?
Ma'am, we have got flavonoids, alkaloids, and terpenoids. So basically, the main phytoconstituents which are responsible are terpenoids, that is sesquiterpene lactones, which are present in both the plants. Hello?
You hear me?
Yes, ma'am. Your voice was breaking. Now I can hear you.
Okay. So it's a good work. So
Thank
I
you
can
so much.
suggest you to go for GCMS studies.
Ma'am, I had done my GCMS studies as well, HPTLC confirmation as well as TLC confirmation. But as far as this is my PhD work, so I have not disclosed all the data in this particular poster presentation. And this particular work is also published in JMPS journal, which is a Scopus and UGC approved journal, ma'am.
Yeah, that's a good, but what happens is you are just telling you have done extraction, you have done antifungal activity.
No, ma'am, the work is quite big, but this is just a part of my work. So if you want to ask further questions, I can answer, ma'am.
No issues, but you could have been just mentioned one line. The constituent was confirmed using this, and then you could have been done the antifungal activity.
Yeah, ma'am. Okay.
Yeah, okay, good work.
Thank you so much, ma'am. Thank you.
Thank you, Dr. Sheikh Sana, for your presentation.
Yes.
Now, I would like to invite Dr. B. Anusha for her presentation from Sri Satya Sai Institute of Higher Learning, India. I hope Dr. B. Anusha is available now.
Yes, ma'am. Yes.
Yes, ma'am. Thank you for the confirmation, and you can start your presentation.
Just a minute, Bhargavi. I am unable to stop my screen share. So,
bottom of your screen, then you can find the option of stop sharing.
Yes.
Yeah.
Stop. Okay. Thank you.
Thank you, ma'am. Dr. B. Anusha, you can start sharing your screen now.
Good afternoon. Am I audible to you?
Yes, ma'am. Your screen is visible and you are audible.
Yes. Thank you. So presently, I will be talking on phytochemical screening and as well as the assessment of medicinal properties of Trachispermum ammi, that is a ajwain plant. So before going to the further details, I would like to add some introduction to the phytochemicals. So we know about the phytochemicals. They are very much useful, like they're commonly called as the botanicals, and they are going to act as complex drugs with multiple potential targets and actions.
And these botanicals are more economical than the synthetic drugs, and there are no side effects. So henceforth, they are more advantageous over to the allopathic medicine. And here are some of the bioactive compounds which has been extracted from some of the plants, showing the therapeutic effect. Like we have well-known Catharanthus roseus, from which the vinblastine and vincristine has been isolated, and Rauwolfia serpentiana reserpine, which is going to show the anti-hypertensive activity, and Taxus brevifolia, Taxol, which is a well-known anti-tumor effect. And then like we have cinchona, which is which is having the quinine, so which is going to act like an anti-malarial drug. So the present plant that I have chosen for my work is the Trachysperma mummi, which is the ajwain plant. And here, like many studies have been done on the seeds, but not on the aerial parts. Henceforth, we were investigating on the aerial parts of the Trachysperma mummi. So it's commonly called as ajwain, and it is called by different names. It's commonly called as bishop's weed in English, and it is native to Egypt. Even though it is native to Egypt, in India, it is mostly grown in Rajasthan and as well as Gujarat. So, and there is a misconception between these two plants, that is Trachysperma mummi and as well as a Plectranthus umbilicus. So many people think that Plectranthus umbilicus has the Trachysperma mummi, but it is not so, because we have cultivated the seeds and we have cultivated the plants by sowing the seeds. So we had done the antioxidant effect primarily. So like we know very well about the oxidative stress, which is the imbalance between the pro-oxidants and as well as the antioxidants. So whenever our antioxidants in the system goes down, so then our pro-oxidants rises up, and that leads to the oxidative stress, and that is going to affect nucleic acids, proteins, lipids, and as well as the carbohydrate. And that leads to a number of diseases like oxidative stress disorders of the diseases like cancer, diabetes, neurodegeneration like the Alzheimer's, Parkinson's, and it is also going to cause asthma, infertility, atherosclerosis, and so on. And there are various herbs and spices which are going to show good oxygen radical absorbing capacity, like clove, cinnamon, oregano, turmeric, and all. So we were looking for our plant, that is the Trachysperma mummi, whether it has the antioxidant effect or not. So this is the methodology that we had taken. So the ajwain plant parts which were grown in the campus, it was shade dried and as well as powdered. And we had extracted it with methanol thrice 24 hours at room temperature. And then we have got the methanolic extract, and later the fractionation, sequential fractionation has been done from non-polar to polar the solvents by using the hexane, which is the non-polar, and then dichloromethane, then ethyl acetate and butanol. And finally, whatever residue was left, that was the aqueous fraction. So then this is the methanolic extract and various fractions of the methanolic extract, and we had actually looked for all these activities. So anti-diabetic, anti-hypertensive, and then anti-inflammatory activities and so on. So the objectives of the study was like to test for the anti-inflammatory activities. So we had done the bovine serum albumin denaturation assay, and also inhibition of hyaluronidase and inhibition of xanthine oxidase activity. So this, the results section, coming to the results section, like when we have seen the radical scavenging activity by DPPH and as well as nitric oxide radical scavenging assay. So we have got wonderful results, especially with the ethyl acetate fraction. And these were some of the results, the preliminary results. These were the qualitative analysis that we had done. So just to test whether the carbohydrates, presence of carbohydrates are present in our fractions and as well as the extract. And then here like amino acids, and then here we see the qualitative test for flavonoids. Here the ethyl acetate fraction and the N-butanol fraction has shown the maximum flavonoids amongst the other fractions. And this is the tannins and as well as the phenols. So mostly dichloromethane fraction has shown the better tannins and phenols, and next was the N-butanolic fraction, and next was the ethyl acetate fraction. And detection of steroids and terpenoids was also done as to test the steroid and terpenoids, so in the fractions. And this was the inhibition of bovine serum albumin denaturation assay. And in this, like we could see the DCM fraction, that is a dichloromethane fraction of the methanolic extract, it had shown the significant effect, followed by ethyl acetate and butanol and aqueous and hexane fractions. And we have used the positive controls, that is the BHD and the synthetic antioxidants like lutein and quercetin. And then inhibition of xanthine oxidase activity here as well. The butanol fraction of the methanolic extract has shown the best activity amongst the other fractions. And likewise, the inhibition of hyaluronidase activity here, we saw the ethyl acetate fraction showing the significant inhibition compared to the other extracts. So even the DPPH activity, like if we see, the N-butanol fractions have shown the best results compared to the other fractions. So it has proved that, and even the nitric oxide radical scavenging assay also, like we have seen the ethyl acetate fraction showing the best results. So henceforth, like we have concluded that the compounds which are present in the ethyl acetate fraction, like the flavonoids and also the butanolic fraction. So mostly the polar compounds like the flavonoids gets extracted into this. So they were causing the effect of all this. And the plant extracts can be considered as a significant source of natural antioxidants, and it can antagonize the deleterious effects of pro-oxidants, and also it can prevent many diseases which are caused by the oxidative stress like cancer, atherosclerosis, diabetes, inflammation, and aging. Thank you so much.
Good presentation, but when you are going for qualitative studies,
yes, ma'am,
you are writing 1+, double plus, triple plus.
Yes, ma'am.
What do you mean by triple plus and double plus?
Yes, ma'am. So double plus is like more amount of
but how do you know
is present? Like for example, based on the color, ma'am, like we have got the color. So if high intensity of the color, because we know very well that these qualitative analysis, they are all
but
color
based. Can we tell in the qualitative test, it is more, it is less? We cannot say, only we can say present or absent, right? When we are doing quantitative, then only we can define it is more, it is less, this much more, this much less, right? We cannot write like this, the 1+, double plus, single plus, which is showing the less, maybe having less extract you have taken, so it is showing less.
Actually, what we have done was we have taken in a like 10 mg per ml, and then we have checked, and then each fraction was showing different color. So we have
even though you have taken the 10 mg per ml, yes, but you have dissolved in a suitable solvents, right?
Yes, yes.
What was the solvent you have dissolved?
Ma'am, first initially it's the methanol.
Okay.
And then later, like we had taken the non-polar solvent like the hexane and then the dichloromethane.
So first when you take the methanol, yes, so other constituents will not, which is not soluble in methanol, will not come, right? Because methanol is a polar solvent.
Yes, ma'am. Ma'am, actually what we had done was we had taken the entire powder and we had mixed it in a methanol. We have maceration, it's just a process of maceration.
I understood.
And then after, yes, ma'am.
What is my concern is you cannot write double plus, triple plus.
Okay,
Only
okay.
qualitative, right? So just
Okay.
plus or minus.
Okay.
If you want to further confirm, please go for the quantitative estimation.
Okay.
There you can say that it is having this much more amount of flavonoids by doing total flavonoid content or total phenolic content, and you can tell there it was more.
Okay, okay, ma'am. Thank you for your good suggestion. Thank you.
Yes, thank you, ma'am. Now we are moving with the other presentation by Desai Priya Ajit Kumar. This is her second presentation, ma'am, from Tatya Pharmacy College, India. And the topic is a comparative evaluation of anti-cancer effect of Panchagavya and Panchagavya Grita by Levy's lung carcinoma cell line. Dr. Desai Priya, I hope you are there online. Dr. Desai Priya. Okay, ma'am. Moving with the other presentation by Divya Iyengar. Dr. Divya Iyengar, could you please confirm us with your present? Yeah,
Yes.
okay,
Am I
ma'am.
audible?
Dr. Divya Iyengar from Saraswati Vidya Bhavan's College of Pharmacy, and her topic presentation is in silico screening of the phytoconstituents from Ayurvedic polyherbal Panchakaval preparation against fungal targets and development of modern formulation for fungal infection. Dr. Divya Iyengar, you can continue with your presentation.
Hello, is my screen visible?
Yes, ma'am, it is visible and you are audible too.
Yes, yes. Thank you so much for confirming. So a very one and all. I am Divya Iyengar, and I'm pursuing my master's in quality assurance department. It is my immense pleasure that I'm getting this opportunity to present my study with all of you today. The title of my study is In Silico Screening of Phytoconstituents from Ayurvedic Panchavalkal Preparation against the fungal targets and development of a modern formulation for fungal infection. So, wherein a complete focus is made on learning the antifungal activity of phytoconstituents against a familiar fungal target. Since there are increasing number of fungal infections, which can be both, say topical, which can be ringworm, or it can be atlas foot, or it can also be systemic, wherein the Candida albicans infection of throat or mouth. The main problem with the regular marketed antifungal agents is that their resistance and their side effects. So the main aim of this study was to get an agent from natural origin where they will have low toxicity and they will be safer to use. So on the similar lines, I have found this modern Panchavalkal in the literature, wherein they have proved that these have antimicrobial activity as well as wound healing activity. So what is modern Panchavalkal? Modern Panchavalkal is a combination of 5 drugs. In the name itself, as we can see the word punch, it is combination of Ficus bengalensis, Ficus religiosa, Ficus infectora, Ficus leccor, and Albizia lebec. So the aim of this project was to evaluate the antifungal activity of its phytoconstituent against the fungal target. And the method used was molecular docking. And then developing a modern formulation for fungal infection. As a part of experimental work, the first step was selection of ligands and then identifying the fungal targets. So a past test was done to know whether your phytoconstituent is having the activity, antifungal activity or not. So the past test is that prediction of activity spectra for substances, wherein all of these 10 phytoconstituents, which is present in Panchavalkal, was tested for its antifungal activity. There you get 2 values, that is PA and PI, wherein PA is the probability to be active and PI is the probability to be inactive. As you can see, almost all the 10 of the phytoconstituents are having PA value approximately near to 0.5. That is, this assures you that each and every phytoconstituent is having some or the other antifungal activity. Then a Swiss ADME prediction was done to know the pharmacokinetic. Hello, any problem?
No problem. Sorry for the interruption, ma'am.
Okay.
Ma'am, Pushpalata ma'am, I request you to mute yourself, and I request to each and all participants to mute your mics so that it will not create any disturbance during the presentation.
Thank you. So I would like to continue with Swiss ADME prediction, wherein all the 10 phytoconstituents were tested for their H-bond accepting ability, H-bond donors. For instance, that to make sure that they are forming H-bond with the fungal target. And then TPSA was checked, that is TPSA is topological polar surface area of the molecule, wherein you can see if your TPSA value is nearby 100, then you can say that that is having a good GI absorption. Whereas if your TPSA value is nearby 60, then you can say that it would have a good blood-brain barrier penetration. And then log P was checked to see the lipophilicity of the phytoconstituent, log S to check the solubility, and then GI absorption was checked. And finally, Lipinski's violation was checked, wherein you can see as listed in the table itself, 6 of them are violating one of the Lipinski's law, which is that the log P should always be less than 5. Moving on to study the pharmacokinetics of these phytoconstituents, we had conducted a ADMET prediction test. ADMET is testing the absorption, distribution, metabolism, excretion, and toxicity present with this phytoconstituent, wherein in case of absorption, we had tested this CACO2 permeability, which is actually human intestinal absorption testing, wherein you can see the CACO2 permeability is optimal, so that we could analyze that there is a positive human intestinal absorption. Then as a part of distribution, as we always know that the more amount of plasma protein binding will indicate that our drug is properly distributed in the body. So we can see almost it is approximately near to 50%. Therefore, it is having a medium plasma protein binding. And then blood-brain barrier, here, as I already said, that the TPSA value is approximately nearby 60 or below 60. So we are having BBB barrier penetration as positive. And then as a part of metabolism, the cytochrome P450 family was analyzed as whether the phytoconstituent is non-inhibitor or it is a substrate for the cytochrome P450, because cytochrome P450 is a super family of enzymes which actually oxidize steroids and then xenobiotics, fatty acids, which is very important in clearing of these substances. And then as a part of excretion, I studied the T half-life. And then for toxicity, it was checked that hepatotoxicity is present or liver injury is seen. These are the same ADMET test for the next 5 constituents. As I already said, there are total 10 phytoconstituents in modern Panchavalkar. So in the similar basis like that past 5, these also have optimal CACO penetration. These some of them except gallic acid and catechin have good intestinal absorption, whereas gallic acid and catechin does not have that perfect intestinal absorption. When comparing the plasma protein binding, gallic acid has low plasma protein binding when compared to others. And then here also we can see the CP450 inhibitors, non-inhibitors, and some are substrates. And then for excretion, T half-life is approximately near to 0.7 or 1. And then human hepatotoxicity and drug-induced liver injury. And then this molecular docking was done. The proteins used for this molecular docking was actually prepared by Biova Discovery Studio, which is a software which actually helps us or assists us to prepare a protein before molecular docking. And then molecular docking was done with the help of autodocking. Docking of both the reference ligand as well as the phytoconstituent was done. Here you can see the binding energies are given. Here these are the binding energies of the phytoconstituent, and this is the reference ligand, and then this is the fungal target that was tested. This fungal target is specific for this reference ligand. This reference ligands are the main antifungal products which are present in the market. As you can see, the boxes which are highlighted in red, these have more binding affinity towards the fungal target as compared to the reference ligand. So this assures us that these phytoconstituents present in modern Panchavalkar is having more affinity towards the antifungal targets when compared to posaconazole or any other antifungal agent which is present in market. So we can see that modern Panchavalkar is a promising compound for antibiotic and as an antifungal agent. And then this is 2D interaction of beta-cetosterol with fungal target and 3D interaction of catechin with their fungal target. And then to move on with the study, a in vitro antifungal activity of polyherbal extract was checked against Candida albicans. Here, the ketoconazole drug was used as a positive control and DMSO was used as a negative control. And here you can see the readings of the polyherbal extract. The zone of inhibition is given in mm, and this was the dose mg per ml. And then these are the readings for ketoconazole. As you can see, the polyherbal extract is giving a comparable result with ketoconazole. Then moving on to the other part of the work, a main focus was kept on preparation of a dusting powder formulation under the brand name of Pan-B. Here, this is the formula which was used to prepare the dusting powder formulation, wherein there were 4 formulations that was done, in which 2 of them were done with normally air-dried extract because the extraction was done using methanol. So it was dried using a vacuum oven, and 2 of them are done with spray-dried extract, and 2 with talc as base and 2 with cornstarch as base. This is how the container looked and how the powder looked like and its label. Moving on to evaluation of the dusting powder. In part of evaluation, the 2 main things was evaluated, that is micromeretic property, since it is a dusting powder, and then phytoconstituent testing of alkaloids, carbohydrates, and so on. So the extraction yield, as I already said, extraction was done using Soxite extraction with methanol as solvent. And this extraction yield was found to be approximately nearby 6%. And then, as I said, we did 2 types of drying, that is vacuum oven drying and spray drying. So in spray drying, yield was 4.9%. The pH was found to be weakly acidic, that is 5.5. Then coming to the results of micromeritics. In micromeritics, various parameters like angle of repose, bulk density, tap density, Hosner's ratio, and Carr's index was analyzed. And checking this among the 1, 2, 3, and 4 formulations, the formulation 4 had all these properties properly. This formulation 4 had all these properties in balance. Therefore, formulation 4 was selected. Then results of phytochemical testing. As a part of phytochemical testing, the phytochemicals were tested that the drug powder, the crude drug powder of Ficus bengalensis, Ficus religiosa, Ficus racemose lacquer, and Albizia lebec were individually tested for alkaloid, for carbohydrate, tannins, saponins, flavonoids, steroids. And these were found to be positive. So by this, I would like to conclude stating that this study revealed that there are many phytoconstituents that act as a potent inhibitor of fungal targets. And by assessing these various phytoconstituents, it is concluded that Panchavalkal exhibits the antifungal activity mainly due to the phytoconstituents that are beta-cetosterol, catechin, sigma-sterol, fredalin, amyrin acetate. The in vitro antifungal study of Panchavalkal showed zone of inhibition as that of standard ketoconazole against Candida albicans. Among the 4 formulations, the formulation 4 was found to be more suitable, more apt, containing a spray-dried extract in cornstarch base. Therefore, a further study can be conducted to commercialize a formulation under the brand name Pan-B dusting powder. These are certain articles that I referred during my study. And last but not the least, I would like to thank my college for allowing me to conduct these studies in the college itself. And thank you so much for Startup to giving us this platform to express our ideas and views. Thank you.
Yeah, very good work, excellent work.
Thank you, ma'am.
Is it your M.Pharm work or PhD work?
Ma'am, this was actually M.Pharm work. I'm actually studying on this. I'm doing my master's 2nd year.
Okay. Very good work. One question, why you have selected only methanol as a solvent?
Because ma'am, all of my phytoconstituents were soluble in methanol only. So I thought it would be better.
No, if it is soluble in methanol, that means it might be soluble in other polar solvents also, right?
Yes, definitely.
Have you got any review of literature which shows methanol?
Yes, ma'am. My senior has done this method of development, HPTLC method development over there. So she had used that. So I followed it only.
Okay. But she has not published?
Not yet, ma'am.
Okay, fine. Okay, very good work.
Thank you, ma'am.
Yeah, all the very best.
Thank you.
Thank you, Dr. Divya Iyengar, for your presentation. Now we are going with the other presentation, poster presentation by Sabina Hussain Syed from Ishwakarma University, India, and her topic is on health assessment of toxicity and safety. Dr. Sabina Hussain, I hope you are there online.
Yeah, hi everyone. Thank you, Bhargavi. Let's.
Yes, ma'am, you can start sharing your screen now.
Okay, thank you. Yeah. Is it visible, Bhargavi?
Yes, ma'am, it is visible and your voice is also audible.
Okay. Good afternoon, everyone. Myself, Dr. Sabina Hussain Syed. And I thank the platform for giving me this opportunity to present my paper. So today I'm going to talk about the assessment of toxicity and safety regarding the herbals. As we all know that since morning we are seeing that how the herbal medicines are having where the herbal medicines are having edge over conventional medicines. But sometimes this is not always it is not going to be the case. There are certain events where herbal drugs are also having toxic effects on the human beings. So I thought that it is going to be promote some awareness among all the participants. So let's start with this. So globally now, most of the human populations are diverting since COVID times. Since COVID times, more human population are edging over the herbal formulations. Now, if you see the recent statistics which I have placed over here, that it is projected that from this herbal industry is going to be going from $165 billion industry to the $347 billion by 2029. So you can see the huge market regarding the herbal use. So that is good for all the pharmacognosis, but there is also as this usage is increased, we have certain responsibilities to minimize those toxic effects and produce some awareness regarding the usage of these herbal medicines. So in this context, I want to tell you, highlight some of the points where the herbal toxicity is going to be two factors are going to be majorly two factors are responsible if you take the toxicity levels of the herbal products. So it is going to be the extrinsic or also intrinsics. So first let's go with the intrinsic adverse effect. Intrinsic adverse effects means the adverse effects are going to be present within the herb itself, and it is going to produce certain toxic effects if you are not going to be used within the limits. Okay, so there is a notion among all of us that herbal are always safe drugs. They do not produce any side effects, and we can take any amount of the drug that is being prescribed. But this is not always the case. If you see certain well noted example, that Aristolochia and Aconitum, these drugs, if you are taking in a increased quantity due to this aristolochic acid, it is proved that that leads to the carcinogenic. It is carcinogenic, means it leads to cancer, and also it is Aconitum alkaloids. Now these Aconitum alkaloids are also having this carcinogenic effects. Now next one is the kava. As we have all been heavily dependent upon the herbals for nutrition, but all the nutrients are not safe to use. Like for example, kava, which has been reported for its hepatotoxicity. Like ephedra, echinacea. Ephedra, we all know that ephedrine is the alkaloid which is having sympathomimetic action. But if you are taking this ephedra, in the higher quantities, in the more than the prescribed quantities, it also leads to the cardiotoxic effects. So within the herb itself, there are certain qualities which may promote some toxicity. Now coming to the extrinsic adverse effects, like in extrinsic means which are not within the herb, but there are certain other factors which may also promote the toxicity towards the herbal drugs. So among them is the adulteration, lack of authentication, and contamination. Now in the contamination, if the processing is not been done according to the GMP manufacturing guidelines, then that leads to contamination well-known contaminants includes the lead, mercury, cadmium, and arsenic metals, heavy metals, which may have this toxicity on almost all the organs. And also microbial and fungal contaminations are also been well noted in the literature, that it leads to the contamination of the herbal drugs, with this leads to the toxicity. Most importantly, this herbal and contamination, like for example, we are going to take the crude drugs which are being underground levels, like for example licorice, if you take the roots and stolons, and also Rolfia. So all these are going to be underground. If they are not being processed well, there may be chance that it may leads to the it may have a contamination with the microorganisms that may lead to this toxic effect. So other one is adulteration. So adulteration, sometimes even if you see nowadays that many of the herbal formulations, they are going to add certain the conventional medicines to it. So most of the formulations where they show that this is an antibiotic drug. Now we have seen
Dr. Prachi, please mute your mic. A small request to everyone, please mute your mics so that it will not create any disturbances during the presentations.
Thank you, Bhagavi. So let's continue with this, that adulteration also, sometimes adulteration also leads to the adulteration of herbal drugs with the conventional drugs also is one of the major factor that leading to the toxicity, where the some of the Ayurvedic manufacturers are mixing the conventional drugs like diabetic, like glibenclamide, adding to their formulation. With that, what may happen is they may have a synergistic effect. So the synergistic effect leads to the hypoglycemic. So that is one of the extrinsic factor. Other factor is authentication. The most important, I think, in any herbal formulation, the pharmacognosics play a major role in the first parts itself, that is authentication. If you do the authentication very well, because many plants within the species, they look similar. Within the species, within the genus itself, they look similar. And even if you take, here I have taken two plants which are even genus, the genus is totally different, but if you see the leaves of the drugs, they are very much similar. And if you are going to not take the drug which we want to, that also leads to authentication errors and leads to some toxicity. For example, Plantago major, if it is being replaced with the Digitalis lanata, if you see the leaves now, they are almost same, shape of the leaves. So this has been taken for the laxative effect, and this is as we all know, it is have a cardiotonic action. Now, if you want a laxative and you are taking to this cardiotonic drug, it leads to the cardiotoxicity. So authentication is also one of the extrinsic factor which leads to the adverse effect of the herbal medicine. So these are the some of the extrinsic and intrinsic factors that leads to toxicity of the drugs. So we have to be very careful that while you are selecting any plant, you have to go for the literature survey, where you have to check whether there is a toxicity that is being present over this herbal drug. Then only you have to go for the proper authentication, then processing, then formulation, then that will leads to a safe drug. Otherwise, there are many in literature survey, we can see many cases where that leads to the adverse effect. Now, most important and very widely known toxic effect that is been we all know that it is herbal drug interaction. So what may happening is we are taking this while we are going for a physician, if you are not saying that you are also on the herbal medication, then that leads to a major error. So I suggest anyone who is going for the conventional treatments, even if you are on an Ayurvedic treatment, you let your both the physicians know about each other. Otherwise, what happens is this leads to these major interactions. Here, if you see, for example, I'm not going to go with all the examples. I will take one of the example, and that is the well-known one is the garlic one, lysan. Now, while you are taking lysan, garlic as an herbal formulation, at the same time, if you are on a blood thinning medications like aspirin. It will increase, it is going to produce a synergistic action and it will increase the bleeding time. And most of the other herbal drug interactions are being present with the what pharmacokinetic interactions, where the cytochrome 50 P450 enzymes may be altered, so the drug may be have potentiating effect, or it may not have any effect at all, So other example is licorice, mulethi. If you are taking with any other laxative drug, it will decreases the potassium level. It may decreases the potassium level. So these are the certain factors that may also have this toxicity effect on the human beings. Now, what we have to do? Okay, we have learned about the toxicity related issues with the herbs. So what we have to do for inhibiting, or we have to do, what are the factors that, or what are the steps we have to do to regulate it? So this is one of the conference that was held, and I have taken this figure from that paper itself, that they have very much in precisely they have told us how to measure the quality and safety of the herbal drugs. That conference is Global Coalition of Regulatory Science Research Conference that was happened in 2018. So they have given certain parameters where we can go with this, where we can minimize all these adverse effects. So in this, first one is we have to go for the conventional toxicity studies, monographs, well-established monographs, and also conventional analytics. These are the three major things that we have to check for the quality of the herbal drug. In this, I want to highlight with this, as Divya, the previous presentation, what she was saying in the in silico studies. So if I take that example, now antifungal studies, what we used to do is in vivo studies. Now if you go with the in vitro like simulation studies, what she has done is some of the parties simulation studies. Now from the simulation studies, we will get to know that whether the molecule is going to be active or not, how much it is going to be active. So those parameters we can already know, and that can be that can be placed in this monograph, which is going to be useful for the others to refer and go with the safety issue parameters. In this, one of them is advanced analytical methods like conventional analytics and advanced analytics. In conventional analytics, what we used to do is we used to do this HPLC studies. In the advanced analytics, we can go with the HTS, like high throughput screening methods, where it is going to allow us to automatically test number of chemical or biological compounds in a more proportions as compared to this conventional analytics. And also this DNA barcoding, this will be this is going to be it will avoid the fraud in like dietary and also in the herbal supplements. As we know, if one barcoding is being placed to this one plant, it is from the source to the finished products, it will help us to identify the plant at any stage of its formulations. Next, blockchain technologies. It is also blockchain. It is also helpful us to monitor the herbal medicines from its source to the destinations. Next, in silico methods, the previous study which we have seen, that in silico methods, in silico methods, like we are going to have this simulation studies, combination of a simulation studies plus conventional toxicological methodologies, which will also helpful in determining the quality and the safety of this herbal drugs. So with this presentation, I want to make awareness among all the pharmacologists and also the pharmacologists that not all the herbs drugs are going to be safe. We have to do the literature survey, and we have to establish good data, and which we have to preserve that literature survey by which we can reduce the adverse effect, and also we can promote a very safety herbal medicines in the market. Okay, this is my conclusion. Thank you so much. I would like to thank all the organizers and also College of Pharmacy, School of Pharmacy, Vishwakarma University for giving me this opportunity. Thank you.
I can say it's a need for the time, because the herbal drugs is in demand now. Everybody is using it, but as the myth, which is herbal drug is the safest drug, we can take it, so which is not practically correct. So there is a need, and I think this topic you have taken very nicely, and you have taken each and every aspects. Very good presentation.
Thank you so much, ma'am. Thank you so much. It means a lot. Am I audible? Hello?
Yes,
Yeah.
ma'am. Yes, ma'am.
Yeah,
thank you for your presentation, ma'am. Now we are moving with the next presentation by Ashwini Makta Gavit from D.Y. Patil College of Pharmacy, and her topic is on preclinical evaluation of anti-cholinesterase and antioxidant activity of an Ayurvedic formulation, possible efficacy in Alzheimer's disease. Dr. Ashwini Makta, I hope you are there online, Dr. Ashwini. Okay, ma'am. We are moving with the next presentation. Mrs. Sakina Yusuf Punjab from Ravindra Institute of Pharmacy, and her presentation topic is on formulation and development of anti-hyperpigmentation herbal gel containing acacia couture extract. Mrs. Sakina Yusuf?
Yes, madam.
Yes, ma'am. You can start your presentation, ma'am.
Yes, give me a minute.
Yes.
Is my screen visible to everyone?
Yes.
Hello?
Hello.
Hello? Am I audible?
Yeah, you are audible.
Yes.
Is my screen visible to everyone?
Yeah.
Yes, ma'am.
Shall I start?
Yes.
Please.
No problem.
Yes, ma'am. You can start with your presentation.
Good afternoon, everyone. Myself, Sakina Punjab. As you can see, I'm a research scholar at LIPS Research Center. I'm doing my PhD from European International University, Paris. So my presentation today, I would like to start with an introduction brief about formulation and development of anti-hyperpigmentation herbal gel containing acacia catechu extract. So hyperpigmentation is nothing but a mismatched or discolored patch on the skin. So as a definition, hyperpigmentation of the skin refers to as a dermatological condition which the color of skin making it discolored or darkened. In other words, or in a medical term, it is used to describe as a dark patches of skin from the excess melanin production. As you can see, this one picture briefly summarized how does the pigmentation or hyperpigmentation looks like on your face. So as you can see on this head, I don't know whether you will be able to see my cursor, but on the forehead, these dark patches of hyperpigmentation is caused melasma, which is caused by the sun exposure. Over here, the small dots of pigmentation is called as fractals, which is also caused by sun damage. And this is over here at the corner of the lip. You can see post-inflammatory hyperpigmentation, which is dark color towards the corner of the lip, which is caused by acne, rashes, or inflammation. So these are the types of hyperpigmentation, which over here you can see. Now I would like to tell you something about melanin. What is melanin and how does it cause hyperpigmentation? So melanin is pigment which is formulated, or you can say generated, in the skin cell. When UV radiation is caused on the skin, it activates the reactive oxygenating species, ROS, which activates mutagen activated protein kinase. Now this process gives parts to amino acid L-tyrosine, which is already present in the skin cell. So tyrosinase enzyme causes a reaction with amino acid L-tyrosine, causing it to form a L-dopaquinone, which is also known as L-dopa. This L-dopa again react with the tyrosinase enzyme and form dopachrome or dopaquinone. This dopaquinone again react with the tyrosinase enzyme and forms melanin. So this is what which has been described in this picture. If you all can see, this is melanin synthesis pathway in which L-tyrosine forms L-dopa, L-dopa forms dopaquinone, and dopaquinone forms melanin in the skin, which gives color to your skin, or I would say darker color to your skin. If you all are following, I would like to tell you something about statistics. This is my review which I have done. This statistics have been done from Statista Research Department, and it shows that out of 100% patients, 35% patients are of hyperpigmentation. This is done via walk-in survey in a dermatological setups in all over the country.
Ma'am, sorry for the interruption.
Yes.
We are unable to see your slides, ma'am. We are seeing only one slide.
Okay.
The first slide we are seeing ma'am. 3 types of hyperpigmentation. That slide we are seeing.
Okay, just a minute.
Yes.
I'll again share my screen. Is it visible now?
Sorry, ma'am. The first slide itself we are seeing. You can share your screen for one more time. Or else you can proceed with your presentation, ma'am.
Yes, I'm trying. Can you see the second slide now?
Yes, ma'am. Now we can see introduction. Yes, ma'am.
Okay. Shall I start again or should I continue?
Oh, ma'am, you can continue from the past.
Okay. So this is the 3 types of hyperpigmentation which I explained if you couldn't see in the previous presentation. So these are the 3 types of hyperpigmentation which I was talking about. First, you can see on the forehead, then at around the cheeks, and then at the corner of your lips, of this picture. Now the thing is, these pigmentations or these hyperpigmentation is caused by melanin or extra production of melanin. I would say this melanin accumulates at one place and which gives a darker color than the rest of the skin. So the melanin synthesis pathway is somewhat like this, whereas in melanocytes, L-tyrosine, which is an amino acid which reacts, tyrosinase enzymes reacts with L-tyrosine and form L-DOPA. Then L-DOPA again react with this tyrosinase enzyme to form dopaquinone. This dopaquinone again react with the tyrosinase enzyme to form melanin. So this is melanin synthesis pathway, or you can call it as pathophysiology of melanin synthesis.
હા યાશના જોયો તારો મેસેજ હું તમને કહેવાની પણ હતી કે તમે લોકો આમાં જજો.
Sorry.
Ma'am,
એટલે
could you
આપણે આનું જ આપણે આનું
Miss Ma'am, I request all the participants to mute their mics. Yes, ma'am. Dr. Sakina, ma'am, you can go with your presentation.
Okay. So this is the stats or literature whereby I got to know that 35% of the walk-in patients in dermatological setup, they come with the complaint of hyperpigmentation. Now, hyperpigmentation, for the treatment of hyperpigmentation, I would first like to tell you about the synthetic approach, which will briefly give you idea of my objectives or primary goals, which for which I conducted this study. So first line of therapy and second line of therapy, this is a synthetic approach towards the treatment of hyperpigmentation. In first line of therapy or in first line of treatment, topicals with triple combination therapy, including hydroquinone, topical retinoids, or topical steroids are given. Now they are given in the triple combination. Then in second line of therapy, chemical peels in combination with topicals like salicylic acid peels may be given with concomitant acne. So this is the first line and second line of therapy. Again, topicals such as zinc sulfate, arbutin, azelaic acid, kozic acid, and some cosmeceuticals can be added as a second topical, but can be tried as a monotherapy also. So this is about topical therapy. Now there is chemical peels, and then there is laser therapy, which also reduces the appearance of hyperpigmentation. And Q-switched NDYAG is also a type of laser treatment, which is conducted to reduce the appearance of hyperpigmentation on the skin. Now, as you can see, this is a synthetic approach, so it is bound to have some sort of side effects. As you can see in this pictures, chemical peels or retinoids or steroids can give you these types of skin irritation, erythema, dryness, swelling, itching, peeling, all these types of side effects, or you can say unfortunate experience of the hyperpigmentation treatment. All this complete treatment, laser, chemical peels, and steroids, and all of this can be very expensive too. Hence, novel discoveries of plant-based or herbal formulation with lesser side effect is need of the time. Now, while I have established my ground for the study, I would like to tell you this was the reason that this study is conducted. Formulation and development of anti-hyperpigmentation gel containing acacia catechu extract. This is a herbal and natural answer to the hyperpigmentation. I would like to tell you the formulation of this preparation. Acacia catechu extract, TEA, that is triethanolamine, carbopol, glycerin, and distilled water. These all are the formulation. Can you all see my screen?
Yes, yes.
It is moving, right?
Right.
Okay. So this is the formulation of the gel. Now I would like to tell you something about acacia catechu. What is acacia catechu and why I have chosen this as an active cosmeceutical agent? All my pharmacist friend must be familiar with API, that is active pharmaceutical agent. I belong to cosmetics field, so I would like to call it as active cosmeceutical agent. So my ACI is catechu extract. Catechu is very well known as katha, right? So this is used in various food additives or astringent or tannin or as a dye also. This is extracted mainly from Senegalia catechu. This active cosmeceutical agent contains chief constituent like catechin, catechinic acid. The wood contains epicatechin, adzelchin, dicatechin, galochin, gossiptin, phlobetanine, camphorol, and quercetin. Now, a hero of my study, quercetin is biologically highly active. Ayurveda literature says this bark is antihelminthic, antipyretic, anti-inflammatory, and anti-leprotic. This is
sorry to interrupt, ma'am. Please stick on to the time, ma'am. You're exceeding your time limit.
Yes.
Please stick on to the time and complete your presentation before the time.
Yes, I'll hurry up. Yeah, give me a few minutes and I'll wrap it up.
Yeah,
This
thank you.
in this study.
Go to your methodology and results.
Sorry, ma'am.
Please go to your methodology and results.
Yes. So this study is ethanolic bark extract of Acacia catechu were examined for its presence of quercetin content. Now, quercetin content was 0.070% determined by HPLC method. Now, this is not my study. I have taken this data from estimation of quercetin in Acacia catechu ethanolic bark extract by HPLC method. So keeping this in my mind, I have conducted efficacy testing of my catechu extract for tyrosinase inhibition activity of quercetin. This analysis of final product in which I have conducted stability testing, pH, centrifugal test, spreadability, absorbance, and microbial testing for my gel preparation. These are the results. And now the important or a key factor of this study, that is percentage of tyrosinase inhibition at concentration of 120 microgram per milliliter. So this is the study which shows on an average of 86.19% catechu extract was able to inhibit the tyrosinase enzyme, which will reduce the production of melanin, as compared to kojic acid, which gives 98.73% on an average. So the summary of this study is that pigmentation is on the skin is due to compound called melanin. In excess, melanin cause hyperpigmentation, and hyperpigmentation can be treated through herbal extracts or through herbal preparation formulation by using Acacia catechu as a star ingredient. So Acacia catechu extract is rich in polyphenol called quercetin, which act as a substrate in tyrosinase inhibition. That's all. If you have any questions.
So what is your formulation?
My formulation is a gel which contains Acacia catechu as an active constituent.
Just a simple gel?
Yes, just a simple gel.
So what is the for gelling agent, what you are using?
Carbopol. It's a polymer, synthetic polymer.
Okay. And this formulation of the gel, and you are finding out the activity, whether it is able to do the hyperpigmentation
I am
or not,
finding
right?
out the activity of Acacia catechu extract, which is going to inhibit the tyrosinase enzyme, which helps in melanin production.
But this is already proved, right?
No, I have proven it. Quercetin, amount of quercetin is already proved. You want me to
but
share
the effect of quercetin on pigmentation also, right?
Yes, the effect
No need to share.
Yes, the effect of quercetin
So,
is.
but in the case, but you are also demanding that the quercetin which is present in your acacia is only responsible for it, right?
Yes, ma'am. That's my study.
Then what is your novelty?
Content Acacia catechu extract.
That is already proven, right?
No study has been done to propagate that Acacia catechu extract gives fairness or which helps in treatment of hyperpigmentation. There is no study which proves that.
Is it so?
Yes, ma'am. Acacia catechu has not been proven as an fairness agent or anti-hyperpigmentive agent.
But I think no.
Okay. I will check again.
Yeah, but you are talking so much about this thing, but I was very much excited to hear about your formulation. You are bringing some novelty there. I was looking for it.
Formulation,
As you said, that quercetin is present in Acacia catechu, but it
Yes.
is not the major constituents, right?
It is not a major constituent, no, ma'am.
Yeah, so the amount also will be very less. So
The
how to
amount
get
is only 0.070% weight by
Yeah.
weight.
So whether you are applying some novelty there so that it will increase the amount of quercetin or something else also is responsible in
I'll
acacia.
keep it as a base for my study, definitely, ma'am.
Yeah, so that is what I was looking for, but I could not find anything exciting in your formulation. And as you said, your PhD work. So I think
But this is
something
not my
novelty you
PhD
have to
work.
bring
No,
there.
ma'am, this is not my PhD work.
This is not your PhD work?
No, no, ma'am, this is not my PhD work.
Okay. In the beginning, you have mentioned that, right?
I said I'm a student, a research scholar.
Okay. So this is just a work, not your PhD work,
This
right?
is just an attempt on towards the research in whitening.
Okay. Okay. Okay, fine. But I think you should work on the formulation.
Yes, ma'am.
Which will be more exciting.
Definitely.
Yes, thank you, doctor. Thank you for your presentation. Ma'am, shall we move with other presentation now?
Excuse me,
please,
Bhargavi. I have a
Yes,
small
ma'am.
doubt. I am a listener. I am a PhD scholar too. So I have a small doubt with Dr. Rokanya. Can I ask now?
Yes, ma'am.
Actually, if we find there are many traditional medicines that is being well treated with by Siddha doctors for cancer and other main diseases. So, if you are taking some of the medicines, is it mandatory to know what is the real bioactive component, or we can take as a churnam or the crude extract?
See, when you are taking the churnam or crude extract, you are blindly telling someone that this will be helping you to do this. But when you are finding out the bioactive constituents, you are very much confident. This is the thing which is responsible for it. And also at the same time, you will be knowing what is the safety parameters, what are the other things. So until unless you know what is the component is responsible, so you cannot go to the safety parameters and all other things.
Okay. For that, we can use GCMS, right?
Yeah, any kind of estimation. So if it is a volatile oil constituents, you can go for GCMS. If it is a normal, you can go for LCMS. Other than that, you have many methods to estimate how much amount it is present.
Okay, ma'am. Because I found many literatures that are suggesting many good medicines, Siddha medicines with very good activities, but there is no proof it. So I thought like I can take it, but taking a bioactive component is very difficult from the crude extract, right? So I thought like whether I can give it a try. So I asked you, ma'am.
Yeah. It is a challenging, it is a very challenging, but I think through the advanced technology, whatever is available till now it is becoming much easier compared to the previous days. So I think the traditional system of medicines, which are actually very much beneficial for our health. So if you are making it as per WHO standards, if you are able to make it as a safe, and if you are able to tell that, yes, there is an evidence that it is safe. So I think it will be better move.
Okay, ma'am. Thank you so much for it. Thank you.
Yeah, please call the next presenter.
Yes, ma'am. Thank you, ma'am. So now I would like to invite Dr. Lokesh Patil from Institute of Pharmaceutical Sciences and Research, Sardar Patil University, India. And his topic is on design and development of floating drug delivery system for 5-HT3. So Dr. Lokesh, you can
Hello.
start your presentation.
Yes, ma'am.
Dr. Lokesh, you can start sharing your screen and you can go with your presentation. I hope Dr. Lokesh was there online. Sir, could you please confirm us with your presence?
Yes, ma'am.
Yes, sir. You can start
Yes.
sharing your screen now. Seems to be Dr. Lokesh is having some network issues. We'll be going to the other presentation, going back to Dr. Ashwini Maktha. I hope Dr. Ashwini was there here online.
Yes, ma'am, I'm here.
Yes, ma'am. You can go with your presentation, ma'am. And Dr. Ashwini presentation is on preclinical evaluation of anticholinesterase and antioxidant activity of an Ayurvedic formulation, possible efficacy in Alzheimer's disease.
Is my screen visible, ma'am?
Yes, ma'am. Your screen is visible. Could you please be a bit louder?
Yes, ma'am.
Yes, ma'am. You are audible now.
Good afternoon, all of you. First of all, thanks for giving me the chance to present my poster here. I'm Ashwini Gavit. Uh, I'm I'm completing my M Pharmacy in Pharmacology from Dr. D.Y. Patil College of Pharmacy, Akudi, Pune. Today, I'm going to discuss interesting topic entitled preclinical evaluation of anticholinesterase and antioxidant activity of an Ayurvedic formulation and possible efficacy in Alzheimer's disease. Uh, as we know, Alzheimer's disease is the most common neurodegenerative disease with approximately 44 million people worldwide. More of our old people from our houses are living with Alzheimer's disease as it is age-associated neurodegenerative disease. Various pathways like extracellular amyloid plaques, microtubule tau protein aggregation, oxidative stress, cholinergic hypothesis, neuroinflammation, and metal hypothesis are included in the pathogenesis of Alzheimer's disease. And current pharmacotherapy like donepezil, rivastigmine, galantamine gives only symptomatic relief. Now, global therapeutic research is shifting towards the herbal medicine or Ayurvedic medicines. People presume that Ayurveda is safer than the synthetic, and Ayurveda manages Alzheimer's disease as preventing, promotive, and curative levels and improves quality of life of individual. It is essential to conduct study which evidences the potency of Ayurvedic formulation. Here we use the marketed Ayurvedic formulation named Memory Medha syrup, which contains various herbs like Shankushpi, Gulab, Elaichi, uh, black pepper, Phonicum vulgare, Vitis vinifera, Centella asiatica, and Crocus sativus, which are individually known for their neuroprotective action, antioxidant action. In present study, 30 male Vista rats weighing between 180 to 220 grams were used, and this is the ISA approval number. An experimental design was 30 male Vista rats grouped into 5 groups. First one group was the normal control, which was treated with the distilled water, and second one was the induction control, which we had treated with the AlCl3, that is aluminum chloride. Aluminum chloride is the induction inducing chemical here. Dose was the 100 mg per kg orally for the 42 days. Third one group was the standard control group where we have treated with AlCl3 100 mg per kg, and after 1 hour of treating with AlCl3 we have given donepezil as a standard drug 1 mg per kg orally. Then fourth one group was the low concentration syrup group where we treated ALCL3, and after 1 hour of the ALCL2 treatment, Ayurvedic formulation with the dose 4 ml per kg given orally. Then fifth group was the high concentration syrup treatment group, in which we have treated with the Ayurvedic formulation 8 ml per kg after treating with the LCL3 orally. After treatment of 42 days of LCL3 and the treatment of Ayurvedic serum, behavioral study were performed like locomotor activity and Morris water maze test. In locomotor activity, uh, each animal placed in the digital actophotometer for 5 minutes. The apparatus was consisted of photoelectric photoelectric cells. When the beam of light falling on the photo cells is cut off by the animal was recorded as one count, and it was counted for 5 minutes, and it is related to anxiety, and as anxiety is risk factor of Alzheimer's disease, as increased symptoms of anxiety is related to the higher levels of beta amyloid plaques, and also stress can affect the brain immune system, which is lead to the AD symptoms. And next one was the Morris Water maze test, which was performed for the special memory task of the animals. It is performed as per the norms, and a large circular tank was used, which was with 150 centimeter in diameter, consisting of 4 quadrants: N, W, N, E, S, E. Hello, am I audible?
Yeah, please continue.
Uh, the last circular tank was consisting of 4 quadrants, that is northern west, northern east, and southern west. And it was filled with the water with temperature 25 plus minus 1 degree Celsius. Then submerged platform of 10 by 10 centimeter was kept 1 centimeter above the surface of water for acquisition phase, that is training phase for the animals. And animal were placed into the tank facing toward the wall of the tank and allowed to locate the submerged platform and guided to locate the platform for 120 seconds. And this training was given for 4 days, on 17th, 18th, 19th, and 20th day. And after that training period, retention phase was carried out. The water was made opaque by using milk powder, and the submerged platform was kept 1 centimeter below the water level, and animals were placed in the tank and allowed to locate the submerged platform. And like this way, the retention of memory of animals were tested, and escape latency of animal were tested in Morris water maze test. Then supportive studies like acetylcholine esterase level was carried out. It was carried out by Illman's method. Here, after performing behavioral study, brain of the animal was excised and homogenized. Then supernatant was used for further testing. Here, 0.4 ml of supernatant mixed with 2.6 ml of phosphate buffer and 100 microliter of DTNB. And the absorbance was checked at 412 nanometer using UV spectrophotometer. And then add 20 microliter of substrate, that is acetylthiocholine, and change in color was observed, and it was recorded at absorbance was recorded at 4,1200 nanometer. Then antioxidant status was checked using supernatant. Here catalase and GSH was performed. Catalase activity was performed by Luck method, and GSH activity performed by Moron method. Moving forward to the results, after treatment with the Ayurvedic formulation, locomotor activity was found to be increased as compared to the aluminum chloride treated group on 21 day and 42 day, we can see here number of counts was increased as compared to the ALCL3. Then Morris water mesh test. Here are the three bars. So like 20 was the training day, 21 and 22 was the retention phase test. Here escape latency was increased, decreased means time to locate the submerged platform was decreased in treatment group, and it was as like the Donepezil. We can compare it with the Donepezil. They are significant improved in escape latency. Then acetylcholinesterase level was decreased in Ayurvedic formulation treated group, as acetylcholinesterase is the enzyme mainly involved in pathogenesis, which clears acetylcholine into choline and acetic acid and decrease the level of acetylcholine. Here, our formulation decreased the acetylcholinesterase activity, which was increased in ELCL3 treatment group. Then, in antioxidant parameter, catalase activity and GSH activity was increased as compared to the acetylcholine group. Here, the histopathology. These are the 5 groups: A, B, C, D, E. A1 was the normal control. B1 was the induction control. Here we can see the beta amyloid plaques, and here we can see the neurodegeneration of neurodegeneration. In C standard control, there was less neurodegeneration and beta amyloid plaques. And in treatment of low syrup formulation, there was less neurodegeneration and hemorrhages found. And also in the high syrup concentration treatment, there was decrease in the neurodegeneration and beta amyloid plaques. Moving to the discussion and conclusion, aluminum chloride is a cholinetoxic which causes apoptotic neuronal loss, that is neurodegeneration. Also activates the ACHE enzyme and spatial and recognition memory deteriorated. Our Ayurvedic formulation showed the neuroprotective effect by improving the behavioral parameters and inhibition of ACHE activity, and also reduced the oxidative stress. Hence, the Ayurvedic formulation may provide a safe, economic, and therapeutic alternative in the management of Alzheimer's disease. Here are the references I have used for this study. Thank you.
This work is done by you?
Yes, ma'am.
Okay. Ajay from project?
Yes, ma'am.
Okay. So you have done anti-Alzheimer's this thing model, right?
Yes, ma'am.
Other than this model, any other models are available?
No, ma'am, but other studies like in behavioral study, other 2 methods were performed, like elevated plus maze and Cook's pole climbing apparatus test. And in supportive study, and in antioxidant status, LPO and SOD were performed. And also in histology, I have performed in 2 various sections, like in hippocampus and cortex. These are the main parts of brain which are involved in Alzheimer's disease. And in 2 different strains I have used, like we are using H&E as a normal histopathology dye, and I have also studied in it Congo red dye. But due to lack of space and lack of time, I have not included here, and publication is undergoing. That's why I'm not revealing the data as well.
Okay, fine, good.
Thank you, ma'am.
Yeah.
Can I stop sharing, ma'am?
Yes, ma'am, you can stop sharing your screen.
Thank you, ma'am.
Now we are going with the other presentations. So for this day, we are pending with the 3 presentations, ma'am. These are the last 3 presentations. Now I would like to invite Dr. Lokesh Patil from Institute of Pharmaceutical Sciences and Research Center, Sardar Patel University, India. Dr. Lokesh. Dr. Lokesh. Okay, ma'am. We are moving back to the other presentation by Dr. Dr. Desai Priya from Tatya Pharmacy College. Dr. Desai Priya.
Yes, ma'am. Yes, ma'am. Yes, ma'am. I will
Yes,
start my
ma'am. You
screen
can
share.
go with your presentation, ma'am.
Okay. Good afternoon to all, ma'am. Present over here. Can I audible? Hello.
Yes, yes, yes.
And can you see my presentation whole poster? Hello.
Yes, ma'am. You are audible and your screen is also visible. You can start.
Okay. So first of all, good afternoon to all present over here. And I personally thank you Startup to share my research area over here. So today I'm going to be present a poster on the competitive evaluation of anti-cancer effect of Panchgavya and Panchgavya by Lewis lung carcinoma cell line. That is a pre-study before we are going to be in vivo study. So personally, I Priya Desai, Assistant Professor of Satya Pharmacy College and student of PhD of Ukata University. I will start with introduction and background. What is a malignant tumor? As we already know, according to my oral presentation, the malignant tumor is a cancerous, uncontrollable growth that is called as a cancer. And the lung carcinoma is a malignant lung tumor with various histological variants that is arise from the different cell types such as bronchial, epithelial, alveolar, and bronchial glands. So in this study, the mainly two type of lung cancer cell are observed. That is non-cell lung carcinoma and small cell lung carcinoma and non-small cell lung carcinoma. The non-small cell lung carcinoma is 85% of total. In this study, I will be induced the model in which the Lewis lung carcinoma, which is established from the cancerous lung of the C57BL mouse. This cell line is collected from the C57BL mouse. In this, I will treat this cell line to check the effectiveness of my medication, that is traditional medication of cowpathy. You can see that nowadays whole world is shift towards the Ayurvedic, or we can say herb patterns, in which the Panchgavya and Panchgavya Grita is a new novel drug because there is a no side effect, as well as this undercomes to Ayurvedic part, as well as Panchgavya and Panchgavya Grita is not yet reported for any lung cancer treatment option. So I choose this Panchgavya and Panchgavya Grita for my pre-study. After that study, I was go with the in vivo study that is already presented on over the oral presentation. So the Panchagavya and Panchagavya grit is nothing but the mixture of 5 cow products, that is milk, dung, curd, urine, and ghee. Milk contains lactoferritin, which is proven as individually as anti-cancer activity. The dung and the curd have a disinfectant activity, whereas urine have that particular uric acid that is used for the immune booster agent. Now, curd contains conjugate linolenic acid, which is a proven anti-cancer effect, and in ghee there is a CLA, conjugated linolenic acid. So this 5 component together are used for the anti-cancer, provide anti-cancer medication or anti-cancer effect. Now, this individual component, for example, milk, dung, ghee, are reported for the immune booster as a treatment of breast and prostate cancer, already reported, but there is no single data or no single study about a comparative or not an individual Panchagavya and Panchagavya Grita is reported for the lung cancer. So that is a new era for the research for my perspective. Now, for cell line study, I will go with the first cell viability test. Now, that is a material method. In cell viability test, prepare a stock solution of kaolin AM dye, which is a viable dye. So we can see the live cell of a particular cell growth. Now, in DMSO, dimethyl sulfoxide, and prepare PI dye, that is called as propodium iodine dye, This is particular for the dead cell in sterile water. For example, 10 milligram a particular dye in ml. Now, adherent cell may be cultured on sterile glass cover slip as either conflict and subconflict monolayer. It means a layer will be single on the slides. Now, the cell may be cultured inside the 35 mm disposable petri dish or other suitable container. In non-adhesive cell may also be used, because if you are going to be adhesive cell, you can show that all the lump are present over that. But there is a non-adherent cell and non-adhesive. That means a single layer will be easily formed and excessive cell growth will be easily removed. And the remove media from the cell in plate. Now, wash twice with PBS buffer solution. Now, this PBS, phosphate buffer saline, is particularly used for the preventing aggregation of the cell. Now, fresh add buffer and add kaolin AM dye. So, ubiquitin intracellular esterification is activated. Now, incubate 37 Celsius in dark and for 15 minutes. So maximum cell will be strained and give a result. After that, add PI dye for 10 microgram per ml. So the dead cell will be strained. Incubate in dark at 30 Celsius for 5 minutes. So we are providing the time for the reaction for calcium dye as well as PI dye. Now remove dye containing buffer and add fresh buffers. So you can see the easily see the all growth available in that. Observe under the microscope FITC, which is 400 to 450 nano lambda. So easily you can see the all the viable as well as non-viable cell. Now live cell of stained with the calcium dye, and that cell will be stained with the PI dye. Now, if you want to count that cell, count the number of the cell contained PI dye, which is in orangish yellowish color. So you can easily check the viability of cell. And the formula for checking this viability is equal to calcium in positive cell divided by the total number of cell, which we can take it in 10 raised to 5 or 10 raised to 6 into 100. That is all about the cell viability test. Now we are moved after that cell viability test. We are going to be perform the MTT assay. Now this in this MTT assay, we have to take 1 into 10 raised to 6 cells per plate in microliter of media or well. So the column is 2 to 11 cells are present. Now add the 150 microliter growth media. So easily they can grow from 1 to 12 total column of the plate of our experiment. Now in only we are going to be plate 2 to 11 column of for selling growth and 1 to 12 for growth media. If there is any contamination present in our study, they can show in 1 and 12 number of the column. So we can identify the we can say debris, or we can say extra colony group from the 1 and 12 column. The plate are incubated at 37 Celsius in CO2 incubator for 24 hours. So exponential freeze. So maximum growth in particular time with the manner will be shown. Now prepare sterile dilution of the cytotoxic drug in growing media of the either giving a different concentration, highest to lowest kill the most of the cell and lowest to kill of none of cell. So Panchagava and Panchagava will be prepared in different concentration and put in the different row. As per the we can say start with the 2 column to 11 to highest to lowest or lowest to highest as per our need. The media needs to remove from the well from 2 to 11 and feed fresh growth medium contain 5% fetal column serum. That means they have cytotoxic column 3 to 10. So second column is only for normal growth. Now incubate at 37 Celsius for CO2 chamber, incubator chamber for different exposure phase. So maximum reaction will be take in this particular phase. So killing of all microbes that is need for the according to drug concentration will be stored in this period of time. At the end of the drug exposure period, the medium will be removed from the vial and cell is fed with the 100 microliter of the fresh medium containing 0.5 mtt solution. Now wrap the plate in aluminum foil so there is no other reaction will be possible because of the aluminum foil and a covering of aluminum foil, and incubate for 3 hours at 37 Celsius and media remove MTT contain. Remaining MTT frozen crystal dissolved into 100 microliter DMS solution. That means remaining concentration of dye will be dissolved in that, and we can see clear solution of particular things and particular cell. So we can observe the kill or the amount of drug required for the inhibition of 50% of the whole growth of colonies, and recorded at absorbance 570 nanometer. Now we can move towards the preparation of Panchagavya and Panchagavya Grit. As I earlier told you, the Panchagavya contain and Panchagavya Grit contain 5 cow products, that is ghee, curd, milk, dung, and urine. In Panchagavya Grit, we have to take equal amount of each and every component. In which, in Panchagavya Grit preparation, we have to mix well urine and dung. After that, filter 8 times in cotton cloth, and after that we have to add even curd and ghee in particular are filtrate. Sorry,
this part you can leave because already explained,
yes,
right?
Yes. Thank you, ma'am. Now a result, that is a cell line study. In x-axis I have a concentration milligram per ml, where y-axis I have a percentage inhibition. So you can see my different dilution concentration on x-axis, and y-axis there is a percentage inhibition. Now as you see, if the concentration will be increased, the inhibition will be also increased, same as Panchagavya and same as for Panchagavya Grit. But in Panchagavya there is a higher amount of concentration will be sourced. In minimum dose there is a higher amount of inhibition will be sourced. So my discussion from this result is I say 50 value, that is effective concentration 50. That means a need of drug to kill the 50% of total growth for Panchagavya is 7.75 milligram per ml, whereas Panchagavya Grit is 6.78 milligram per ml, whereas standard drug doxorubicin IC50 is 1.087 into 10 raised to minus 10 milligram per ml. But that doxorubicin has some side effect, whereas Panchagavya and Panchagavya Grit has no side effect. So we can prefer as an Ayurvedic treatment as an anti-cancer agent of Panchagavya and Panchagavya Grit. So my discussion is, from the result, I will conclude that Panchagavya and Panchagavya Grit is effective in LLC cell line lung carcinoma. Thank you, ma'am.
Okay, good.
Okay, ma'am. Thank you, ma'am, for sharing your best experience with me.
You can stop sharing so that the next presenter can present.
Okay, thank you, ma'am.
Yeah, okay. Thank you for your presentation, ma'am. Now, I would like to invite Pranita C. Vani from MGV's Samajastri Prashanta Hire, College of Pharmacy, India, and her topic is on sleep-inducing cream. Dr. Pranita, ma'am, you can start your presentation.
Yes, ma'am. I'm audible, ma'am?
Yes, ma'am, you are audible. You can start sharing your screen.
Okay, 1 second, 1 minute. Screen is visible, ma'am?
Yes, ma'am. Your screen is visible. You can make the zoom.
Yes, ma'am. Thank you.
Yes, ma'am. Now it is clear. You can start your presentation.
Okay. Hello. Good afternoon, ma'am. My name is Pranita Chandrakantwani. I am a final year B.Pharm student from MGV's SPH College of Pharmacy, Malegaon, Maharashtra. So my title of paper or my topic is a sleep-inducing cream. Actually, we prepare a cream for the insomnia patients as well as normal people also. Insomnia means insomnia. It is a sleep disorders in which people's having problems to falling and staying asleep. And nowadays it is very common because the causes of causes of that it is a stress, mental health issues, lifestyle, busy schedules, then medications, poor sleeping habits, etc., and many more. So, for this reason or for this problem, we are preparing a cream, we are preparing a solution, and the solution or our product is a sleep inducing cream. The benefits of our cream is it gives you a peaceful sleep, it gives you a calming effect, it gives you a cooling effect, release your stress, tension, and pain relaxation also. It is also anti-anxiety. So in that we are using various types of materials. We are our main ingredient of in that our cream. The main ingredient are the lavender oil, chamomile oil. These two main ingredients are the natural sedative agents. They produce a sedation by externally. And after that, we are using a cream base like beeswax, borax, water, liquid paraffin, and etc. Lavender oil, the biological source of lavender oil, it is a Lavandula angustifolia, flower of the Lavandula angustifolia. And the biological source of chamomile oil, it is Matricolia chamomilla, flower of this plant. So these main ingredients we are used in our sleep inducing cream. Actually, how our product works? Our product works on the basis of the aromatherapy. Aromatherapy means in that using of the aromatic compounds or the using of compounds which having an aroma, which improves your psychological as well as the physical health. So for this, we are using these two, lavender oil and chamomile oil, because these two are having a pleasant aroma, and their aroma produce a sedation. They produce a sedation by the way of it acts on the parasympathetic nervous system, the central nervous system also. So it's increased the aroma of lavender oil. It's present in that there are two ingredients are present. First one is the linalool and the second one is the linalool acetate. Actually, these two are the terpenoids, and this mainly having effect on the sedation purpose or having a calming effect properties. So when the aroma of lavender oil, it sensed by the nose. So nose, smell should be passing towards passing through the brain, and in that they are act on the sympathetic nervous system, and then relaxation of the receptor should be occurs. In the lavender oil or the chamomile oil, it should having sedation properties, and the lavender oil it's especially the known as a excellent natural remedy for the insomnia. Then it also increase the melatonin level in the system. Then how we prepare our cream? We prepare our cream on the base by the simple method. First, we prepare a oil phase. In that, we are adding a liquid paraffin in beeswax and melt this mixture on the water vat. After that, we adding a water phase, or we prepare a aqueous phase. In that, we are adding a borax in water and dissolve in water vat. And after that, these two phases we are mixing at the same temperature by the drop wise. Then we are adding in that the preservative. And after that, our main ingredients are lavender oil and the chamomile oil by the order of this. So these are the formulation of our cream. These are the formulation of our cream. We are using beeswax, is an emulsifier, then liquid paraffin, it is a skin protectant, then borax stabilizer. We are using methyl paraben as a preservative, then water as a vehicle, and these two main ingredients. We are taking the evaluation of our cream by some physiological parameters such as a texture or their appearance, then their order, then their color, then their spreadability and the pH. Every cream having a standard pH, and the pH it should be alkaline. So we are checking our cream pH, so it is alkaline, so it is not harmful to skin, or it is useful. Then conclusion: sleep-inducing cream or our cream gives you sedative effects or relieves your stress or attention and produce calm and a cooling sensation. Actually, we are using in that chamomile oil. Chamomile oil, it is it is a mild sedative, but it's having more the cooling effect properties. So because of that, we use the combination of that products, and by the route of the aromatherapy, it gives you peaceful sleep that every insomnia patient wants, or it's normal patients or normal people's wants. Then we are using these references for our preparation of a cream, and the acceptability of our cream is more, because cream, it should be our body type, it should be, or our body should be familiar or the habitual with our cream, because we are using the various types of cream in our daily lives, such as moisturizing and the other types of cream such as cold. So we are using a cream dosage form as a we are choosing a cream dosage form because it is familiar to our body. So it is very habitual to our skin also, and it is very marketed, or it is people accepted more cream. In market, there are many types of sleep inducing products are present, such as like gels and oils, but every product having a drawback. So I think our cream reduce the one drawbacks of the another product. So this was our sleep inducing cream. We are prepared, and thank you so much for giving me opportunity to present. Thank you so much to the startup for presenting my ideas in front of you. Thank you so much.
Good presentation. You are a B.Pharm student?
Yes, ma'am.
Okay. Which year?
Final year.
Okay. Project is good. I just want to know where you want to apply the cream.
Okay. The application of cream is on the forehead. It is on the forehead. So and nose also, because in that we are using the aromatherapy. Aroma, it should be smell or sense by your nose. So it should be applied on your forehead, or it should be in a nose. So it gives you better effect or peace.
When you will apply, just before the sleep, or what is the duration?
Yes, actually, ma'am, it is a sleep inducing. So before going to bed, you have to apply the cream, and it's a very beneficial for the insomnia patient mostly, but as well as it's act as well as normal people also, or they use for the normal people also.
Normal people also you can apply.
Yes.
Right?
Yes.
So based on the constituents, you are telling that it is going to induce, right?
Yes.
So have you done any work on that? Like, have you treated on animals or something that it will tell you that it will induce the sleep?
Okay. Actually, ma'am, we are only taking the few parameters or evaluation parameters, and the animal study, it should be we are planning, because the animals, it should not present at in our college. So we are planning to taking an animal study after that, or we are going to study after that competition. So.
Oh, good.
Thank you for your presentation, ma'am.
Thank you.
I hope this is the last presentation of this day. In any case I have missed any number or any person name, you can intimate me now.
I've been waiting for a long time now.
Okay, ma'am. This is the last presentation for this day, ma'am. Thank you, everyone, for joining with us.
Hello. Can I say something?
Yes, sir, please.
I've been waiting to present for a long time.
Sorry to interrupt. Dr. Shankar Joshi, your presentation is on day 2, all C. Your presentation. We have sent the agenda also.
So I'll have to join in there.
No, there the presentations got over because you were not available. So you can present here, sir.
Okay. So
Yeah.
will I get the opportunity to present here?
Yeah, you can present here.
I'll not take long. I'll just.
Yeah,
very
no
brief.
issue, sir. You can do it. Yeah. Thank you.
Yeah.
Am I audible? Shall I go ahead with it?
Yes, sir.
What I'm going to present it here is there is a polyherbal formulation from Ayurveda, which is called as Panchatikti. It has been there in the use since last many centuries. And there are different types of Ayurvedic formulations, out of which this one is a Dhrita formulation, a cow ghee based formulation. Dhitas are generally prepared by initially extracting the different ingredients, dried ingredients, the powders, with water by decoction, usual decoction, and then it is counter extracted with the cow ghee, which is previously purified by the methods that are given in the compendia, like Ashtanga and all the rest of books that are available, the compendia that are available. Since it is a polyherbal formulation, the composition of this Dhrita, it has 5 main ingredients and there are 31 additional ingredients. The 5 main ingredients and the Kalka Dravya is what is called. They are separately treated. Kalka Dravyas are macerated in water and kept overnight, whereas the 5 ingredients are the decoction of them is synthesized, prepared first. And then these two are mixed together and boiled along with a specified amount of purified cow ghee. The Shodhan process is another process. The cow ghee is again differently prepared, and then it is used for the actual formulation. So it takes a lot of time. Then we have prepared this formulation in the laboratory itself, and we have taken a marketed preparation which is available from the Kerala Ayurveda as a comparative for this present study. And two types of studies were undertaken. Since all the ingredients, most of the 5 ingredients, then phytoconstituents are already reported in different research papers in the established journals. Those phytochemicals were screened through different tools that are available. Their structures were obtained from PDB database, 3D structures using canonical smiles and the rest of the parameters that are used. And then they were subject to docking studies for 2 targets for basically in liver disease which are involved. One is called as the regular CYP450 enzyme, which is the initial enzyme which generates the superoxide by oxidation of this oxygen, and the other one is reactive nitrogen species, which is generated by nitric oxide synthase. These 2 targets have not been explored much by the most of the researchers, and that is why I've taken it. And they do have a lot of important role in pathogenesis of liver disorders, including the chemically induced liver damage as well as virally induced liver damage. Since liver disorders are one of the leading causes of morbidity and mortality across the globe, more so with viral hepatitis, which may lead further 35% of the cases of this viral hepatitis may lead to liver hepatomas and hepatocarcinomas. So that, and in India still we are still developing, and the issue of contaminated water, infections with hepatitis virus, particularly HPV infections and HCV infections, which can lead to disastrous consequences on the health of the individual subject, especially coming from the lower socioeconomic background. So it needs a lot of exploration. This particular formulation is not directly reported in the granthas to have usefulness in the liver disorder, but the individual ingredients do have antioxidant and hepatoprotective activities that have been reported. So that again, many Vaidyas, many Ayurvedic practitioners use this formulation off-label in liver disorders also. And dramatic results have been obtained, so that I've selected this formulation. These two molecules, target molecules, were selected, and the basis for it I've already told. Different phyto ingredients that are reported from the individual constituents have been screened, and out of that 30 were again screened for Lipinski violations, and those phyto constituents which have zero violations as per Lipinski, through Swiss admit, the freeware that is available to check, that is used, and out of that only 3 have been found to have strong interactions with the targets that are selected. Both inducible nitric oxide synthase as well as this CYP450 reductase enzyme. So, and the freeware that is used for finding out binding interactions was BioVia, which is freely accessible and a very useful tool for the small time researchers or the beginners or the PG students. So that is being used for this particular docking studies. And the 3 constituents that have been found was synapic acid, apigenin, and the third was nimbinol. So synapic acid from Adathoda, whereas nimbinol is from neem, that is there, and the third one is from Chitraka. So all these you can see in here the different conformations. The upper ribbon and the helical structures represent the 3D structures, whereas the 2D structures are on the lower level, and the kinetic energies have been also recorded, and they are found to be very they show quite good binding interactions, very strong binding interactions. So the inhibition of these 2, particularly CYP2 450 reductase, will inhibit the generation of reactive oxygen species, and AYUSH, that is inducible nitric oxide, will also reduce the generation of peroxynitrile radical, which is very damaging to the liver cells and induce overall inflammation of the liver. This is also associated with hepatitis B viruses also. So that is how the docking studies are done. The 2 simple in vitro methods are used for assessment of antioxidant potential. Was this superoxide anion scavenging activity, as well as DPPH assay, which are commonly used to preliminary assess the antioxidant property. And the results, different concentrations of these, there was a difficulty in testing the formulation as such. So what we had to do was to prepare unsaponifiable fraction by saponifying the ghee formulation with alcoholic ethanolic potassium hydroxide, and then it was washed with diethyl ether many times, and the diethyl ether fraction was then dried out, and the extract was prepared. This was then dissolved in the combination of DMSO plus chloroform, and then it was made soluble so that it can be used for assessing the DPPH as well as superoxide scavenging activity, so that the reagents could mix together. So that is how we did it, and we have consistently found that the increasing concentrations of these extracts show increasing antioxidant or free radical scavenging activity, so that it is consistent with the reported claims of these different phyto ingredients, as well as the use of these this formulation Panchatattva in liver disorders is supported. That is what we have found. So that is my presentation. Thank you for organizing such a wonderful event that has went on for the last 2 days. Thank you, Ladda sir, and rest of the organizers. Looking forward to better interactions through and the outcomes out of this conference. Thank you so much, the chairperson, for this evaluation. Thank you. Hello.
Yes, thank you, sir. Thank you for your presentation. I hope Dr. Rukia ma'am is having some network issues. She will be getting back within 1 minute.
Yeah, sure.
Yes, sir.
I'm
Yes,
back.
ma'am.
I'm sorry. I had
Yeah.
a network problem.
No
It's all,
issues, ma'am. No
it's
issues.
all right, madam. Thank you so much for attending and giving me this opportunity at the last moment to present this, because I was confused about which hall to be there. I was not sure in which hall I was. So, thank you again for your kind courtesy and letting me present this small work in front of you.
So
Thank you.
you have work on in silico studies, right?
Yes, yes. As well as in
Uh,
vitro antioxidant. Both.
Okay. In silico studies you have done and in vitro antioxidant activities.
Yes, yes, both.
Okay. Okay. Good.
Yeah.
Yeah. Thank you.
Thank you so much.
Thank you, ma'am. Thank you, sir. I hope each and everyone has a great day and good experience in the Startup, a journey from innovation to entrepreneurship in Herbals conference. So here in the chat box, I have mentioned you the link to download the certificate and steps to how to download. So I can explain you in brief. First, you need to sign up into the account if you are a new user of APR. If you are an already existing user, you can log in into it and you can download the certificate. If you are a new user, sign up and create an alphanumeric password, and then you will be receiving the OTP to your registered mail ID. And please make sure that you are checking all your inboxes like spam box and junk box also for your OTP. And then you can enter into the dashboard after you log in. On the left side of the menu, you can find some menu bar and then click on the certificates and rewards. And from there, you can able to download your certificate. And here is the link for new member to register. And here is the link for existing member to register. And I have mentioned you the link for feedback also. You can give your feedback by using the link.
કે કેવી રીતે ડાઉનલોડ કરવાનું છે.
And the best presentation winners will be informed to the presenters via mail, and winner certificate will be sent to you mail within a week. And presenter certificate download link and the listener link I have mentioned you in the chat box. You can download your certificates by using the link that I have mentioned here. And you can drop your feedbacks also through the link that I have mentioned here. And I would like to thank each and every participant and the session chair, Dr. Rukia ma'am, for joining us together with this conference and making this a grand successful event. Now, I request Dr. Shefali Thakkar, Assistant Professor from Dr. D.Y. Patil Deemed to be University School of Pharmacy, for vote of thanks. Shefali ma'am, you can continue.
Okay. Good afternoon to one and all present here. It is an honor for me to thank all the dignitaries. On behalf of D.Y. Patil Deemed to be University School of Pharmacy and Association of Pharmaceutical Research, I extend my gratitude to all esteemed delegates for the conference for their presence and contribution to make this webinar a great success. I extend my gratitude to all session chairs, especially to take out their time from the busy schedule and to grace the event. Thank you for inspiring and encouraging us with your work on this special day. A special thanks to all the speakers for sharing their experience with such enlightening talks. I would like to thank all the moderators for simultaneously handling all breakout rooms. I extend my gratitude to Dr. Rakesh Somani, Principal of D.Y. Patil Deemed University School of Pharmacy, and Mr. Rudra Satpathy, CEO and founder of APR, to organize a webinar. I must thank the organizing team, the volunteers, the scientific committee for working so hard for the past few days to make this conference a successful one. I congratulate all the participants for their active participation. Thank you everyone once again for making it a great success.
Thank you, ma'am.
Thank you, ma'am.